students, welcome to Vedantu NEET English. I am Ms. Gopika, your own biology master teacher. Welcome all of you to Biology 360 day 2, and today we are going to do biology in human welfare. So I hope all of you are super duper excited. I can already see in chat few people are like, ma'am, where are you? Okay. So I am here. Okay, I am here. I hope I'm audible and visible to all of you. Can you all see me, hear me? Okay. Tell me. Yes. So the first class had a super duper response from all of your side, and obviously if you respond like this, then Gopika ma'am and Baswa sir can never stop teaching, right? So your response is everything for us. How you people are, that is how we are. So it's like your mood is directly proportional and your reaction is directly proportional to our classes. So today we are going to do biology in human welfare. So quickly smash the like button and also inform all your friends. Okay. Yes. Inform all your friends, and let me also drop a message in the Telegram channel. Okay. Yes. Very nice. Very nice. I can see. Everything is perfect, right? You can see the presentation also right behind me. If there's any issue, please let me know, okay? Wait, students, 1 second. I will just share. Yes, done. Perfect. Very nice. Very nice. Okay. Yes, so we will start. Awesome, Trisha. That's the energy we want. Okay. Yes. Okay, it's okay. Manu, people will come and go. That's how YouTube is, right? If it is good, people will come back. No matter what, people will always come back to, good classes, and people will always appreciate good teaching, right? So that, don't worry. Our students are going to stay till the end. So students, those who were not there in day 1, it's okay. Today, welcome all of you for day 2. Even if there are a lot of students who are a part of us on day 3 also, it is okay, not a problem. We just want to make sure that you people study and you are constant throughout these classes. It might go for 6 hours, it might go for 5 hours. I have tried maximum to put as much as NCERT line by line, so I do not write a lot, and also we can speed up the process, okay? So shall we start, students? I am going to start now. Biology in human welfare. So this chapter weightage is around 4 questions, okay? So this is our 2024, this is 2019. So from 2019 to 2024 weightage this is, and this chapter has a 16 marks, right? Almost 16 to 20 marks weightage is always there. So if you are aiming for 360 out of 360, there is no way that you can even skip a chapter that gives you 2 questions. Now microbes are going to give you 2 to 3 questions, and you cannot skip that chapter because that chapter has a lot of connection with your human health, biotechnology. So we are going to do everything, okay? Very nice, very nice. Lot of new students. Okay, thank you so much for joining. Do subscribe to the channel if you haven't subscribed, and first of all, like the video, okay, because the class is going to be awesome and genetics is going to come soon. Okay, don't even worry about genetics. Without genetics, how can biology go forward? So yes, let's start. First of all, health. Ma'am, we have heard lot about health, right? But in your chapter, in your NCERT, they say that Hippocrates and Ayurveda has always considered health with humor word. That means liquid or the fluid content in your body. Could be if you are, let's say, if you do not have any breathing issue, any respiratory related issues, then that means you're healthy. If you do not have any gastric related issue, any liver bile kind of issue, then you're healthy. So a doctor or an Ayurvedic doctor will check all these 3 things, that do you have gastric, do you have any issue with your liver, do you have any issue with your respiratory organ. If none of this, okay, in this all 3 spectrums you are okay, then they will be like, yes, you are fine. Okay, you are fine. Now, here, according to WHO, okay, according to who, students? WHO, World Health Organization, health is a state, okay, health is a state of physical, mental, and social well-being, So I have also put NCERT lines, a lot of it today, this time, because why? Human health is a chapter that students hate to read. Okay, students don't like to read only that chapter. Ma'am, this chapter is so long, ma'am. It's like one, novel I can't finish reading. So I have put NCERT snips so that I will read. I'll make you read it in class itself. So tomorrow you don't have to sit and cry. So World Health Organization, students. So in the first paragraphs, you will see about Ayurveda and Hippocrates, but it's not very important. But what directly question can come? What is health according to WHO? So according to WHO, health is a state of well-being, that is your physically, mentally and socially well-being. Let's say you're physically very fit, 6 packs you have, okay? You are like, ma'am, I can have more packs also. So 6 packs I have. Okay, fine. You're physically fit, but you're mentally very disturbed. You're always disturbing your neighbors. You're always making noise. You're playing DJ in the night. Are you healthy? Tell me about this person. Are you healthy? Okay, night full you're making noise. You're disturbing the neighbors. You're disturbing everyone. Okay, and you're mentally very disturbed. You do not talk to your parents well. You do not talk to your friends well. You do not talk to anyone well. You're not mentally well. Okay, let's say socially, since we are called as social animals, you're supposed to be socially good also. That means when you're going somewhere, okay, what you have to be, how you have to be, those things are also very, very important. Okay, now understand that when you are all the three, even when we study reproductive health, we talk about reproductive fitness. There might be a lot of people, they're very rich, okay, they have everything, they are well to do, but then because of the absence of that one child, okay, because of that they might not be, doing really well in life, right? So that means reproductive fitness also comes under this health. So according to WHO, your definition for health should be physical, mental and social well-being. Okay, now health can be related to 3 main categories. So students, I'm going to focus on from where your questions are going to come, because this is a Biology 360's main aim is to get you 360 out of 360 in biology, so that even if you get 120 and 120 both on chemistry and physics, you still get into a good government medical college. Okay, so I am going to stress on topics which are fetching PYQs, and I've also taken PYQs of previous year. You will also be practicing questions during the break time. Okay, during the break time. Clear? Yes. Studied in your principles of inheritance, no? Down syndromes, Klinefelter syndrome, and all the syndromes that you have studied, those are genetic disorders which are not contagious. It will not spread to someone else. So genetic disorders, or it could be even a deficiency with which a child is born, and deficiency defects which the child inherits from the parents from birth. Okay, so it could be because of a genetic disorder, or it could be an infection, a normal infection. It could be because of change in weather. It could be because of having contaminated food or water. It could be because we went to a new place and we could not get accustomed to the climate there, an infection can come. Or it could be because of our lifestyle. We are living a very serene life. We are living a very sedentary life. Like we are not moving, we are not getting up, we are only eating, and we are eating junk food and we are not getting up. Because of this also you can have a health issue. So these are the 3 main lines given in your NCERT that you can have. That means a health is getting affected by a genetic disorder which a child is born with, and it is a defect. Okay, and it could be an infection that you acquired during your lifetime because you are exposed to something, or it could be because of your sedentary lifestyle where you are consuming food which is not enough, having enough protein, fiber, carbohydrates, etc., etc., and you're not moving, you're not exercising. These 3 things could lead to health. Okay, any doubts in this? Perfect. Okay, fine, very good. Okay, understood. Now this is the first paragraph in your NCERT. Now most important thing, when we talk about health, the next thing that we have to talk about is what? Disease, right? Just 1 second, students, I'll just move this or else I can't see your chat. Are you all enjoying? If yes, keep answering. Don't wait for me to, tell you to answer. Just keep answering. Okay, just keep answering. Whatever you know, keep answering. Yes. So, types of diseases or pathogens. That means when I talk about health, I should also talk about something called as disease. And students, all these notes will be available on Vedantu NEET English Telegram and also on Biology by Gopika Telegram. You can join this Telegram for all my class links and everything, and Vedantu NEET English Telegram for the main PDF of PCB, all the 3 subjects. Okay. Now, dis-ease means what, students? Let's break this word. Dis-ease, that means you are at an uncomfortable, okay, uncomfortable situation, or you're not at ease, right? Not in ease. Now, when you get a fever, what do you feel? ma'am, I'm not able to eat properly, I'm not able to smell anything, I'm not able to feel anything, right? This is what you feel. That means what? You're in an uncomfortable situation, or you're not at ease, you're not feeling normal. This is called as a disease. Now, disease is caused by whom? Disease is caused by none other than a pathogen. What is a pathogen? Any organism, okay? Any organism who is capable of causing a disease is called as a pathogen, okay? Organism capable of causing, okay? Of causing a disease is called a pathogen. Or else, in other words, what can I call it? Very good. Otherwise, I can call it disease causing organisms. Disease causing organisms. Today, who is going to put the target? Every day, one student can take up the role. Who is going to put the target today? Target of today's class. Who's going to put? Let me see in the chat box how much have we reached. Very poor. Attendance is poor. Where did all of them go? Yes. Okay. Okay, Rani is putting. Okay, Madhumati, everyone are putting. Very good. Everyone put, no problem. Okay, no problem. You people are the ones who are going to do that, right? Oh, Dilip, that is too much, yaar. That is too much. That will not happen. Okay, chalo. Let's fix on 200 and 150. Okay, this range we will go. Yes, 362 I will make you get in biology. That is so my target. Okay, so yes, pathogen is disease causing organism that is capable of causing a disease. Now students, when we talk about disease, there are two types. What are the two types of main diseases? That is, ma'am, a disease could be something that actually spreads or something that does not spread. So we have two types: non-infectious, exact lines of your NCERT, and infectious. Now tell me some examples that all of you know. Non-infectious and infectious. Non-infectious means whatever happens, it is not going to spread. Okay, it is not will not spread. Okay, and we can also use the word non-contagious. Non-contagious. Okay, that means it is not going to spread. So that means these are the diseases which are not going to spread. Exactly. We can use the word non-infectious, infectious. We can use the word communicable, non-communicable. We can use the word contagious, non-contagiousness. So all these words are related to it. Okay. All these words are related to it. So now understand students, non-infectious means now a cancer patient is there. I am taking care of the cancer patient. There is no way cancer is going to spread. Okay. That means what? It is a non-infectious disease, but cancer is very, very fatal. But infectious means what? I have common cold. There's two other people in the, in this room. I sneeze or I touch this pen and I pass on the pen to the next person. Because of my aerosols or droplets, the infection gets spread to another person. That means what? It is infectious, ma'am. It is communicable, ma'am. It is going to spread. Okay. So here it is going to spread. It is going to spread from a infected person. Okay. From an infected person to a non-infected person. Okay. Non-infected person. Those who haven't seen day 1, definitely take some time and watch day 1 because it's very good and you get to solve a lot of questions. Okay. Now here. Non-infectious, infectious. Now here what is going to happen? Example or some of the non-infectious going to be cancer, diabetics. Okay, these are non-infectious. Infectious, so many of them you can remember. So many of them. You can write flu. Okay, flu, common cold, AIDS, Out of infectious disease, AIDS is very, very severe, no? Because it is going to be spread by fluids or by sexual contact, not by saliva or, you know, shaking hands. Okay, not by shaking hands. Okay, clear or not? Yes, reproduction unit is covered in that. Okay, students, so this is clear to all of you. So when we talk about health, we speak about diseases and we also speak about pathogens. That means disease causing people. And then disease means you're in an uncomfortable, not at ease situation. And there are two types, communicable and non-communicable, non-infectious, infectious. All the terminologies are same. Just understand that it will be spread from one person to another. In other case, it will not be spread at all. Okay? Clear or not? Yes, perfect. Okay, let's continue. Let's continue. Now, we are going to start with the first disease. So, here in this whole chapter, you are going to study some bacterial infections. Okay, you are going to study some viral. You're going to study some fungi. You're going to study helminths. You're going to study protozoans. So, these are the categories of how much you're going to study. So here first we start with the bacterial disease, that is your typhoid. So here you will have NCERT. Here I'm going to write the notes. Okay, don't worry, notes, everything is going to be included. I will just go through NCERT because sometimes what happen here and there, in case some line is missed, no, then when you read it later you will be like, oh my god, Gopika ma'am, you missed this one line. So that's why we have kept NCERT also in such situation. Now the topic is common diseases in man. First one is going to be typhoid. Students, first thing, you have to know that typhoid, every year one question has come, neither about the confirmatory test, that is the Vidal test or the Vidal test, or about the causative agent. I will show you how many questions I have only taken from the previous year about typhoid. So typhoid is very important. So let's focus on it. First thing that you have to know is the causative agent, students. Who is the causative agent? All of you in the chat box. Salmonella typhi. Salmonella typhi is the causative agent. Very good, very good. All of you have answered. Now, Salmonella typhi, okay. Typhi murium you can write, but here you can write typhi itself, it's okay, okay. Salmonella typhi. Now, mode of transmission, MOT. Mode of transmission is how, students? It can be transmitted by contaminated food, okay. By contaminated food and water. Very problematic, okay, very, very problematic. Then, when you talk about this, you should also know mode of transmission, you should know the causative agent. Then, you should know the symptoms which are related to it. So, what are the symptoms? What are the symptoms of typhoid? The first thing that you're going to have is fever, okay, you're going to have fever. So, the fever temperature is given specifically in NCERT, so you should remember this, 39 degrees Celsius to 40 degrees Celsius. So you will have fever which is at this temperature, okay, and it is going to be a sustained high fever, okay. It is not going to be like recurring. It is not like today it is coming, then 3 days later it's coming, not like that. Sustained high fever you will have for around 39 degrees to 40 degrees Celsius. Then you will have, students, extreme weakness will be there, okay, weakness, stomach pain, or you can write abdominal pain, because obviously it is spread through contaminated food and water. You should know that you are going to have abdominal, headache, okay. All these are the symptoms that are associated with it. Now, most important thing, what is that? Yes, chills will be there, headache will be there, you will have constipation, you will have abdominal pain because it has come through contaminated food. But a very important thing here, it is the confirmatory test. I don't know how many times the question has come, how can you confirm that you have typhoid? Who is going to confirm? Is the next lady going to touch and tell, oh, you have typhoid? No, it's not going to be like that. It is going to be the confirmatory test, okay. Confirmatory test is going to be Vidal test, okay. This will tell you that if you have typhoid or not. Very, very good, very good. Vidal test, okay, perfect. I love the Josh. Quickly like the video as well and share it with your friends, okay. Vidal test. So the students, this is very important. Many years PYQ this is, okay, many years PYQ this is. Now we have a beautiful story of one person. Whose story that is? Okay, whose story that is? Mary Mallon. Mary Mallon was a cook, okay. She was a cook during that time and she used to cook food. She had fever, she had symptoms, but she kept ignoring it, telling that, hey, today I have to go to work, otherwise I will not be able to earn, no? So she kept going to every house with carrying Salmonella typhi in her body, and she started contaminating food over food or food of every single person there where she used to cook. And she ended up, you know, making it so fatal that many people lost their life because of it. So her name is called as Typhoid Mary. What is her name called as? Typhoid Mary, and she was a cook, okay? Thyroid Mary. Your autocorrect is still in endocrine gland, okay? Typhoid Mary, okay? Typhoid Mary. And Typhoid Mary went on spreading the typhoid. That is why she is called a human carrier of the disease, okay? She was called the human carrier of the disease. And later she was, you know, isolated, and after that she passed away, okay? Her name is called Mary Mallon, okay, or famously known as Typhoid Mary. Typhoid Mary. Clear to all of you? This is the whole story about your typhoid, okay? This is the whole story about your typhoid. Clear? Yes. So here, students, I have underlined and highlighted the words. So when you're doing the same, you can just go through your NCERT and just look here. Look here. Salmonella typhi is a pathogenic bacterium. So this is a bacterial infection, okay, which causes typhoid fever in human beings. These pathogens generally enter the small intestine through food and water contaminated with them, and migrate to other organs with the help of blood, because blood is there, no? He'll take everywhere and go, okay? So blood takes this everywhere and go. Because of it, you will have sustained high fever, weakness will be there, stomach pain will be there, headache, loss of appetite are some of the common. Now what happens during severe situations, okay, when like Typhoid Mary also kept ignoring all the symptoms, what happens? These Salmonella typhi will make perforations. That means holes, pores in the intestine. That is very problematic, okay? That can get very fatal. So that is what they have told here. Intestinal perforation and death may occur in severe cases. Intestinal perforation means in the intestine, okay? On the intestine, they make pores, okay? Perforin is there, okay? Perforin proteins, they make pores, and that will lead to more severe cases, okay? Typhoid fever could be confirmed by viral test. Very, very important PYQ. Very important PYQ. Clear? This is clear. Typhoid is clear. So here I am writing it here. In severe cases, okay, severe cases, you have intestinal perforation. Perforation word is clear, I hope. Students, you can learn it with me in this class. You do not have to go home and study or anything. You learn it with me. Just keep answering whatever you know, okay? Just keep answering whatever you know, okay? Yes. It's not typing error. It is basically her keyboard is like, you have to study chemical coordination, okay? We are still at thyroid. Good thing, no? Keyboard starts typing thyroid. Good thing, students. It's better than typing other words, no? It's better to type some educational words. Okay, death may occur. Very good, very good. So severe cases, intestinal perforation happens. So that is one bacterial. Next bacterial disease is pneumonia, and my students know everything about pneumonia. Why? Because Griffith has done an experiment where he took Streptococcus pneumoniae, and then when we came to study restriction endonuclease, we have studied about hind 3, hind 2, Haemophilus influenzae, right? Haemophilus influenzae. So here goes our one and only pneumonia, and the causative agent. Causative agent is what, students? Haemophilus influenzae. Okay, Haemophilus influenzae and Streptococcus pneumoniae. Streptococcus pneumoniae. I've always noticed when I take this class, no, rhinovirus will get to know that, oh God, Gopika is taking class somewhere. I need to attack her. Every time I take this class, I will have common cold. Now also throat is like, shall I stop it or not? So that's like, you know, the connection that we have, the bond that we share, students. Okay, please understand. Yes, so Streptococcus pneumoniae, Haemophilus influenzae. These are the two causative agents of your pneumonia, and this is also a bacterial infection. Now what exactly happens here? In pneumonia, the biggest problem is that the air, alveoli, gets filled with fluid, okay? That is the most important. So this alveoli, so let's write MOT, okay? Mode of transmission, students, tell me that. Let's go in order itself. Mode of transmission, obviously, if it is something related to respiratory disorder, it has to be with the help of droplets or aerosols, okay? Droplets or aerosols. Yes, very good. Droplets, aerosols. Okay, that means when a person sneezes, okay, infected person sneezed, there are droplets and aerosols. Because of that, we inhale that and we get the infection. Okay, that's what they mean. Okay, MOT is mode of transmission. Then students, what is the symptoms of this? What is the symptoms of this, or where it is affecting? It is going to affect the alveoli. It is going to affect who? The alveoli. Very good, very good. That is, alveoli is going to get affected and there is going to be fluid filled. Okay, fluid filling is going to happen. Because of that, breathing issues is going to happen. That means the difficulty of breathing will get okay, difficulty in breathing will happen. This will lead to severe problems. Why? Because alveoli is the place where exchange of gases happen. If alveoli gets filled with fluid, if alveoli gets filled with mucus, that means exchange of gases also will become problematic. Yes, ma'am, exchange of gases will get problematic. On a longer run, when it gets very severe, what happens? The lips will turn grayish to bluish, nails will turn grayish to bluish. Why? Lack of oxygen in the body. Oxygen is not there. Exchange of gases is not happening. Okay. So pneumonia, students, what exactly happens? The alveoli will get filled with fluid. Okay, leading to severe problems. Now, this will also lead to all kind of respiratory, okay, respiratory problems. And in severe scenarios, okay, in severe scenarios, okay, severe cases, what happens? Lips and nails, okay, lips and nails will turn gray to bluish. Gray to bluish. See, I can just take NCERT and read, okay, but that will not help. When I write, even you will, oh, ma'am is writing, I should also read what she is writing. So, lips and nails will turn gray to bluish. Clear or not? Other symptoms are what, students? Cough will be there, headache will be there, breathing issues will be there. All this also are the symptoms of your pneumonia. Clear? Cough, headache, fever, chills, all this will be associated with these things only. Obviously, these are all bacterial infections. Okay. Now, let's see NCERT in case we miss something. So, here, look here. Again, the bacteria is Streptococcus pneumonia. All of you, please remember Griffith. Okay. God he is, okay, he found transforming principle and that's how we study the entire molecular basis of inheritance. So, Streptococcus pneumonia and Haemophilus influenzae are responsible for the disease pneumonia in humans, which infects the alveoli, which is the air-filled sac. Okay, and what happens in the alveoli, students? The alveoli gets filled with fluid, leading to severe problems in respiration. The symptoms of pneumonia are fever, chills, cough and headache. In severe cases, the lips and the fingernail turns grey to bluish. Okay, grey to bluish in color. A healthy person, how it is transmitted? By aerosols or by droplets. Okay, droplets. Even for all of that, okay, vaccines are there, like your TY21 in typhoid. Here also you have PCV13. So all that vaccines are there, but you don't have to study now. You will be studying this in your MBBS. Okay, which are the vaccines, which are the medicines. Again, further you will keep studying about this. Now, students, there are a lot other bacterial infections like your dysentery, plague, diphtheria, cholera, TB. So like that, many more bacterial infections are there. If you're interested in that, you can definitely read through all the bacterial infections. Okay, I can even send you a book for reference, so you can read through all the infections, but I have not seen many questions or anything on that basis. That's why we will stick to NCERT right now. Okay, perfectly clear. All these notes will be available, so don't take tension. Only do lecture notes. Keep underlining in NCERT or writing the causative agents, because sometimes you forget the causative agents. Okay, yes. Moving forward, that are the two bacterial diseases that you had to study. So I will just put a B here. Okay, bacteria and bacteria. Now we are going to virus. Okay, virus, that is common cold. And who is the example? I am the example. Okay, now students, common cold. Okay, common cold. Yeah, even tetanus. Okay, common cold, tetanus is usually, it happens because of you have got hurted with a rusted iron, okay, because of any rusting or something that was sharp and it has got rusted, okay, there's iron deposition on it, because of that you will get that, okay. Yeah, many bacterial infections are there for you, like if you want to study about it, lot of bacterial infections are there. I just told you generally few of them, okay, generally few of them. Now common cold, students, causative agent, I just told about this, that is called as rhinovirus. Okay, what's the causative agent? Rhinovirus. Every single time rhinovirus come, it tries to come in a new variant. This is the problem with virus. Even though this virus might look like, it's just a cold. But the problem is, virus always comes with different variants, so that our memory cells are not able to capture them every single time. Definitely immune system is going to try, cell-mediated immunity, innate immunity is going to try as much as possible with the help of skin, mucus, and also with killer NK cells, they will try. But the problem is, see, sometimes we can definitely react to all the variants, but sometimes we cannot do, okay. Yes, sometimes we cannot do. So that's why this problem is there. Rhinovirus is the causative agent. And students, mode of transmission, who doesn't know mode of transmission? Common cold, it will be spread by droplets, okay, droplets, aerosols, by touching an object, right, by sharing with an infected person, sharing utensils, with an infected person. How many of you have cold right now? Raise your hands in the chat box. Let's see if we can make a full gang out of it. Okay, yes, okay. Yes, so many strains are there, exactly, okay. Now, one good thing about cold is they only infect the nose and the respiratory tract. They do not affect the lungs or the alveoli. So that is why every time a person gets a cold also, they are like, okay, our immune system is going to fight it. There's nothing to worry. But in cases of pneumonia, that is not the case, because the direct fluid filling, mucus filling is happening in the alveoli. Here what is going to happen, your nose is going to get runny nose, blocked nose, depending on what, depending on the rhinovirus. It's like, today I want to make nose block, okay, I will do that, okay. So depending on that, it will make traffic jam or it will make everything free. That depends on the rhinovirus. But normally it does not affect the lungs, it does not affect the alveoli, which is in one way, we are lucky, okay. Yes, wow, we have lot of people, okay, lot of people with cold. I didn't know that. Okay, so yes, students, so here what is going to happen, the symptoms, okay, lot of symptoms are associated with this. First thing is sore throat. Okay, sore throat, you will have headaches, you can have that hoarse and that hardness in your voice, okay. This is even, we also tell, when the boys have maturity, like when you reach puberty and when your secondary sexual characters change, you get this hoarseness in the voice. That means very bold, very rough voice comes, so that also happens. Then you will have cough. For some people, cough not very normal, okay, headache, runny nose, okay. Sometimes slight issues of taking the breath because your nose is blocked, only that. There is no direct affecting the lungs, okay. It is not directly going to affect the lungs. Now students, other than that, how much is the incubation, like how long these people are going to stay? These people are going to stay from 3 to 7 days. That means it can go up to 1 week. It can go up to 1 week. This is the time period given to it. It could be because you are sharing anything, any material things you are sharing with an infected person, you are going to get cold, okay. Yes, exactly. Rhinovirus club, one whole gang is there for rhinovirus club. I thought it is only me, but then if you see the chat box, you'll know how many of them have cold. Okay, so rhinovirus, see students, rhinovirus represents one such group of virus which causes one of the most infectious, diseases in human, that is the common cold. They infect the nose and the respiratory passage, but not the lungs. Very, very important. They do not affect the lungs. That is why we are like, oh, it's you, fine, it's okay. Target completed. Which target completed? First one or second one? We have more targets, no? So this will not affect the lungs. This will not affect the lungs. Okay, yes. Now students, as you can see, all the symptoms we have written, like nasal congestion, discharge, sore throat, hoarseness, cold, cough, headache, tiredness, which is usually lasting for 3 to 7 days. Again, could be asked as a PYQ because it is mentioned specifically. Now, how is the transmission, ma'am? It is going to happen with the help of droplets, by sharing objects, sharing things, sharing a pen, sharing other things. This can happen. Okay, this can happen. Clear? Yes. Okay, second target we have to like, come on. Okay, now contaminated objects such as pen, book, cups, doorknobs, computer, keyboards, mouse, and cause infection in a healthy person. So infected person to a healthy person, transmission is very, very easy. Okay, transmission is very, very easy. That's why they tell when you sneeze, use a tissue paper or a handkerchief so that the aerosols are not spread. Now, most important thing, malaria. Now tell me about malaria. I think all of you should know about malaria. I like that, Abhinaya. I think I should change mine to like that. Okay, incubation period from 3 to 7 to whole life. No, whole life. Yes. Students, also tell me other virus, the diseases that are caused by virus. Now we are going to study AIDS. Okay, other than that, you have polio, you have measles, you have mumps. Lot of viral diseases also are there. If you want to study about that, that also you can study. Now tell me, what is Plasmodium vivax? We have studied about Plasmodium vivax before. When? Don't know, ma'am. Okay, this is a protozoan. So from virus, only virus, common cold is there, and then we will study AIDS separately. Okay, here you are going to study about a protozoan. Now, malaria is very, very important. Okay, what is it? Yes, here malaria causative agent is going to be Plasmodium vivax. Plasmodium vivax. But is this the only type of causative agent? Of course not. There are many other causative agents like Plasmodium malariae, Plasmodium ovale, Plasmodium falciparum. So these are the 4 types that are there which causes malaria. In that, the most malignant malaria or the most fatal malaria is caused by Malaria falciparum. This is the most fatal one. This is the most fatal one. Okay, question has come on this, so please make a note of this. Okay, falciparum. This one also is important for you to remember because in a bracket it is given in your NCERT. Even though they have not highlighted it, they have given it in a bracket, so question can come. Okay, you cannot argue telling that you did not highlight it. It is there in the textbook, you have to answer. So students, commonly the malaria caused is going to be by Plasmodium vivax, but then these varieties are also available, and in that falciparum is the most malignant one, that means the most fatal one. Okay, now Plasmodium vivax is not a very simple fellow. He's like, you know what, I cannot be like common cold and all. I am going to need 2 people to transmit or to continue my life cycle. That is, I am going to require, I am a digenetic. Okay, I need digenetic host. That means what? I need 2 hosts to carry my life cycle. One is going to be man and one is going to be female Anopheles mosquito. Female Anopheles mosquito. If these 2 people are there, I will complete my life cycle. How are you going to complete your life cycle? If you ask them, what are they going to tell? They will tell that I am going to complete my asexual phase. Okay, asexual phase of my life cycle in man. Asexual phase in man, and I am going to complete my sexual phase of the life cycle in female Anopheles mosquito. Clear or not? Easy? Yes. Okay. Very good. Yes. Awesome. Okay. Female Anopheles mosquito. So this is what malaria is telling us about. Okay. Now students, remember there is going to be sporozoites. Okay. There is going to be sporozoites. sporozoites which is going to enter the man. So this word you please remember. Now sporozoites, two things you have to remember from PYQ point of view. Sporozoites, it's going to be stored in the salivary gland. It's going to be stored in the salivary gland. But it is fertilization and the formation. Fertilization and the formation is going to happen in the gut of the mosquito. Gut of the mosquito. This is a PYQ and many students make mistake. The problem is they do not read. Okay, they do not read NCERT well. That is the reason. So please make a note of this. This could be a PYQ. And this is a PYQ, like it could be a PYQ for you as well. Okay, this is clear. Now we will go to the life cycle. Okay, I'll teach you in detail the life cycle. Come on, let's do this. So man, let's keep it in green color. Okay. Okay, this is man. Okay, this is what is going to happen in man's body. And this is what is going to happen in mosquito's body. Listen to me carefully. Don't write anything. Just listen. Okay, just listen. I will teach you the whole thing and then you can write. Okay, you can scribble whatever is required. Now, look here. These are the two body. Okay, man and mosquito. Man and mosquito. Which mosquito? Female Anopheles mosquito. Okay, and this is Plasmodium vivax we are talking about. Now, an infected mosquito. From where? What came? An infected mosquito. Infected mosquito means in the body of the mosquito, what is there? Plasmodium vivax is there. One infected mosquito bit this man. Okay. So, bit the man. Now what is going to happen? It is going to release. It is going to release sporozoites. Okay, it is going to release the sporozoites. Obviously, ma'am, in the bite, it's going to release the sporozoites. Yes, ma'am, definitely. Now, the sporozoites that it released, it is going to directly go into the liver cells. It is going to go to the liver cells. I can also call liver cells as hepatocytes. And students, Plasmodium vivax knows that I need to do all the asexual part of my lifespan in this man. So what it will do, it will go to the liver cells, that is the hepatocytes, and it will asexually keep multiplying and multiplying and multiplying and finally burst open. Okay, finally burst open the liver cells. Burst open the liver cells, and obviously if they burst open the liver cells, what is going to happen? It is going to get released into the blood. Okay, so it is going to get released into the blood. Okay, released into the blood, and in the blood, where is it going to go? It is going to go into the RBC. Okay, it is going to go into the RBC. In the RBC also, these people are going to do the same thing what they did there. That is, they are going to asexually reproduce. Okay, asexually reproduce continuously in the RBC. Burst open the RBC. Okay, important. Burst open RBC, and this is going to lead to the release of what? All of you tell me in the chat box. So students, remember here, okay, it is going to go into the RBC. Okay, after going into the RBC, it is going to multiply, burst open the RBC. Burst open the RBC, and these are going to release. Very good, very good. Who is that? Trisha Jashnavi. Okay, am I calling your name right, dear? Okay, Rani. Yes. Meghana. Very good. Kubera. What are all names here? Very nice. Avengers. Yes, good job, good job. Pratibha, very good. Okay, all of you, good job. Now, it is going to do what? Ma'am, it is going to release something called as an hemozoin. So, burst open the RBC, releasing, I'm writing it here, releasing Zoin. Now don't tell me, ma'am, it's out of NCERT. It is there in NCERT. If you read the paragraph, you will find this word. Okay, release hemozoin. Now it is believed that this hemozoin also helps, because students here, even though they're bursting open the RBC, remember that the gametes are already formed here. Okay, gamete formation has happened here. Male and female gamete formation has happened, but we cannot do fertilization. In our body, fertilization will not happen. Male and female gamete fertilization will not happen in our body. So the mosquito is very brilliant, no? But they tell that this hemozoin also helps with the gamete formation. But main thing of hemozoin is it is going to be the reason for having recurring fever every 3 to 4 days. Okay, this is the reason for having recurring fever, okay, every 3 to 4 days. Clear to all of you? Is the life cycle clear in man? Thumbs up if you understood. Thumbs up if you understood. Okay, so here, okay, just tell me if you understood. Remember, in this, your male gamete will be formed, okay, and also the female gamete will be formed, okay. I'm just putting some two shapes, male and female gamete will be formed. Yes, 3 to 4 days recurring fever, very good. Now that gamete, okay, this gamete will be taken up, okay, will be taken up as a blood meal, okay, by a mosquito. It need not be the same mosquito. Maybe this mosquito bit and it went and we killed the mosquito somewhere else, okay. It's not necessary that the same mosquito comes back. It could be another mosquito. Now in the other mosquito also they will take up the gamete, okay. So in the blood meal, the gametes will be taken up. After the gametes will be taken, what will it happen? Okay. Yeah, I think chats are slow. Yes. So after the gametes are taken up, what will happen, students? Definitely, ma'am, fertilization will happen. Then, students, fertilization will happen. Fertilization will happen. That means the male plus the female gametes are going to come together, and this fertilization will lead to the formation of zygote, and that zygote will mature and develop to form your one and only sporozoites. Okay, sporozoites. Clear to all of you? Sporozoites. Now this sporozoites, this whole fertilization is happening, in the gut of the mosquito. Okay, whole fertilization, everything is happening in the gut of the mosquito. Remember this, in the gut, okay, in the gut. But the sporozoites, after formation, it is going to be stored. Okay, it's going to be stored in the salivary gland. Okay, it is going to be stored in the salivary gland. And then the cycle will continue. Okay, then the cycle will continue. Understood? Okay, clear or not? So, yes, DDLC, here also I am teaching the same thing, no? So it's the same. Okay, come here, look here. It will be stored in the salivary gland. Again, it is going to bite another person, and from the salivary gland, the sporozoites will be released into the person, okay, healthy person. So this is how the cycle is going to continue. So this is what exactly happens with your Plasmodium vivax, okay. Any doubt in this? Please let me know in the chats if any doubt you have. So remember, asexual phase, okay, asexual, okay, I will do this job, okay, this is going to be the asexual phase, okay. Asexual phase will happen here. This is going to be the sexual phase. sexual phase. Female Anopheles mosquito. Okay, yes, quick recap. So what happens, students? One mosquito came which already had Plasmodium vivax and that has infected or bit a man. Okay, this has happened during a bite. Okay, mosquito bite. Now, as the mosquito bites, you know that in the salivary gland, the sporozoites is already, okay, sporozoites is already ready, present, telling that, yeah, I want to bite you. Okay, so release the sporozoites into the liver cells, and the liver cells can also be called as hepatocytes. In the hepatocytes also, asexually, okay, asexually, it is continuously multiplying, and it will burst open the liver cells. So, students, remember, these are all cells. Right, these are all cells like this. As the multiplication happens, it is normal that if you have more than what should be there, it is going to burst open. Right, it is going to burst open. So here the liver cells will burst open. Liver cells will burst open, and that will lead to release of these people into the bloodstream. Release of these people into the bloodstream. Now, once it is released into the bloodstream, it is going to go into the RBC. In the RBC also, they will continuously asexually reproduce, burst open the RBC, and this bursting open of the RBC will lead to the release of hemozoin. This hemozoin will cause recurring fevers, show you the first ever symptom that you have malaria. By the time the symptom is shown, you know that one cycle has already happened. Okay, schizogamy, all that words you don't have to remember. Okay, you don't have to remember because all those words are not there in your NCERT, and most of the students don't study it like that also. So this is fine. Okay, this is fine. You can stick to this. Burst open the RBC, and this hemozoin also helps with gamete formation. Slightly, I'm not telling like full. Okay, gametes will be formed in the RBC itself because the Plasmodium vivax asexual phase will happen in the man. Now that hemozoin will lead to recurring fever in 3 to 4 days. Now, students, what is going to happen? The gamete will be collected by a blood meal, okay, by the mosquito. Now, in the blood meal, gametes are there. We know that in our body, fertilization will not happen. So, what will happen? Fertilization will happen in the mosquito's body. Fertilization will happen in the mosquito's body. Write the notes, write, Dilip, write lecture notes. Keep writing, okay? Students, we use the word schizogamy, we use the word, you know, gamy, enterogamy and all of that, where the multiplication will happen, okay, like this bursting open will happen. No, we can use those words, okay. Some people use the words, but it's not necessary, okay. Yes, fertilization. Now male and female gamete fertilization happen, lead to the formation of zygotes. In this, that zygote develops into your sporozoites. Now this entire thing happened in the gut of the mosquito, but the sporozoites will be stored in the salivary gland. Once it is stored in the salivary gland, it will be ready for the next. It will go and bite the next person and it will infect the healthy person. Clear? Yes, this is your entire malarial life cycle for you. Okay, malarial life cycle. Clear? Okay, perfect. Now moving forward, this is what is given in your NCERT, and I will just go through that so that you people know. Okay, so you people know. Look here. So when a mosquito bites another human, the sporozoites will be injected with the bite. That will go directly into the liver. Okay, through the blood it will reach the liver. Then the parasite will reproduce asexually in the liver cells, bursting the cells and releasing into the blood. Parasites reproduce asexually in the red blood cells, bursting the red blood cells and causing the cycles of fever and chills. That is because of the release of hemozoin. Now in the blood itself, you can see the sexual stages, that is the gametocytes, develop in the red blood cells, but fertilization does not happen. Gamete formation happens, but fertilization does not happen. Now the gametocytes will be taken up by a blood meal by another female mosquito, and that will lead to the fertilization and the development, which will happen in the gut of the mosquito. From there, it will lead to the formation of sporozoites, which will be stored in the salivary gland. Again, mature ineffective sporozoites escape from the gut and migrate towards the salivary gland, and again the cycle will continue. Done. Why, Sangeeta? You don't want to study human health. Microbes you will directly study. Microbes will happen after I finish my class. Okay, after I finish my class. Yes, perfect. Clearly visible. Okay, yes. Okay, now next is amoebiasis. Okay, next is amoebiasis. Now malaria is done and dusted. Now amoebiasis, all very simple, students. Amoebiasis, ascariasis, all just causative agent and mode of transmission. Okay, now causative agent is going to be entamoeba. Okay, it is going to be entamoeba histolytica. Entamoeba histolytica. Now this entamoeba histolytica, students, it's very important for you to know that this comes only because, okay, this comes only because of what? Yes, depending on our cooperation. Very good. Okay, entamoeba histolytica is totally going to, that means this disease, amoebic dysentery, is going to spread when there is fecal matter. Okay, there is fecal matter, and on the fecal matter, if a housefly, okay, if a housefly, this looks like a honeybee, but it's okay. If a housefly sits on the fecal matter, and then the housefly comes all the way flying. Okay, let's say we have kept food. Okay, we have kept food. Okay, something we have kept. Now this housefly has come all the way from there, and it has come all the way here near our food, and it is planning to sit on the food. Okay, sit on the food. Now what is going to happen? These people have carried the fecal matter, and in this fecal matter there could be chances that there are eggs of the entamoeba histolytica. So this is a fecal oro route. That means because of the fecal matter, it has gone through our oral route. No, the fecal matter has entered our oral route by contamination with the help of a carrier, which is called the housefly. So this fellow acts as a carrier. Now students, remember, female Anopheles mosquito, I forgot to write that actually. Female Anopheles mosquito is a vector. Vector means vehicle. Now this vehicle can be a part of the life cycle. Not necessary, but it can be a part of the life cycle. Okay, it plays more role. But when you come to carrier, they are just going to carry the infection. Okay, they are going to just carry the infection. Clear? Home fly does not sound nice, no? Now, if I change your name and call, how will you feel? So, let's stick on to housefly wordle, okay? Now, students, because this is a fecal oro matter, you are going to have abdominal pain, extreme pain will be there. You can also have, you know, blood and mucus. All that is possible. So, look here. Entamoeba histolytica, again a protozoan parasite in the large intestine of human, which causes amoebiasis or amoebic dysentery. Symptom of this disease include constipation will be there, abdominal pain, cramps, stools. Cramps means again extreme pain. Okay, stools with excess mucus and blood clots. So the fecal matter can have mucus as well as your blood clots. Clear? Symptoms are clear? So who is the problem here? You are the problem for not closing the food. You can't expect housefly to be more responsible. No, you should be more responsible. Yes? 𝐂𝐡𝐞𝐤. This 𝐂𝐡 only caused all the problems. So, constipation, stools with mucus and blood, extreme abdominal pain. Okay, I'm writing few points here. Since you already have it in the notes, you can directly see it. Now look here, students. Houseflies act as mechanical carriers to serve to transmit the parasite from the fecus to the infected person, to the food, and then to the food products, okay, thereby contaminating them. Drinking water and food contaminated by fecal matter are the main source of problem, main source of infection. That means if you have kept some food open and you know that in and around your surrounding there are a lot of garbage dumps and people also, you know, excreta, they pass on the excreta there, then you should definitely make sure to first of all clean that place by informing the BBMP, or that is here, okay, or you should make sure to close all the food and water so that the houseflies does not come, because housefly does not know, students. How will housefly know that if I am sitting on food or not? You should be the one who should know. Clear? Clear? Yes? Okay. So, Yes, perfect. So that is Entamoeba histolytica. Then we have Aspariasis, which we have studied in our Ascalminthus, right? Ascalminthus. So Aspariasis is caused by Ascaris lumbricoid. Okay, causative agent is going to be Ascaris lumbricoids. Now this is also again a fecal oral matter. What is exactly going to happen, I'll tell you. Let's say there is an empty land. Okay, there's an empty land. Okay. Yes, very disappointing. Those who haven't liked the video, quickly like the video. Don't disappoint our class. Okay, they are putting, the targets. Okay, now listen here. Very important for you to remember that let's say there's an empty land. Okay, there's an empty land and someone has passed the excreta there. Now that land later will be used for farming. Now in the excreta that this person has passed out, there could be eggs of Ascaris. Ascaris again will be in the intestine. There could be eggs of Ascaris in the fecal matter. That will dissolve into the soil, and when the person does a farming or cultivation of something, it will be absorbed by the plants. Now this plant, this vegetable or whatever will come to the commercial market and we will buy it. So do you understand the route map of how this Ascaris will reach us? We might not be directly connected to the person who has this Ascaris lumbricoid or Ascariasis, but it can be passed on with the help of contamination of soil, water, all of this. Okay, soil, water, vegetables, all these medium, this can reach to us. So exactly what is happening here, students? This is again a Helminth. Okay. So, Ascaris lumbricoids, this person, the common roundworm, this is going to be passed on again with a fecal oro route or oral route. Okay, oral route, and this is again from the fecal matter to the person. What are the symptoms? Symptoms, you can have internal bleeding. Okay, this is little dangerous. Okay, you will have again abdominal pain. Abdominal pain. Anything with contaminated food and water, it is very normal, no, that you will first have stomach pain. Okay, stomach pain. Then you can have anemia. These are some of the common, symptoms. Now, mode of transmission, as you people know, it is again contaminated food, water, vegetables, okay, etc. This happens a lot in rural areas. Okay, rural areas. What happened? Okay. So here, look here. Now, Ascaris, the common roundworm, and Wucheria, the filarial worm, are some of the helminths which are known to be pathogenic to man. Ascaris, an intestinal parasite, causes Ascariasis. Symptoms of these disease includes internal bleeding, muscular pain, fever, anemia, blockage of the intestinal passage. The eggs of the parasite are excreted along with the fecus of the infected person, which contaminates soil, water, vegetables, plants, etc. A healthy person acquires this by contamination of water and vegetables. Clear or not? Yes. So what we have to do? We have to wash our vegetables. We have to, keep it in salt water and turmeric water for a very long time. Brush the vegetables outer layer, then only consume. Nowadays, I see a lot of people just walking by, they'll see cucumber. They'll be like, oh, give 10 rupees cucumber. They will eat like that only and go. And I'm like, and when we ask them, let's say we ask some of them, like, why don't you wash it? They're like, purest form. Yes, it is definitely the purest form, because there could be Ascaris on us. There could be, there could be anything on that. There could be Ascaris egg. There could be Amoebiasis, that is Entamoeba histolytica. There could be anything, because who knows where it was cultivated? Who knows from where did they pluck it? Did they wash it? Did they bring it? No idea. He's just spraying from morning so that it looks very glossy and shiny like my hair. Okay, that they'll put serum on my hair. After blow drying my hair, they'll put serum telling that, ma'am, now your hair will look very shiny. This guy from morning, he's putting perfume, that is spray on the cucumber, so the person who looks, oh, so fresh. I do not get this in market. I should buy it right now. I should eat it right now because it looks fresh. So it's all polished, students. Nowadays, you see apple also, no? Don't you feel apple looks like it's been coated with some lipstick and blush? So red. so red, so shiny that I am standing here, apple is shining from there. Okay, so I'm like, what is all this? Everything is color. Everything is color, wax. On the apple, if you see the apple and if you scrub it with a knife, no, you will start seeing white powders. And I read it somewhere and I think I saw a video or something. They told me it is wax. So that's why whatever looks shiny, no, don't take and eat it immediately thinking that, oh, it is very fresh. Fresh. How they made it fresh, God only knows. Okay, God only knows. Okay, clear? Yes. So this is what is happening in your Ascaris lumbricoid. So next time if you see someone telling that this is fresh, eat, please ask them to wash it and eat. Okay. Now next is elephantiasis. This is again a helminth only. This is caused by causative agent. Causative agent is Wuchereria. Wuchereria bancrofti. Okay, so Wuchereria bancrofti or Malai, yes, even I saw that, yes, yes. Now what is going to happen? I just told hair because they spray so many things on the hair so that it looks shiny when you go outside, no? That's how they're doing for the vegetables nowadays. Yes, so look here, Wuchereria bancrofti or Wuchereria Malai. Now this two, any one of the variants, no, in that bancrofti we usually tell that causes elephantiasis, a filarial worm. Filariasis also we can tell because it's caused by that. Now students, what exactly happens here is the swelling of the lymphatic vessels happen. So as you can see, the lymphatic vessels of the lower limbs get swollen, and elephantiasis means the entire leg region, no? So the leg would look something like this, okay? Something like this. This much swelling will happen, students, and this swelling will also, okay, this swelling will also go into the genital area. So it is very dangerous, and but what is most important is the mode of transmission. Mode of transmission is by Culex mosquito. Female Culex mosquito is the reason for causing this elephantiasis or filariasis, and this happens in gross deformities in the genital area. Okay, clear? Yes or no? Yes, very good. Swelling of the lymphatic vessels in the lower limbs, which can also spread to the genital area, and the entire body will have gross deformities. That means obviously equal, you know, the proportions will not be equal, no? So the proportions are going to be different. Clear? Now the pathogens are transmitted to a healthy person by the bite of a female mosquito vector. Female mosquito is going to be the Culex mosquito. Now dengue and chikungunya also, two lines are given in your NCERT. Aedes mosquito. So three mosquitoes you have studied: Anopheles mosquito, malaria; Culex mosquito, elephantiasis; Aedes mosquito, dengue and chikungunya. Dengue and chikungunya, if you want to study, just type dengue and chikungunya video by Gopika ma'am. Video will come for you. I have taught in detail, but last time question did not come, so this year we are thinking, okay, should we teach or not? Baswa sir and I am not siblings. How do you even think that we both are siblings? Okay, we are colleagues and we were college friends. That's all. Okay, all unnecessary questions you people have. Do you know anything about Malai and bancrofti? Do you know they are siblings or not? No, ma'am, I don't care who those two people are. I'm bothered about how you are going to, okay, are you having any siblings? Now, many fungi. So students, we have done bacterial, typhoid and pneumonia. Virus, we have common cold. Again, many others are there, but as of now in your NCERT, then we have studied protozoan, plasmodium vivax. Then we have studied, again, entamoeba histolytica. Then we studied ascaris as well as your Wuchereria bancrofti, which are helminths. Okay, so these are the ones that we have studied. Now we have fungi. Fungi, we have only one disease that is mentioned to us is ringworm. What is it? Ringworm. Now students, ringworm is mainly caused because you should know that fungi loves places which has a lot of moisture content. It loves moisture. Now moisture in our body could be sweat. Okay, could be sweat. That means let's say we are working out every day and we are not washing our body after workout. We are wearing the same clothes over and over again. Okay. If this is going to continue, what is going to happen? The place where the moisture content is there, the fungi can grow. Now, which genre of fungi can go? Trichophytum, Microsporum. See, look here, Microsporum, Trichophyton and Epidermophyton. These are the 3 types that you have to study. Okay, 3 types that you have to study. So ringworm fungi. Now ringworm, how it is affected, students? How does it look like? It looks like white lesions. Okay, the area which has affected, no, the area where the affection will have, it will look like this. It will have this white lesion. Okay, now same like how we have studied in plants and all, no, the white spot, like that only, white lesions. This can come in your skin, okay, on your skin, on your nail, okay, on your hair. These 3 regions very commonly these can affect. That's why we get dandruff also, no, very oftenly. That is because of that. Okay, so that is why, because the hair, the scalp, okay, it's very difficult to keep it clean every time. So now oil is there. So when you're oiling it and you're going outside, what happened? Dust gets accumulated. Now fungi loves to live in a place where there's a lot of moisture, wetness, okay, dirt is there. These places fungi will grow. So these will grow in areas like your so near your groins here, these regions, okay, right behind your neck, okay, they can grow there. Some people, they also get it between their, you know, armpits. Some people get elbow fold here. They can get wherever moisture can be locked or where the wetness can be stayed for a long time, and places where there is no enough sunlight. Okay, now every time we are not tying our hair like this, no, so here what happens, the wetness will be there or the moisture content will be there. In these regions also the fungi allergy can happen, and between your nails, okay, between your fingers also, these places you can get allergies. Is this clear? This is most important for you: Microsporum, Trichophyton and Epidermophyton. These 3 names are very, very important for you to learn. Clear? I hope no doubts. No doubts? Yes. All of you drink some water? Yes. Madhubanti is reminding me, ma'am, please drink water. Yes. Did you all mark the attendance? Done. All of you. Why can't I see it? Yes, very good. Okay, let's do a question then. First question on all of you. Last time I saw many of them got 40 out of 40. I was so happy. Yes, match list 1 and list 2. So students, 4 marks for one question. 1 mistake, -5 marks. Okay, -5. Ringworm, filariasis, malaria, pneumonia. This is like a revision so that you do the question now itself. Haemophilus influenzae, Trichophyton, Wuchereria bancrofti and Plasmodium vivax. Choose the correct answer and from the given option. Yes, tell me quickly the answer. Okay, very good, very good. Okay, let me see. I have not solved it. So yes, ringworm is going to be 2, filariasis, Wuchereria, malaria, Plasmodium vivax, pneumonia, Haemophilus influenzae. So what is the option? D1, okay, C4, B3 and A2. Very good. Option number D. Option number D. Ready? Yes. So +4. You can put +4, and when you are writing the comments, you will put GM, okay, GM and your total marks. That means how much you got in my class. I am seeing all your comments. Okay, I am seeing all your comments. So keep. Yes. Next question. The infectious stage of Plasmodium that enters the human body is female gametocytes, male gametocytes, trophozoites and sporozoites. Okay, it's a NEET 2020 question. This was a NEET 2023 question. Okay, this is also +4 marks. Please tell me the answer. Very good. +4. Everyone should get +4. Okay. Yes, those who made mistake. Yes, good job, good job, good job. Sporozoites. This is the stage that enters the human body, right? It is not the male gametocytes, not the female gametocytes, but after the zygote has got matured, that only enters our body. So +4. So that means you are right now at +8. Okay, +8. As and when I am teaching, you will get questions. Okay. Yes, next question. Match the following diseases with the causative organism and select the correct option. Typhoid, pneumonia, filariasis, malaria. Same question, huh? No, different question. Yes, this is NEET 2020 question. So you can see, this is how much questions will come. You don't have to break your head. So just understand, remember. Okay, tell me the answer for this. Let me also solve. If you can't see, please let me know. Okay, D also is there. A, B, C, D. Today is a D day. Yeah. Correct. Very good. That is typhoid, salmonella. One was enough to solve, but let's go further. Pneumonia, we have Haemophilus influenzae, filariasis, wuchereria, malaria, plasmodium. Okay. So it is going to be option number D. This means you have got plus 12 in your wallet right now. Okay. Plus 12 in your wallet right now. Okay. Shall we continue immunity? Okay. Immunity. Okay, very important topic, students. From now on, I want full 100%, 200% concentration. Okay, and as and when you feel the class is going nice, like the video as well. Okay, now immunity. So we have completed common diseases in man. We went through NCRT and we also did the causative agent, mode of transmission and the symptoms. Okay, now next is immunity. Here you need a lot of understanding. So I'm going to help you understand every single thing. What is immunity? Now, one person has come and hit me. Okay, one person is coming to hit me. I will definitely try to resist that person, right? When your siblings fight, what do? When she or he is coming to hit you, you try to resist it. Same way, whenever an antigen is coming, whenever a foreign particle is coming, our body also has the ability to fight it out. That is called immunity. So, immunity simply means the ability of our body to fight a foreign particle. Okay, a foreign particle. This is immunity for us. That means if something is coming, okay, the ability of my body to fight it. And if it is a disease causing organism, I call it a pathogen. So the foreign particle can also be called a pathogen. It could be a pathogen, right? So this is what immunity means for us. Okay? Yes. Now, in our life, we are born with some immunity because when as soon as a child is born, okay, the child is already born with some immunity. But as the child is going to grow, some immunity will be acquired during the lifetime. Now, none of us expected that COVID would happen. Right, COVID is an infectious disease. It is going to spread from one person to another. None of us knew that COVID is going to happen when we are at the age of 25, 26, 27. No one knew. Now, what exactly happened, students? Remember that during the lifetime, I got a vaccination to fight COVID. Now, in our body, there will be two types of immunity. In our body, what will be there? There will be two types. Oh, 1 second. Yeah, there will be two types of immunity. The one that we are born with, which is called as innate immunity, and the one that we will acquire later in our lifetime. Acquired immunity. So these are the two immunities, innate immunity and acquired immunity. Correct? These are the two immunities. Now, I am telling that innate immunity with birth, acquired during our lifetime. Important, during our lifetime we acquire this. Clear? This is clear what is immunity and what are the two types of immunity to all of you. Any doubts? Okay, very good. Now remember, immunity does not just mean that they are going to fight all the time. No, they have protection. So these people are going to mainly act as barriers. These people are mainly going to act as barriers. They are also going to do the job of protection, and not only protection, students, most important thing that these people do is they hold a memory. They hold a memory of the person who came to attack. Now let's say one person came to attack me. Okay, one person came to attack me. Now obviously I'm going to remember that person. Am I going to forget? Or telling that, oh, he hit me he slapped me, so I'm forgetting him. No, the next time I see someone similar to that also I will always remember. I will always remember, right? So here remember that these people are going to act like a barrier. They are going to do the job of protection, and they are also going to hold a memory. This is important. Now, when I go further, the first topic that I need to learn is innate immunity. Students, the chapter is big, but let's make it as interesting as possible and also learn everything today itself in the class. Okay, today itself in the class. Yes, defense mechanism. Very good. Now, innate immunity. When we talk about, there is mainly 4 types of innate immunity. First type is called as physical barrier. So these are the barriers which you are born with. Okay, first one is called as the physical barrier. Now ma'am, what is exactly physical barrier? Now students, in our body we have one and only, okay, one and only skin. Okay, one and only skin. Entire body is lined by skin. And that skin is going to protect us from most of the dust particles, most of the foreign particles, right? But they are not specific. They are not like, oh, today this dust came, today cement came, next time silicas is coming, asbestos is coming. It's not that kind. They are not specific, right? So here, when I talk about physical barrier, remember the first physical barrier is skin that lines, okay, that lines our entire body. Clear? In animal tissues we have studied epithelium, lining of the entire body. No? Second most important physical barrier is mucus. Okay? Mucus lining. All of you think and tell me, okay, all of you think and tell me, this is a person. This is a person. Okay? Now in this person, okay, in this person, I am telling that there are 3 main areas or 3 main pathways through which any foreign particle can enter. First one, what it is going to be? Respiratory system. Through my nostrils, I will inhale something and I can get a problem. Next is digestive system. Okay, I take in something through my mouth and digestive system. Third thing, urinogenital system. Right, we get UTIs and all of that. So in this child, 3 ways, okay, in 3 ways the pathogen can enter. One is through the nostrils, one is through the mouth, and one is through the urinogenital tract. So in all these 3 regions, there is going to be mucus lining to protect us from such kind of entries. Okay, from such kind of entries. Clear or not? So mucus lining that is present in the digestive system. Okay, digestive system, in the respiratory system, and in the urinogenital system. So mucus lining is present in all these 3 systems, so they can help us with, yes, very good, they can help us with entry of, okay, entry of unnecessary, they can help and also prevent unnecessary things from entering our body. Okay, so mucus lining, epithelium will be present on all these 3 layers. Next most important barrier is going to be what? Physiological barrier. Physiological barrier. Now physiological barrier, students, what are the barriers? Let's start with all of you opening your book and starting to cry. Okay, yes, very good, target achieved, very nice. Okay, first thing, what is going to happen? You start crying. That is, you have lysozymes in your tears. Okay, correct? Okay, when we go outside and come back home, students, what do all the girls observe? They go touch their face and they'll be like, oh, I have oily skin. That means what? Secretion of sweat and oil from your sweat glands and sebaceous glands. Sweat glands and sebaceous glands, which are also one way to excrete out or throw out the waste materials from our body, unnecessary things. Then we eat food. Once we eat food, what happens? When it goes to our stomach, HCl will be released. Okay, HCl in stomach. These are the These are the main physiological barriers. So whatever is present in our body, it is made in a form so that unnecessary substance can go out. Okay, unnecessary substance can go out. Clear? So here you can see lysozyme, sweat, oil and HCl in the stomach. Like that we have ear wax. Okay, all these are going to help with removing unnecessary things. Okay, removing unnecessary things from our body. Now, this is clear. Physiological barriers and physical barriers, exact lines of your NCERT, just study these 3 words. Okay, these 3 words. Clear? That is like that. Some people will be there, no, I am leaving, so I will just not like also. So, all that. Okay. Yes, cerumen, that is your ear wax. Done. Clear? Now, next 2 people. Okay, I think here. 1 second, students. Next 2 barriers that we have is what? Cellular barriers and cytokine barriers. Are you all understanding? Are you all enjoying? Let's put some fire emojis in the chat box. Come on, all of you. Come on, no sleeping, no giving up. Okay, we are going to step into new year in another 3-4 days. 4 days. So we cannot be like, okay, let me do it later, let me watch replay. Why? You have the data, your parents have provided you with a phone, laptop. What is the problem? Just sit. I am the one talking, I am the one moving, I am the one shouting. You just have to sit and listen and write lecture notes, right? Okay? Yes. Now, look here. Next is going to be innate immunity. The next is going to be cellular barriers. Cellular barriers. Now, students, cellular barriers, as you can, from the terminology itself, you can remember, no, cells, something to do with cells. Hi, DJV. Okay, hi. Okay, we are almost in the middle of the class. You want me to tell hi. Now, cellular barriers, as you can tell, some cells which are going to fight. Okay, if there is, some antigen in the blood, they will go and fight. If there is some other bacteria that is going to enter, they are going to do phagocytosis. They are present everywhere. They are also present in your cartilage, right? Cartilage, when you studied, all if you remember, we have macrophages. These people are full-time active, okay? So, cellular barriers, first you have PMNL, which full form can be asked for you, that is polymorphonuclear leukocyte. Okay? Neutrophils. Okay, body fluids in circulation we have studied, no? PMNL. Other than PMNL, who is there? You have monocytes, you have macrophages, and these people are all going to help in phagocytosis. They will engulf the problematic fellow. They will just swallow. They will engulf the problematic fellow. And the most important cell, which are called as natural killer cells. Natural killer cells or NK cells. What these NK cells are going to do, students? They are also going to do perforations. They are going to make holes or pores in the antigen or the problematic fellow. They are going to make foreign particles. They are going to make pores, and because of it, everything will enter and swell and break open. Okay, break open. Clear or not? So cellular barriers are PMNL, polymorphonuclear leukocytes, neutrophils, monocytes, macrophages, and natural killer cells who are going to do the job of cellular barriers. Next most important person is going to be cytokine barriers, and these people are the most selfless people that you will see. Okay, in humans we do not have this tendency. In humans the tendency is that in my home current went. Okay, in my home current went, I'll go see in next home if current has gone. If next home current has gone, then I'm at peace. But that is not what is going to happen here. Here what is going to happen, students? Cytokine barriers. Let's take some cells, okay? They're all close to each other. They're all neighboring cells, okay? Neighboring cells. Let's call them 1, 2, 3, 4, 5. Now what has happened? the first cell got affected by a virus. Virus has affected the first cell. The first cell already realized that, okay, my life is going to come to an end. It is over for me now. Now I cannot do anything. I cannot do anything, but I can do one thing. I can make the other cells aware that I got a viral infection and release an antiviral protein, or tell them that you please release an antiviral protein so you can get saved from this virus. So this infected cell will release interferons. What they will release? Interferons, which will signal, okay, which will signal the rest of the cells, telling that I have been affected, okay, I have been affected by a virus. You please be careful, okay. Infected cell will release interferon which is going to signal another cell telling that I am in problem, you please take care, okay, you please take care. They will tell this and they will signal all the cells, okay, they will signal all the other cells. Signal the cells to release what, students? To release antiviral protein so that they can protect themselves from the virus, okay, they can protect themselves from the clear or not? Cytokine barriers work on this principle of interferons. Do we humans do that? I am in danger. Do I inform everyone else telling that I am in danger, please go and escape? No, nothing like that. Have you seen all this videos on this YouTube and Instagram? One fellow will be sitting on the stone, okay, taking photo with knowing that there is high tide, okay. No one will tell him to please get down from that stone and come and sit in the corner. When he is going in the water, everyone will start shouting, okay, they'll keep shouting, okay. Yes, yes, tell me, okay, clear or not? Yes, antiviral protein. So the virus will infect the cells and the first cell will know that I cannot survive, so I will immediately release interferons. That is going to signal the other cells and that is going to help them release antiviral protein so they can save themselves, okay. They save themselves, clear? That is going to happen. Now next immunity, students, acquired immunity. As you can know, the name itself talks about what? Acquired immunity means the immunity that you gained during your lifetime, okay, you gained during our lifetime. Now there's something very important. I told you that innate immunity is not specific. It does not care, okay, it does not care if it is a bacteria, if it's a helminth, if it's a protozoan. It is going to affect everything and it is going to treat everyone the same, okay, it's going to treat everyone the same, no partiality. But acquired immunity is not like that. Acquired immunity is like, okay, I know whom I should do and how I should treat them. Specificity. Specificity. That means if it is a bacteria, I know what to do. If it is a virus, I know what to do. If it's a fungi, I know what to do. Specificity is important. That means what? You also know how you should treat your best friend and how you should treat your so-called friend, how you should treat your fake friend. For some people, you will send long paragraphs. For some people, you will send HMM. For some people, you will put this emoji. That means what? You're very specific how you should treat each of you, right? Now, if some student is not treating me right, I also know how to treat that student, no? That means I'll tell them, be respectful. Like how Nandu is telling that she wants to sleep in the class now and she's sending sleeping emoji. So what I will do, I'll remove her from the class so she can sleep peacefully, right? So we should exactly behave that acquired immunity, no. Specificity is important. Specificity is important and acquired immunity is like I am very specific. Next most important thing is, students, acquired immunity is diverse. It shows a lot of diversity. Ma'am, what is diversity? Let's say many variants have come in our body. Many variants have come in our body. Our body, this acquired immunity, knows the diversity. Knows the diversity that, okay, this has come. This is also coming. They will know everything. That diversity, they will be able to understand. Clear or not? These are the, okay, these are the important words. Yes. I can remove you so that you can sleep peacefully. Very important thing which innate immunity cannot do, discrimination. Discriminate self cells with non-self cells. These are lines of NCERT only. I am not making it up. It's all in your NCERT. Okay. Now what is going to happen? Okay. Look here. Discriminate self cells with non-self cells. Self cells means what? I know who is my family. I know who is not my family. I know how to treat my family and how to treat outsiders. Same way, body also should know who is my own cells and who is the cell that has come from outside and it is pretending that I am yours. But they know to discriminate. Acquired immunity know to discriminate self cells and non-self cells. When our body starts harming the self cells, that is when we get autoimmune disorder. Clear? So that also these people know. And students, see, specificity, diversity, discriminate self cells and non-self cells, and one of the most important thing is they have memory. They hold memory. They hold memory means what? Ma'am, they remember that okay, COVID has come once for me. COVID has come once for me and I have taken vaccination for it. Next time COVID is going to come, my body is going to react. That my body knows. How? Because my body has memory cells. Is this clear to all of you? Yes or no? So, these are the 4 characteristics: specificity, diversity, discriminate self cells from non-self cells, and memory. Clear? Yes. Now, when you talk, okay, when you talk about immunity as a whole, you should remember about some organs, okay, organs which are called as lymphoid organs. Lymphoid organs. Lymphoid organs. Now, why you should know about lymphoid organs? Because you are mainly focusing on 2 people in this whole immunity topic. Who are they? Lymphocytes. Who are they? Lymphocytes. Which are the 2 lymphocytes that you are going to focus on? Ma'am, B cells and T cells. B cells and T cells, which are going to provide you that specificity, which is going to provide you humoral immunity, which is also going to provide you cell-mediated immunity. Yes or no? Correct? I am teaching in the class and you people are putting Rohit plus fire emoji. Why? What happened? Chumma sitting and boasting about each other. Okay? Chumma sitting and boasting about each other. Hey Rohit, I am standing and teaching here, but Rohit, who is in the chat box, gets all the fire. Okay? Yes, B cells and T cells. Okay, lymphocytes are going to be B cells and T cells. Now, before you go into cell-mediated immunity and humoral immunity, you should know that these lymphocytes have to be maturing, right? Maturation should happen. Differentiation should happen. Differentiation should happen. And finally, these people should also be sensitive and should go and fight with the antigens. And they should move to the effector cells. For all of this, lymphoid organs are going to play a very important role. Okay? Lymphoid organs have to play a very important role. Okay? Hari Govind, even I have studied ICSE itself. Okay? So, yes, whatever you have studied in your 9th class, you have to study a little more in your 11th and 12th. Drink some water, all of you. Yes, good job. Okay. Oops. Done? Shall we move on? Yes, ma'am. Okay. Now, lymphoid organs in the body. First, I'll teach you this so that it will be easy for me to take you through the humoral immunity and everything. Now, lymphoid organ, students, 2 are there. Primary lymphoid organ and secondary lymphoid organ. Okay, secondary lymphoid organ. Secondary lymphoid organ. Now tell me, what is primary lymphoid organs? So I told you that B cells and T cells are like the soldiers who protect our city. Okay, who protect our entire country. Okay, let's take it as a small city, and there are soldiers or police officers who are protecting us. No, now before they become the police officer, they have a training camp. Right, all of you have seen, no, in movies and all, they want to somehow get into the training camp. That training camp is the primary lymphoid organ. But the place where they actually fight the criminals, you know, face the antigen, that is going to be the secondary lymphoid organ. Remember this always like this. Primary lymphoid organ is a place where their maturation happens. But differentiation, coming in contact with the antigen, being sensitive to the antigen happens in the secondary lymphoid organ. Is this clear? Is this clear to all of you? Tell me in the chats. Yes, okay. Now, primary lymphoid organ, students. Before I go to primary lymphoid organ, remember, as I told you, origin and maturation. Origin and maturation of your lymphocytes happen here. That is in the primary lymphoid organ. Origin and maturation of your B cells and T cells happen in the bone marrow. Sorry, only origin happens in the bone marrow. Okay, of who? B cells and T cells. Origin happens in the bone marrow. But for maturation, T cells will go to thymus. T cells will go where? To thymus for maturation. Okay, for maturation. Is this clear to all of you? In the bone marrow, students, origin will happen. Okay, in the bone marrow, origin will happen of both B cells and T cells. Maturation will happen in the thymus. Okay, maturation will happen in the thymus. Now, where is thymus present? It is present in the mediastinum. All of you know mediastinum, who has studied body fluids and circulation with me knows it. Okay, between the lungs, near the heart, no, just here, mediastinum, thymus will be present, which we are going to study in our chemical coordination, endocrine gland, which we are already studied. So this thymus will be larger in the younger stage, and then as we get older, it will become the smaller and smaller and smaller in size. Okay, that's why our immunity also reduces. So here what is happening, students, origin and maturation is going to happen in the primary lymphoid organ, that is in the bone marrow and the thymus. Here origin will happen of both B cells as well as T cells will happen in the bone marrow. B cells maturation also will happen in the bone marrow. Okay, B cells maturation also will happen in the bone marrow. So remember like this, B bone marrow, B cells. Okay, thymus, T cells. Is this clear? Clear? Yes, reduced along the age. Show me your degree, Divine Heart NT. Let me see your degree to tell about my degree. Okay, yes. Tell me, students. Students, please ask his degree, no? So that you all can study from him from tomorrow. Yes, clear or not? Yes, this is fine. Bone marrow, origin and maturation of B cells. Thymus, maturation of the T cells, but origin is going to happen in the bone marrow. Okay, bone marrow. That means what? Where is the bone marrow? This is your bone. Okay, this is your bone. This portion, no? This is your bone marrow, where the RBCs are formed. So here only the maturation and origin of B cells will happen. If it is clear, we'll go to the secondary lymphoid organs. Okay, secondary lymphoid organs. Now, secondary lymphoid organs, what is going to happen, students? As I told you, here the training has happened that you are going to become a police. Okay, you are going to catch people like Divine Hearts. Okay, and put them behind the bars. So, for that, they are going to train you. Then what happens? You come to the police station. You come to the police station and then you actually face these people who are sitting behind the camera and taking 1,000 rupees and putting this message. After looking at them, we'll feel, oh, for this I got trained. Right. So here what happens? Okay, here what happens, students? Secondary lymphoid organ is the place where they are going to face the antigen. And all of you tell me the secondary lymphoid organs in the chat box. Tell me in the chat box. Quickly. What is Vipul asking? Yeah, now I saw. Exactly. That is the kind of people that comes. Okay, what is Vipul asking? Ma'am. Yes, yes, Vipul, yes. Yes, Vipul, yes, yes. He doesn't understand the language. Okay, clear? Why you all want break? Okay, wait, I'll give you break. Okay, come finish this. Okay, now look here. Secondary lymphoid organ, as I told you, is the place where the fighting is going to happen. Now, so here you have to remember Peyer's patch. Okay, Peyer's patch present near the intestine. Okay, you have to remember tonsils, spleen, lymph nodes. Then students, very importantly, one more word is there, malt. Okay, M-A-L-T, malt. Okay, now look here. Very important, Peyer's patch, tonsil, spleen. Spleen is going to fight blood-borne diseases. That means, let's say in the blood, there is some, pathogen, something is there. Spleen will make sure to attack. Okay, spleen will make sure to attack. This is your lines of NCERT. So, spleen is going to attack blood-borne pathogens. Okay, blood-borne pathogens, and also these are the larger reservoirs of erythrocytes. We have already learned in body fluids and circulation. Where does RBC go and die? Life over for RBC in the spleen, the graveyard. Okay, spleen is also the graveyard of RBC. And there is one line given in your NCERT, the largest reservoirs of erythrocytes. Clear or not? Yes, clear or not? Okay, yes, we will not take a break. Yes, now this one, students, you take a break and come. I am anyways writing, no, you can come back and join, or you can just keep this and drink a coffee or use the restroom or whatever required. Malt is very important for you because it's a PYQ. It's a PYQ. What is the full form of malt? Mucosa associated lymphoid tissue. Now students, this is going to cover 50% of the lymphoid organs. That means what? Mucus lining is there. Everywhere it is there. Just now we studied digestive system, respiratory system, urinogenital system. Some portion will be there in the breast region. Some portion will be there in our bodies, other parts. Okay, so in these regions also, the mucus lining will be there. So malt, like that students, BALT is the BALT, the I'm so sorry, that is bronchus associated lymphoid tissue. Like that NALT, NALT is there. Okay, nasal associated lymphoid tissue. Like that, many tissue linings are there. Okay, depending on the. So here you are talking about mucosa associated lymphoid tissue. Small amount of mucosa will be there in the breast, in the lungs, in the other parts of the body, small regions also. Some amount of your lymphoid tissue or your mucus associated tissue will be there. This is going to contribute for the 50% of the lining. Where is the 50%, ma'am? From the respiratory, digestive, urinogenital. Is this clear? Are you able to associate? Okay. Now, most importantly, secondary lymphoid organ, this is clear, spleen is clear, lymph nodes are clear, tonsils, mucus associated lymphoid tissue. Question has come as the full form or about the number, 50%. Okay, the 50% number also is come. Okay. Yes. Now, primary lymphoid organs, secondary lymphoid organs. I'm not going to answer your questions if it's not pertaining to what I'm teaching right now, okay, because it's a very disturbance to the class. So please avoid, if you want to search something, go to Google and search. Don't waste your time asking the same doubts here and spamming. I'm not going to answer until it is related to this topic. Okay, yes, you might know a lot of things, you might not know a lot of things. Very good for you students. Please search elsewhere because it will stop the flow of the class. See, this conversation also is unnecessary. Clear? Please try to be cooperative. Last class I did not have any issues. My students were very full-time concentrated. They were just learning. Okay, this time also I require the same thing. Yes, this is clear. Primary lymphoid organ, secondary lymphoid organ. Yes or no? Yes or no? If it's clear, I'll move to acquired immunity. Yes. Thymus is clear, no? Okay. Now students, lymph nodes also, they trap, problematic pathogens from the tissues. Spleen on the other side is a bean-shaped structure which will trap blood-borne diseases. So try to remember that. Okay, blood-borne and your pathogens. Okay. Yes, I'll give you 5 minutes break. Two minutes you people wait. Okay. Now acquired immunity, because the topic is a continuation, so we will just continue, because otherwise the break will happen. Okay, and these people will run away, they will not come back. Now look here, acquired immunity students, two types are there. Two types of immunity is there. What are the two types of immunity? Humoral immunity, and cell-mediated immunity. Now humoral immunity, as I told you, when we spoke about Hippocrates and your Ayurveda, they also spoke about human in the body. That means what? The fluid content. Here also, humoral immunity we are talking about. Okay, we are talking about what? We are talking about the fluid part, right? The fluid part. So here we are talking about blood. We are talking about blood. So in the blood, there is going to be the B cells, and B cells are going to continuously release antibodies to protect our body. Okay, antibodies. Now this is what you are going to continue. So humoral immunity means the human. That means what? The blood, the fluid content in our body. So blood is going to be the fluid in our body. Yes. In that, your B cells are going to be there, and these B cells are going to release antibodies just like the bullets. Okay, just like the bullets. So now imagine I am the soldier. Okay, I am the soldier and I am let into a field. Now I have bullets. Okay, I have bullets. What is that? Antibodies. Now on the other side, I have cell-mediated immunity. Cell-mediated immunity, the main person who is going to be involved is your T cells. Is going to be your T cells. Remember, T cells are not going to form antibodies, but they are going to help. They are going to help T cells, B cells form antibodies. Understood? T cells are not going to directly involve in the formation of antibodies, but they are going to help B cells. Telling that I will do all the help. You continue. Okay, so I will help you form the antibodies. Clear? So T cells are going to be part of the cell-mediated immunity. You will also see the word CMI. You'll also see the word CMI in the books. CMI in the books. What is happening? What is happening? I can't understand your language. I understood that someone is disturbing someone. Tell me who is disturbing. Tell the person's name. I will remove the person from the class. Okay? There is no excuse for those students who disturb in my class. Not even a minute I will let you be in my class. Okay, I understand it is YouTube. I understand that you all have phones and you can tell anything and everything. But no, not in my class. Okay, yes. Okay, just tell the person's name. Anyone who is spamming, I will remove the person from the class. Okay, I care about the students who have come here to study. Yes, humoral immunity is clear. It is going to be the blood content, okay, the fluid part, and B cells are going to release antibodies. Cell-mediated immunity is going to be the T cells. They are directly not going to be involved in forming antibodies, but they are going to help B cells, telling that you form antibodies, I will help you. Now students, remember that your B cells, okay, B cells have two major cells. What are they? Plasma cells and memory cells. Plasma cells and memory cells. These plasma cells are not very specific, okay? They are not specific and their lifespan is not very longer, okay? It could be for 5 to 6 days, but memory cells are not like that. Memory cells are going to be specific and they are going to hold the memory, right? They are going to hold the memory of who attacked me. That memory they are going to hold, okay? So these people are going to be more specific compared to plasma cells. Then when you come to T cells, okay, after this I'll give you a break. Listen to me, very important. T cells, students, T cells, multiple types of T cells are there. What are they? They are going to be called T helper cells. Okay, I'll just write TH. TH, which is T helper cells. Okay, then what? T K cells, that is T killer cells. T killer cells. Then what do you have, students? T suppressor cells. Suppressor cells, and obviously T memory cells, right? T memory cells. These are the types of cells that the T cells is going to have. Is this clear to all of you? Very good. TK, TH, TSUP and TM cells. Okay. Okay, clear or not? Yes. So, B cells have plasma cells and memory cells. Plasma cells are not going to be that specific and their lifespan is going to be less, 5 to 6 days. But memory cells are not like, they are very specific. They know why they have come here and they know what they are doing. T cell, students, there will be T helper cells, and all of you remember, in AIDS, we are going to study about T helper cells who have CD4 receptors, right? And TK cells, which are T killer cells, TSUP, that is T suppressor cells, and T memory cells. This is the whole number of types of T cells. Clear to all of you? Yes or no? Yes or no? Clear? Okay, before I go to antibody, I'll give you a break. Okay, otherwise you people will, I think you will break my class. Don't go anywhere, just keep the live on and go do other works. Okay, go do other works. Clear? 5 minutes break. After that, I'll give you a longer break once I almost finish the class. Longer chapter, students. If I give more breaks, it will not get over. And then Baswa sir can't start the class. Okay, then you people will have to stay awake for a very long time. That's why I do not want to give longer breaks. Okay, yes, Abhinaya, you just be here, dear. 5 minutes break, everyone are asking. So, 5 minutes break, that's all. You people stay on the class. Don't go anywhere. Oh, sorry. I'm so sorry. 2:10. What is 2:10? I'm still in afternoon, students. What is the time? 7, no? Yeah. Sorry. Okay, hi guys. Yes, let's continue. So lymphocytes are clear. Okay, then you understood where they are originated, maturing, and where they are going to fight with the antigen. That is also clear to all of you. Now you're going to study about what the B cells release. No, I told you that B cells, when they are in the blood, they are going to release bullets. Who are the bullets? Antibodies. Now this diagram indicates an antibody. Okay, this diagram indicates an antibody. We have to draw one diagram, a bigger diagram, so that we understand, okay, clearly what is antibody. So I'm just drawing a very simple diagram just to make you understand. Okay, this is an antibody. Now let's write more about it. Now students, as you know, okay, antibodies are glycoproteins. What are antibodies? They are glycoproteins. Antibodies are glycoproteins. Proteins means what are they made up of? Ma'am, obviously, since you told they are proteins, that means they are made up of amino acids. Okay, they are made up of amino acids. Exactly right, students. Antibodies are made up of two heavy chains and two light chains. Two heavy chains and two light chains, and these chains are made up of amino acids. Okay, these chains are made up of amino acids. Now, you know, in biomolecules, when you have studied amino acids, you know that they will have an N terminal and a C terminal. Yes or no? They will have an N terminal and a C terminal. So here also same thing is going to happen. This is going to be the N terminal. Okay, this is going to be the N terminal and this is going to be the C terminal. Okay, this N terminal and C terminal, once question has come, that's why I'm writing it here. Yes, exactly. They are H2. L2 is the formula, two heavy chains and two light chains. Now, understand that, understand that these parts are there, no? These parts are there. These are the heavy chains and these are the short chains. Now, in this, the most important thing is that this is the place where the antigen is going to come and bind, okay? This portion where the antigen comes and bind, I will call this the paratope. What will I call it? Paratope. The region where the antigen comes and binds is called the paratope. This is called as antigen binding site and it is called a paratope. So, one antibody will have two paratope. Yes or no? Agreed, all of you? Tell me yes or no in the chats. One antibody will have two paratope. Agreed, disagreed? Agreed, disagreed? Yes, we are coming to that. Okay, we are coming to that. Paratope. Now, students, to this paratope, antigen has to come and bind. No, to this paratope, antigen has to come and bind. So, remember this way. Let's say this is an antigen. Let's say this is an antigen. This portion, okay, just give me 2 minutes. Okay, look here. This portion where the antigen will come and bind, okay, antigen will come and bind to the antibody, okay, that is called an epitope. The region on the antigen that will come and bind with the antibody is called the epitope. Clear or not? So, this is an antigen example, just like one mushroom I have drawn. This is an antigen. This portion is going to come and bind with the antibody. So, this portion is called an epitope and this portion is called the paratope, which is also called the antigen binding site. Okay, antigen binding site. As I told you, students, this region is going to be variable. Why? Because different antigens are going to come and bind. So, this region is going to be the variable region. Okay, we can also call it as FAB, that means F antigen binding site, and this portion is going to be the variable region. Okay, this portion is going to be the variable region. I think writing is not visible. Is it okay? Is it okay? 1 second. Okay, this is the FAB region, that means antigen binding region, and this is V region, variable region. V I am writing variable region. This portion which is there, no, this down portion, okay, this down portion, okay, here, this portion is going to be the constant. Okay, it's going to be the constant. We can also call it as FC because of the crystallization. Okay, because of the crystallization. Clear? Yes. So here this is the FC region, which is the constant region, and this is the FAB region, which is the variable region. Clear? Now, as you can see, there is a long chain or a heavy chain and a light chain. They have to be clubbed together. They have to be clubbed together. For them to be clubbed together, students, remember, there is disulfide bonds between them. What is going to be there? Disulfide bonds are going to be there. S, disulfide bonds. Like this, 16 disulfide bonds together forms this entire antibody. 16, how many? 16 disulfide. 16 disulfide bonds form this entire antibody. Okay, if you want, you can count it on this diagram. You can count in this diagram. Okay. No, nitrosonium, no. Epitope is the region of the antigen that binds with the paratope. Antigen, let's say there is a lock in key. Okay, let's say this is antigen, this is antibody. This pen, can you see, this is fitting here. Okay, this is fitting here. Now, this portion that is going to fit there is called the epitope. This portion where the fitting is going to happen, that is the paratope. Understood? Understood. This is a lock and key model. Key, that is antigen, epitope. On the antibody, the region is paratope. Is this clear? FC and F constant is just the region. This whole region is going to be variable. This whole region is going to be constant. Those are just regions. Clear? Okay, clear? Any doubts in this? Please let me know in the chats. Who asked me that doubt? Is it clear? Okay. Now, two heavy chains, two light chains. Paratope, the region where the antigen, okay, antigen will bind. Okay, antigen will bind on this region, and this is the region on the antigen where it is going to bind with the antibody. Okay, going to bind with the antibody. Then one antibody will have two paratope, 16 disulfide bond. This is the whole chain. Can you see? This is the light chain, this is the heavy chain. And they will have an N terminal and a C terminal. You have studied in biomolecules. Many amino acids come together. There will be an N terminal and there will be a C terminal where the NH2 is going to be free and where the CH will be free. So, this is going to be called as the N and the C terminal. Clear? This is your entire antibody. Now, remember that depending on the region, okay, depending on your FC region, there is going to be 5 types of antibodies. There is going to be 5 types of antibodies. Now, ma'am, should I know everything? Some questions have come in the past years from this. So, some topics you need to remember. Okay, some topics you need to remember. Ready? Now, look here, students. IgD, IgA, IgE, IgG and IgM. You can remember it as GAMED. Okay, GAMED. G-A-M-E-D. GAMED. Okay, GAME plus D. I just added like that. GAMED is the order of which you can remember this or just the name of this. Okay. Now, look here. You study right now this one antibody. But if you look here, this is a dimer. This is what? A dimer. That means two antibodies are coming together, and always remember, when two antibodies are going to come together, they are going to be joined by a J chain. They are going to be joined by a J chain. This is for people like, you know, who, ma'am, how did they join? Okay, how did they join? That for that question I am telling you. Students, why is this mic doing salsa with me here? Okay, 1 second. Okay, so here this is one antibody that we learned. Okay, heavy chain, light chain. One more antibody has joined downwards. These two are attached with the help of a J chain. Clear? These both are attached with the help of a J chain. Dimer. This is a dimer. Okay, these are just informations for you. If you want, you can remember, but then dimer you please remember. Okay, dimer you please remember. Now, IgA is the second most abundant antibody. Okay, now Ig means what? Immunoglobulin. Okay, immunoglobulin, IgA, IgB, that means they are the ones who are going to do the defense mechanism. Okay, second most abundant one, and IgA is also most importantly asked question is about the colostrum. The colostrum. The milk, the first milk that comes out, right, from the mother will be rich in IgA. This question has come many times, so please remember this. Colostrum will be rich in IgA, which is a passive natural immunity given to the child from the mother. Okay, from the mother. Clear or not? I'm so sorry for the cough. Fine. Okay. Now, going forward, IgD. Students, I'm not going to tell much about IgD. It's a membrane bound one. All that you don't have to remember. If you look here, this is the normal one, right? Single one. So, these people will have 2 paratopes. These people will have 2 paratopes. Here, how many paratopes will be there? 1, 2, 3, 4 paratopes will be there. Okay, 4 paratopes will be there. They can ask you a question. Okay, the 2 paratopes, which are the one that has 2 paratopes? So, this is a monomeric one. This is a monomeric one. Now, if you look here, IgE is very important for you because this is the one that is going to come forward for an allergic reaction. First fighter. He's like, I am ready. Allergy is there. I'll be the first person to come. So, IgE, allergic reaction. This question also has come in the past years as a PYQ. Okay, this question has come. Fine. So, whatever questions have come, I'm just going through it so that you do not miss any marks. IgG, students, it is the most abundant. Most abundant. That is around 75% is IgG. And most important thing about IgG is these people can cross the placenta. Cross the placenta. This is very important. Again, a PYQ. Okay, again a PYQ. They can cross placenta. Very important. Very, very important. How allergic reaction is important. How colostrum is important. The one, they can ask you the immunoglobulin that can cross placenta. Cross placenta means what? From the mother to the child. They can cross the placenta. This is only possible by the IgG. Can you relate to this? Now, when you go here, students, IgM. Look at the complication in IgM. This is a pentamer. This is a pentamer. Now, if it's a pentamer, all of you tell me in the chat box, how many paratopes are going to be there? How many paratopes are going to be there? Chat box fast. How many paratopes are going to be there? Think, just count. See, 1, 2 into 5. 5 2s are 10. Very good, very good. 10 paratopes are going to be there. And students, this is the largest in size. Largest in size. Clear? So, these are the top 5. I have only written the important points. I have not written a lot. Okay, I have not written in detail for any of this, but in years before we have seen questions on this. So, they expect the teacher to teach you. So, I am teaching you and only important words I have written like colostrum, allergic reaction, cross placenta, largest in size, it's a pentamer. This much only I have written. Clear? So, these are the ones. Please make a note of this, or anyway you will get the notes on Biology by Gopika as well as Vedantu NEET English Telegram channel. Okay, moving forward. Now, before I move forward, I have to tell you one more important thing. Okay, here only we will write, students. Students, what are the things that are done by the antibodies? Okay, there are 4 terminologies that usually we all teach in coaching as well as we have also learned in coaching, is that what exactly do these antibodies do? Now, you told me, ma'am, that these people are the part of the humoral immunity. Yes, understood, ma'am. But what do they exactly do? They are going to do something called as agglutination, students. Agglutination. That means they are going to bind with the antigen. Okay, first thing. Then they can do something called as opsonization. Opsonization. Now you can remember this as N neutralization, O opsonization, complemented mediated. NOCA. Can you remember NOCA? Can you remember NOCA? Who is this? Okay, NOCA, can you remember, students? If you can remember NOCA, then it is easier for me to, tell this. So, NOCA, okay, N for neutralization, okay, and A for complement mediated. These are actually the functions of your antibodies. That means what? These are the things that these people do. What is the first thing that they do? Binding. They'll go and bind with this fellow, telling that I'm not going to leave you, I'm going to kill you. Then they can do opsonization. They can either swallow it, or the many antibodies are going to attack and give it to the macrophages. Macrophages are going to do phagocytosis. Okay, so what is going to happen, students? Let's say, okay, this is an antibody. Okay, this is an antibody. Sorry, this is an antigen. I'm so sorry. This is an antigen. Now, from all the 4 sites, okay, okay, I'm just going to draw Y, okay, because full if I draw, it will take time. From all the 4 sites or 5 sites, these antibodies have attached, okay, attached to this, okay. Now, let's say this is your macrophages, okay, this is your macrophages like this, waiting, okay, waiting. Okay, they are waiting. What these people will do? These people are going to bring this fellow here and give it to the macrophages, and macrophages will do phagocytosis. Okay, macrophages can do phagocytosis. Is this clear to all of you? How they are going to function? How the antibodies are going to function? Yes or no? Okay. So you can remember NOCA, okay, like LUCA, NOCA, you can remember. NOCA, N for neutralization, O for opsonization, C for complemented mediated, A for agglutination. This is exactly what will happen, okay, in opsonization. The antibodies will attack the antigen, they will bring it to the macrophages, macrophages will do phagocytosis. Like that, they can do neutralization, complemented mediated ones, okay. So this is the function of your antibodies. If this is clear, we will move forward to active and passive immunity. So far clear? Any doubt so far? Please let me know right now itself. If it's clear, we will move forward, okay? Active and passive immunity, students. What is active immunity? Active is when our body is making the antibodies, right? When the body is given the time to make the antibodies, okay? So when our body makes own antibodies. Then, ma'am, what is passive, ma'am? Students, passive is a situation. Passive is when we have preformed antibodies. Preformed antibodies. Like ready-made. Ready-made. Let's say in our class now, Pragadishwaran is there, okay, Pragadishwaran is there in our class, who is sending, okay, Pragadishwaran, okay, who was spamming the class, right now is kicked out of the class. He has gone to a market, taken a cloth, and he started stitching by himself. That means he's a very active, okay, he's active, he started making his own clothes. Then on another side, we have, let's say, Kubera, who went to the shop, got a ready-made dress and is wearing it. Now what is going to happen? One person is okay, taking the time and stitching, another person is going and buying from a shop and coming. Same thing is this. Here active means we are giving the time for the body to fight, make antibodies. On the other side, we do not have time for it. So what we will do? We will use preformed antibodies. Clear or not? Now active can be divided into natural and artificial. Active can be divided into natural and artificial. Natural, artificial. Okay, passive also can be divided. Yes, ma'am. Passive also can be divided into natural, artificial. Natural, artificial. Now all of you tell me in the chat box, what do you think? Natural. That means what? Now you have got an infection. Your body is going to start making antibodies against it. Yes or no? Tell me the answer for this. Read your NCERT and tell me. That means what, students? Self-infection. Okay? Self-infection, or we can also call subclinical. Subclinical. That means you got an infection and you're letting your body to go against it. Okay, to go against it. That means I got cold. I'm letting my body only to the immune system itself to make antibodies and fight it. Okay. Now, artificial means what? Like your vaccinations. Vaccinations. That means in vaccinations, what happens? And immunization, what happens? We give weakened antigen. Okay, we give weakened antigen. What is weakened antigen? It is not active to harm us, but our body will immediately realize that there is a foreign particle in my body and I will start making antibodies against it. So vaccination and immunization are weakened antigens. Clear? This is artificial. This is artificial because I'm injecting into your body and your body is making, but still your body itself is making. I'm not injecting the antibody, but I am giving a trigger for my body to make the antibodies. Clear? Now, when you come to this side, you just have to remember the example, students, nothing else. Okay, examples they will ask you. Now, here also, students, pre-formed antibodies, ready-made I'm giving. I'm telling that, hey, you do not work. I will give you everything ready-made. Ready-made antibodies I will give you. Okay, here natural example is going to be colostrum. Okay, that is given from the mother to the child. Okay. This is natural but passive. Why? Because I'm not injecting anything into the mother for making the colostrum. She is making it naturally in her body, but for the baby it is ready-made. Thumbs up? Thumbs up? Clear or not? Yes? Okay. Now, artificial means what, students? Like your anti-venom. Okay? Like your anti-venom. Okay? That means I do not have the time to put venom, or not time, forget, it's very dangerous. I cannot put venom inside the body and expect my body to make antibodies against it. Person will die. So what I will do, I will inject it into horse, some cattle, and get the antibodies from it and inject it directly into the body. So this is anti-venom. Clear to all of you? This is all that you need to know. If you want to make a note, my hair has drawn diagrams here. Look at this. Yes. Okay, if you want to write it, please make a note of this as very good. Artificial, anti-venom, anti-tetanus, like that many. Okay, even for rabies, for all of that, we can do this artificial system. Okay, yes, during the emergency, when our body does not have the time to do this whole process. Okay, and it is too dangerous for the body as well. Right? Imagine I'm telling that, no, I'll put venom. Okay, after the person die, what's the use? No, Batman, not like Spider-Man, Batman. Okay, yes. Now, vaccination, okay, vaccination and immunization, very simple, same terminologies, okay, same NCERT it is. So, let's go through this. The principle of immunization or vaccination is based on the property of memory of the immune system. Memory of the immune system means what? Our immune system, your T memory cells, your B cells, all of them are going to be aware that, okay, this has happened to me and this is the memory. COVID also, we got booster dose, we got vaccines. Now our body knows that, okay, this, I have made antibodies against this and now I have to be aware when this, COVID comes back again. Same thing. In vaccination, a preparation of antigenic proteins of pathogen or inactivated weakened pathogens, that is the vaccines, are introduced into our body, but it is always tested. It is not introduced just randomly. Okay, telling that, oh, this pathogen is sleeping now, so I'll inject it into a person. No, it is already checked because it can become dangerous in our body. Now, the antibodies produced in the body against these antigen would neutralize the pathogenic agents during the actual infection. Clear or not? Next time, even though we did not get COVID, some of us got, some of us did not get. Now, vaccination is already given to me. Now, next time my body come across, SARS or your COVID, my body will already neutralize it. Imagine at the first attempt, my body was able to only fight with 10 armies. Okay, 10 army. The second time it comes, this is the primary response. My body did not know it, so it was scared. Second time it comes, it will fight with 1000 armies already ready. Because it has memory. Because it has memory. Clear to all of you? Yes? Okay. Now, the vaccines also generate memory, B cells and T cells that recognize the pathogen quickly on subsequent exposure and overwhelm the invaders with massive production. With massive production of antibodies. That means, students, in the primary response, let's say it was for a longer duration because we did not know. Okay, we did not know. Okay, drink water, ma'am. My voice is cracking, students. I don't know what happened. Okay, I think it's the weather. Yes, we have so much more. Okay, Baswa sir has put the emoji. That is how much this chapter is. Okay, we have reached immunity. We have AIDS, cancer, drugs. Okay, chalo. If a person is infected with some deadly microbes to which a quick immune response is required, as in tetanus, we need to directly inject a preformed antibodies or an antitoxin. Okay, a preparation containing antibodies to the toxin. Even in cases of snake bite, the injection which is given to the patient contain preformed antibodies. So these are the two examples. Okay, antivenom and anti-tetanus. Okay. These are the two examples of your artificial passive immunity. Artificial passive immunity. Okay? Yes, fine. Now, this type of immunization is called as passive immunization. Clear or not? I hope no doubts in this. So, what is happening, students? First time, primary response. Okay, that is my body does not know this has happened. My body is like, oh, yo, this has happened. So, it is going to be for the first time. Primary response, no? So, okay, here I will write it here. What is going to happen? Primary response. What is primary response? It is happening for the first time. It's happening for the first time. Right? Okay, yes. So this is going to happen for the first time and this response is not going to be that strong. Students, imagine first time you are coming across, like let's say very simple thing. First time you come across failure. Okay, in your NEET preparation, first time you would have come across failure. That time it would have taken a lot of time for you to recover. You might be thinking that you're a failure, that I couldn't do it. But the second time when you're going to write the exam, the second time you have already learned from the mistake. Second time, will you do the same mistake that you did in the first time? No, ma'am, of course not. So here also, since this is during the first time, it is going to be a weak response. It is going to be a weak response. Because we do not hold memory of this, we do not know anything about this pathogen, so it is going to take longer time. Okay, takes longer time. Clear? This is the primary response. Now, secondary response. Okay, secondary response on the other hand, have you all liked the video? Tell me. You all have to do quiz after this, no? Yes. Secondary response on the other hand, what is exactly going to happen here? It is not the first time. It is they hold a memory of the first time. They hold a memory of the first time and it is not going to be weak. It is going to be heightened. It is going to be stronger, double stronger, right? When you come for the second time. Okay, first time students, okay, but when you come for the second time, you're going to be more stronger, more powerful. I'm ready to fight anything, ma'am. Whatever be the PYQs, whatever be the difficulty of the paper, I am ready for it. Right? So, heightened. Clear? Does it take longer time? No, you already know the topic, so it takes shorter time. Takes shorter time. And let's say first time we fought with 10 army people, okay, this is the antigen that came. We fought only with 10 people. 10 people. Only 10 people fought in the first time. Second time, our T cells and B cells are going to give a massive attack to show that our body is very powerful. Okay, our body is very powerful. This time you will be fighting with 10,000 people. Okay, this is what they told as a massive attack. Because we know how problematic this is. Acquired immunity has a specificity, diversity. We know what to face. Okay, yes, this is the whole thing. Okay, clear? Yes or no? This is the whole secondary and primary response. Now we have allergy, and after that we should be able to come to an end of immunization. Okay, yes, allergy. What is allergy? Ma'am, allergy keeps coming and going. Students, anything that can cause allergy is called an allergen. Anything that can cause an allergy is called as an allergen. Now, when an allergen enters into your body, okay, it got inside your body. What is going to happen inside your body? Have you ever thought about it? What would happen inside your body? Let's say this is your body. Okay, this is your body. What is happening inside your body? We haven't changed color in 2 days. Okay, if such a thing happens in our body, then mast cells, what cells? Mast cells are going to get activated as soon as an allergen comes in. And these mast cells, students, they are so smart. Okay, they are so smart, they are so active, they will release histamine. Okay, yes, they are going to release okay, yes, what are they going to release? They are going to release histamine, that does what? Vasodilation. That does vasodilation. Ma'am, why will they do vasodilation? It is very important for you to know, no students, that is, let's say blood vessel was like this. Okay, before blood vessel was like this. Now, the amount of blood that is going to enter is going to be normal, right? Yes, histamine. Students, histamine, serotonin, heparin, all this will be released. Okay, all this will be released, but don't take tension now. Only histamine we are going to talk about, okay? This is normal one. Now, what is going to happen, students? When histamine is released, vasodilation will happen. That means the blood vessels will be dilated, and this will lead to more blood to flow inside, so that what will happen? It will immediately start showing redness and inflammation in our body. Redness and inflammation in our body. Understood? Yes or no? More blood will start flowing, leading to redness and inflammation. Redness and inflammation. Clear? This is how you get to know that you have an allergy. If you don't know allergy has happened only, what will you do? Okay, what will you do? How will you react to it? So, the person who is capable of causing an allergy, okay, that is called an allergen. Okay, they get into our body, mast cells will realize it, they will release histamine, vasodilation will happen. Okay, release histamine, leading to vasodilation. Vasodilation will lead to more blood flow. Okay, more blood flow, and more blood flow will lead to redness and inflammation. Clear? Yes, that's it. That is your allergy. That's all is allergy. Okay? Yes. Now moving forward, autoimmune diseases. Okay, this also you people know. So we will just go through it. See, memory-based acquired immunity evolved in higher vertebrates based on the ability to differentiate foreign organisms from the self cells. That is, from the self cells. While we still did not understand the basis of this, two abilities, okay, that means two identities of this ability have to be understood. The differences have to be understood. One, higher vertebrates can distinguish foreign molecules as well as foreign organisms. Yes or no? Yes, ma'am. Most of the experimental immunology deals with this aspects. Two, sometimes due to genetic or other unknown reasons, okay, sometimes due to genetic or unknown reasons, the body attacks the self cells and starts making antibodies against the self cells, okay. This results in damage of the body and it is called as an autoimmune disorder, autoimmune disease. That is rheumatoid arthritis, myasthenia gravis, all of this are autoimmune disorders where they start killing their own cells, they start making antibodies against their own cells, okay. Rheumatoid arthritis, which affect many people in our society, is seen as an autoimmune disorder. So they are telling that the first reason is, even though higher vertebrates can differentiate foreign particles as well as foreign organisms, we do not understand in some cases due to any genetical change or other changes, why does our body start making antibodies against the self cells, that means our own cells. No one knows. So because of this, this leads to rheumatoid arthritis or myasthenia gravis. Myasthenia gravis you have studied in locomotion. Rheumatoid arthritis you can study here. Clear? This is about your autoimmune disorder. Autoimmune disorder. I was thinking, why didn't the board do today dance? Started. Done? Now, students, graft rejection also can happen, okay, which you will be studying again in microbes chapter, where you will study about cyclosporin A, which is an immunosuppressant. That means when we are trying to take a foreign particle, that is, let's say some people do a lot of graft, okay, graft implantation and all, that is from the thighs, they will take the muscle, they'll put it on the face, on different parts of the body. During this time, even though sometimes we take it from our own body or we take it from someone else, okay, like we take kidney from someone else, our body has a tendency to react to it because it's a foreign particle. During that time, we will give an immunosuppressant called cyclosporin A, which will reduce the attack of the body against this foreign particle until the entire process happens. Okay, that is your graft rejection. Okay, 2 lines are given about that. That's why I had to mention. Okay, answer this question, all of you. Autoimmune disorder is a condition where the body defense mechanism recognizes its own cell as foreign bodies. Rheumatoid arthritis is a condition where the body does not attack self cells. 2022 question. 2022 question. Yes, why haven't all of you liked the video? Come on guys, what is this? Poor response. Yes. What is the answer? Oh, mixed answers are coming. 1 second. Is it not visible? Visible, no? Okay. Yes. What is the answer? Statement 1 is correct, but statement 2 is incorrect. Very good. Okay, statement 1 is correct, statement 2 is incorrect. So you have right now plus 16 marks. Okay, those who got plus 16, please put it in the chat box. Okay, plus 16. Autoimmune disorder is the condition where the body's defense mechanism recognizes its own cell as foreign bodies. Correct. Okay. Next question. In which body corpuscles HIV undergoes replication? Okay, we didn't reach HIV. So we will do this after coming back. Okay, tell me this. In which disease does mosquito transmitted pathogen causes chronic inflammation of lymphatic vessels? 2018 question. Answer should come fast. So we can get into AIDS and cancer. Plus 16. Very good. What an all name, yeah? Prawn. Which prawn? Prawn, rohu, cartilaginous fish, bony fish, after some time frog, cockroach. YouTube shouldn't allow to change the names. I feel that rule should come. Whatever first name you kept, no, that only should be your name. Yes, very good. Elephantiasis, it's a chronic inflammation, okay, chronic disorder. Plus 20. Okay, plus 20. Tell me this. Malt constitutes about dash percentage of lymphoid tissue in human body. NEET 2017 question. NEET 2017 question. Tell me quickly. Malt constitutes about dash percent of lymphoid tissues in human body. Yes, good job. 50%, right? Yes, 50%. Malt full form is mucosal associated lymphoid tissue. Plus 24. Plus 24, 2017 question also done. Okay. Okay. Identify the correct pair representing the causative agent of typhoid fever and the confirmatory test for typhoid. NEET 2019 question. You changed your name now, right? Is it I was there? Oh, I'm so sorry, dear. So sorry. W. I'm so sorry, Praveen. So sorry. I called you prawn. Okay. Students, I couldn't see W and I. It looked like prawn. I couldn't see. Now I'm seeing the I. People didn't change the name. No, you people scam me a little. I'm so sorry, dear. But then there are a lot of people who come like this, cockroach, frog and all. It happens. Yes, what's the answer? Yes, very good. Salmonella typhi, Vidal. This is going to be 28. Okay, 28. Those who got 28, please put it in the chat box. Okay. Chalo, let's do AIDS. AIDS. Okay, we will do a standing apology. We'll do an entire apology for who? Our Praveen. Yes. Okay, continue. Okay, so now students, AIDS, 28 marks, everyone. Very good. Now, AIDS, what is the full form? Acquired immunodeficiency syndrome. Okay, syndrome. So from the word itself, you know, acquired, that means it has happened during the lifetime, right? During lifetime until it is, you know, spread from the mother to the child. During the lifetime it has happened. Immunodeficiency means where the immunity, okay, where the immune system will be sacrificed, right? The immune system will be majorly affected. Okay, and syndrome means multiple organs. Okay, multiple organs will be multiple symptoms will be there, students. Multiple symptoms and multiple organs will get affected. Okay, will be get affected. This is the decoding of the name acquired immunodeficiency syndrome. Okay, acquired immunodeficiency syndrome. 1 second. Yes. Now, first AIDS case came in US, okay, and it was found in gay community. So people thought, okay, maybe this is a syndrome that only comes, okay, it only comes in gay community. But later, and it was also called GRID or something, okay, back then. Then people noticed that many other cases also came around the world to different doctors, and then they told that, okay, it's not because of someone's sexual or, you know, because of the community they are in. It is not because of the community, or it's not because of their sexuality that this disease is coming. This is a totally sexually transmitted disease, and this is caused because of a virus called as HIV virus, which is called as human immunodeficiency virus, right? This was what the first finding was, okay, that they told that in 1981 in USA there was a case that came, and they noticed that this is a very rare, okay, rare thing. They thought first it was fungi, then they thought it was bacteria. So multiple, you know, theories came, and it happened in 1981. First in USA, came in gay community. So it was considered that this is because of their sexuality AIDS have come. Then later multiple other cases came, and then they told no, this is not anything to do with sexuality. It is because of a sexually transmitted disease, and this is a viral infection caused by HIV, which is a oh no, 1 second, I'm so sorry. Yes, which is a retrovirus. Which is a what? Retrovirus. What is retrovirus? Retrovirus is the category of virus that has the ability to convert RNA to DNA. RNA to DNA with the help of reverse transcriptase. Okay, with the help of reverse transcriptase. So these people are very dangerous because they have the ability to convert their single-stranded RNA to DNA of the host and manipulate the host and change. Okay, manipulate the host and change. So acquired immunodeficiency syndrome, please understand students, a person can be HIV positive. A person can be HIV positive, but if the person had to be AIDS positive, it will take a span of 5 to 20 years, okay? 5 years to 10 years, 10 years to 20 years it can take for that person to become AIDS positive. Understand that? From HIV positive, some might be tested HIV positive, but to come to AIDS positive, it will take a longer time because these people are very capable of attacking cells which have CD4 receptor. CD means cluster of differentiation. This CD4 receptor is present in our cells like our dendritic cells, T helper cells, macrophages. So these people will affect these cells and they will reduce the immunity in our body for a longer period of time, and the person will die not because of AIDS, but the person will die because of opportunistic diseases. What is opportunistic diseases, students? It could be a common typhoid, it could be TB, it could be tuberculosis, it could be jaundice, it could be any normal bacterial, viral, protozoan, Helminth disease which can be cured usually with a vaccination or, you know, sometime with medications. But because the person's immune system is sacrificed because of AIDS, the person will, okay, get affected with an opportunistic disease. Okay, yes. No, see, what is HIV? HIV is the virus, human immunodeficiency virus. This can be present in your body, but it will take a lot of span to spread all over your body and you become AIDS positive. That's why a child, if you see, who is like, 5 years, 6 years, the child is HIV positive. The child's body condition will be fine. But over 5 to 10 years, you will notice that the body's immunity has reduced drastically and more HIV cells, enter, like more HIV cells will be found, which will automatically we call the person as AIDS positive. Understood? That means HIV does not come today and like tomorrow the person will not die. It will take a span of time. Okay, span of time. No, Here only this, okay, human immunodeficiency virus. Opportunistic diseases causes the death of the person. Clear? Clear? Yes. Now look at the structure of HIV, students. This is the structure of your HIV virus. Now this is the phospholipid envelope. Can you see the circles? These are the phospholipid envelopes. In the phospholipid envelopes, there are docking glycoproteins. Okay, this circle, this lollipop kind of structure that you're seeing now is a docking protein. Now, in that, the top portion, the top circular portion is called as GP120, and the lower that structure no is called as GP41. All are in numbers. Okay, so this is a phospholipid. In that you have glycoproteins. This circular structure is called as GP120. This is called as GP41. Clear to all of you? Yes or no? Okay. You cannot see. You can only give antiviral drugs. Okay. You can give drugs, steroids, which are going to control or maybe increase the lifespan of the person, but we cannot completely eradicate HIV from the body. Understood, Rajkumar? You can just increase the lifespan of that child or of that person by giving antiviral, interferons, other things, but you cannot eradicate completely HIV from the body, because their ability is like that, no? So here you see, students, this is GP120, this is GP41, this is the capsid. All of you remember the capsid in biological classification we have studied. Capsid is made up of multiple capsomeres. They are just the protein code to protect the genetic material. This capsid is there. Inside that, the pink color structure is you're seeing is a single-stranded RNA. What is it? Single-stranded RNA. Now, these people are so capable. Why? Because they have 3 enzymes in their body. Okay, these people have 3 enzymes. One is called as reverse transcriptase, ability of them to convert RNA to DNA. They have protease so that they can integrate to host protein. Then they have integrase to do the integration. So because of this, these people are very, very dangerous. Okay, very dangerous. Why? They have their own enzymes. They have come with full system. Okay, now students, remember, these people will only affect the cells which have CD4 receptors. Which are the cells that have CD4 receptors? Dendritic cell, macrophages and T helper cells. In that, they are going to use macrophages as the factory, telling that I will make all the copies in this factory, but I will not kill the factory. Will you damage a factory that you are producing more goods on? No. But if I affect the T helper cells, I will make sure to kill the T helper cells. Okay, I will make sure to kill the T helper cells. Why? Because T helper cells are the part of the cell mediated immunity. I have to kill them. I have to reduce the immune system. Clear? Now, what exactly happens? Look here. This GP120 comes in attached with the CD4 receptor. In the macrophages, they will bind to the CD4 receptor. After that, the capsid inside the host cell, the entire capsid itself will go into the host cell, no? So, it will enter the cell, and what it will do first? It will convert the RNA to DNA with the help of reverse transcriptase. And this is our host DNA. This is our human host DNA. What these people will do? They will use integrase and integrate the viral DNA with the host DNA. Can you remember all of this? Okay, can you remember all of this? Yes, retrovirus is the entire group, Rani. Retrovirus, leucovirus, we keep telling virus name, no, the entire group we are talking about. HIV is just one we are talking about, okay? Yes. No, we tell lentivirus also, part of lentivirus also, okay? Yes. Now, integration of viral DNA with the host DNA with the help of integrase, and with the help of protease, we will make viral proteins, not we, HIV will make viral proteins, and they will finally form the new virus. Is this clear to all of you? Yes or no? Yes, we are talking about a group, students, group. Okay, retrovirus, lentivirus. Okay. Yes, retrovirus. Lentivirus is actually a subtype of retrovirus. Okay, but we tell lentivirus. Generally, we tell lentivirus sometimes. Okay. There are subtypes, subgroups, no, like that. So much categories are there. Okay, chalo. Clear? Yes. Why is Vikram sad? Yes. So students, those who have that doubt, please understand retrovirus is a group of virus that has RNA as the genetic material, and they have the ability to convert RNA to DNA with the help of reverse transcriptase. Lentivirus also we use the word, that is actually a type under retrovirus. That under, just if you have a doubt about that, just understand. But isn't this very clear to all of you? First they will bind with the CD4 receptor. All this beautiful notes you will get it on the Telegram channel, don't worry, okay? This capsid will enter inside the host, convert the RNA into viral DNA with the help of reverse transcriptase. Host DNA will be inside, that is the nucleus. It will get inside the nucleus, it will use the integrase and it will add, can you see that? It will add the viral DNA with the host DNA, and then they will start making, our host DNA will get confused. What is happening? Okay, Vikram, what is over for you, dear? For him, from that time, game is over. Some game he's playing and his game is over means he's coming here and telling. Okay, now viral protein will be formed because our host DNA will not be in our control. It will be in the control of the HIV, and it will finally form the new viruses. Clear? Yes, fine. Moving forward, I taught you recently this in disorders, okay? So you people should be able to remember. This is the structure, students. As you can see, same thing only. See here, viral protein, viral RNA core, virus infects the normal cells, and as like when it affects the normal cells, they talk about the CD4 receptor, okay? And the viral RNA is introduced into the cell. Viral DNA is produced by reverse transcriptase, that will go and the viral DNA incorporates into the host genome. I just showed you that. New viral RNA is produced by the infected cells. Protein will be done, central dogma, DNA to RNA to protein. Protein will be formed, leading to the formation of new viruses. Okay, clear or not? Yes, this is your whole structure. I have explained it with this 4 steps, so you can use this 4 steps to understand it as well. Okay. That is done. Now, mode of transmission. All of you know that AIDS does not spread by sweat or by saliva, even though that is the fluid, but it will not spread that because the number of HIV is going to be very less. So students, AIDS mode of transmission is mainly by sexual contact with the infected person. That is, if you do not use a contraceptive, if you do not use any barrier method, and sexual intercourse has happened with multiple partners, that sexual contact with an infected person will lead to mode of transmission. So, sexual contact with an infected person, then by intravenous drug abuse. That means what? These people have shown in movies and all, no, with the syringe, these people will be sharing, okay, sharing the syringe and they will be injecting, okay, that also will lead to the blood mixing up, okay. Then student, with the help of blood transfusion, if the infected person's blood we are taking, then we will get AIDS, okay, blood transfusion. And it can also be spread with the help of tattoo, okay, tattoo, piercing, like where the needles are not changed or cleaned. Okay, piercings where there is direct contact with the blood. Then from blood banks, okay, here only I'll write that, blood banks. Then prenatal is there, that is from the mother to the child. Okay, that is if the mother has AIDS, then it can come to the child. Clear? Through the placenta. Yes, very good. Through the placenta, from infected mother to the fetus. Sexual contact, intravenous drug abuse, blood transfusion, blood bank, tattoo, piercing, any direct contact, okay. Always remember that this is all going to happen with fluid exchange, okay, fluid exchange. And this fluid exchange has to, mainly it will happen by blood, okay, or by semen, okay. These are the main 2 ways that the AIDS can be spread. Clear? AIDS can be spread. Now, how do you avoid it, students? What is the prevention? Prevention is obviously taking care. Okay, so the prevention will be, first of all, not to have sexual contact with people who are infected. Okay, so avoid Okay, avoid sexual contact with infected person. Okay, with infected person. Then the most important thing will be always use barrier methods. Okay, always use barrier methods. 1 second, students. Yes. Okay. Yeah. Okay. So avoid sexual contact with the infected person. Check. So students, basically there is no, like, you know, very nice. Okay, I will ignore that question. Yes, so basically there is no cure prevention, just that whatever you see here, no, just be careful of this. Don't do drugs. Okay, don't, when you're going for blood transfusion or to the blood bank, please check the history of the person who gave the blood. Then tattoo, piercing, please ask them if it is a new needle. And prenatal, we can't do anything, students. The mother has to be cautious, and if there is a checkup done, then the mother has to, you know, tell that, you know, the child birth is going to be difficult and all of that. Okay, so the prevention as such, there is nothing to write, just that you have to avoid such. Avoid no, prevent no. Okay, rest is, so prevention is better than cure. Have you heard that? Yes, and December 1st is considered as World AIDS Day. Okay, and many NGOs and many people are working on this, like your NACO. Oh students, NACO, you can remember like this only, no? Functions of your antibodies. Okay, so NACO, many NGOs are working behind this to make people aware, because this happens lot in the rural areas. Okay, in the rural areas. Clear? Yes, prevention is better than cure. Done. We'll go to cancer. Okay, now cancer. What do you know about cancer? Yeah, don't die out of ignorance. Just give me a minute, students. My family is calling. 1 second. Yes. Okay. Okay, yes, let's start with cancer. Ready? HIV is clear? No doubts, I hope. No doubts, right? Students, that's what I told you about the incubation period. Okay, 5 to 10 years, the 8th one. Okay, just remember that. Let me also check your. Okay, we have to do one more thing. No, we have to do this one. Prevention. Under prevention only we have to write about the diagnosis. Okay, we cannot forget that. That is, students, we can do ELISA. ELISA, which is enzyme linked immunosorbent assay. This is basically based on antigen antibody. Okay, antigen antibody principle. Okay, this is how it works. Then to confirm this, you can also do Western blotting, right? This question has come. To confirm this, confirmatory test is going to be Western blotting. Okay, Western blotting. These are the two techniques given in your NCERT. So please make a note of this. This is an antigen, antibody. Okay. Then students, treatment as such, there is no cure. All that you can do is art. Okay, art. That is no cure as such. Treatment is like no cure, but you can use art. That is anti-retroviral, like you can use any kind of therapy. Okay, anti-retroviral therapy. Anti-retroviral therapy. Fine. Okay, this we cannot miss. Okay, yes. Some examples I had made you write last time. Any of you remember? Acidotymidine. Okay, 1 second. Yeah. Ooh. Okay, acidotymidine. Okay. Acidotymidine. Clear? Yes. So partially effective, they can maybe, you know, somehow delay the reverse transcription from happening, or like, you know, stop the reverse transcription from happening for some time, but there is no complete cure. Okay, this is done. So students, always check your NCERT, because sometimes, you know, what happens in a flow, like we are teaching a lot of things, no, sometimes it will go. Okay, clear? I hope no doubts. Okay, fine. Now going to cancer. Cancer, yes. Okay, cancer students, what is happening? Normal cells and cancer cells, the biggest difference is that cancer cells has lost contact inhibition. That means I am standing here, another cell is here. When I touch this cell, I will know that I have to stop my division. But cancer cells have lost the contact inhibition. They know they do metastasis depending on the tumor, and these people also have abiogenesis. That means they can make their own blood vessels around them, take the nutrients from the normal cells. All of this they do. Okay, all of this they do. So cancer cells, if you compare with the normal cells, each of you give me one points. Normal cells have contact inhibition. They have contact inhibition, yes or no? Second thing, what? They have proper lifespan. Okay, that is, they have regulated or timed, okay, regulated lifespan. After some time, the cells will die. Okay, so it is completely timed and regulated. Then students, these people do not do continuous cell division. No, there is no uncontrolled cell division in them. So there is no uncontrolled cell division. No uncontrolled cell division. Clear? Yes. Okay, now when you talk about cancer cells on the other end, when we talk about cancer cells on the other end, what is going to happen in the cancer cells? These people have lost contact, okay, lost ability of contact inhibition. Ability of contact inhibition, and then these people do uncontrolled cell division, and students, these people form tumors. That is, mass of cells is called tumors or neoplast, right? Yes, they also do the stem cell thing. Okay, that we will come into. Carcinoma, all that we will come into. Okay, students, the main problem is what? They don't have this gateway. Like all of you know, no, in cell division you have CDK, cyclin dependent. So that gateway, these people will jump. What is gate actually? Those gateways are telling us that this is the toll. Now you have come from Mysore to Bangalore. You have to pay 70 rupees for car. That means what? It's a toll. Okay, and when you're crossing the toll, people are also checking your car. If you're carrying something that is not, you know, that's illegal, But this cancer cells will jump all the gateways and come out. That is the biggest problem. Clear or not? Yes. Yes, clear? Okay, very good. Yes, they break, so what I just told, they break cell regulatory mechanism. Students, I would change this word to programmed cell death. That is a better word. Okay. Programmed programmed cell death. Clear? This is your what cancer cells are actually doing. Okay. Cancer cells are actually doing. Okay, fine, done. Now moving forward, this cancer cells, especially as I told you, cancer cells have a mass. Okay, mass of cells are called as tumors. Mass of cells are called as tumor cells or neoplast. Okay, neoplast. And students, this tumor cells or neoplast can be into two types. What are they? Benign tumor. Okay, benign tumor and malignant tumor. Okay, benign tumor and malignant tumor. Benign tumor is confined to one place. They are confined to one place. They are not going to move, and most of the time they are not painful and they are not cancerous. Okay, malignant tumor on the other end does metastasis. Metastasis, that is, they move from one region to another region and they will start forming tumors. Okay, let's say malignant tumor is there, from one place to here they will make a tumor from here to here. That means these people are going to do metastasis and they also absorb the nutrients from the other cells. Okay, and these can be painful and most of the time they are cancerous. Okay, but these are not like that. Clear or not? Okay, benign tumor, these are confined to one region, they are not painful, most of the time they are not cancerous. Malignant tumor, they are cancerous, they are metastasis, that is, they go from one place to another and end up forming new tumors every single place. They form new tumors. Fine. Now, what should we know about cancer? Now, students, remember, cancer can be mainly caused because of physical, okay, physical, chemical, and biological reasons. Okay, these are the causative. Okay, if you want to study more about cancer, like your carcinoma, sarcoma, depending on from where they are, you know, the layering, that is a different thing. Okay, yes, physical, chemical and biological, very good. Yes, students, carcinoma is there, sarcoma is there and leukemia is there. Depending on the germ layers, you have sarcoma and carcinoma, as you can say, muscle and leukemia. Okay, so here look here, physical is mainly going to be because of ionizing, okay, ionizing, and because of radiation. So like your X-rays, I'll directly write it, ionizing and non-ionizing. So X-rays, gamma rays, okay, gamma rays, these are the main reason for your cancer causing, okay, cancer causing, that is ionizing, X-rays, gamma rays, and because of radiations, because of radiations. Then you have chemical, that is your tobacco, and biological, students, you have proto-oncogenes, proto-oncogenes. This proto-oncogenes, please understand, these people are present in the normal cells, but when they get activated, okay, when they get activated, they can become carcinogenic. Okay, when they get activated, they can become oncogenic, or we can tell cancer causing. Clear? Cancer causing. Now apoptosis, okay, apoptosis, and you should read NCERT again. These notes are just your reference, okay, but you should read NCERT. Okay, students, here remember the word apoptosis. Okay, there is no programmed cell death, metastasis, all these words are there. Okay, all these words are there. Clear? So here physical, X-rays, gamma rays and radiations, chemical, tobacco and smoke, and biological proto-oncogenes which are present in normal cells but they get activated. They can get activated because of any physical or biological, physical or chemical reasons. Okay. Now students, remember, cancer can be detected, okay, by many methods like your biopsy, okay, histopathology, by CT scan, that is computed tomography, from MRI if you want, 3D, and also this is magnetic resonance, and biopsy, histopathology, CT scan, MRI scan. Okay, these give 3D, and this is from magnetic, and they also give better accuracy. Okay, better accuracy. Clear? Yes, and also in cases of like your leukemia and all of that, you can also do bone marrow and blood test. Okay, blood test. These are the detection techniques. Clear? Fine. Okay. CT scan, MRI, biopsy, done. Okay. Now, since you people asked me to speed up, I'm speeding up. Treatment. Since I taught you people this very recently in disorders, you can just go check out that video. Students, treatment, first thing is surgery. In case of, where the tumor is confined to one position, we can do surgery. Then other thing is we have to do chemotherapy, send radiations, radiotherapy, or we can use alpha interferons. Okay, alpha interferons, because these are good at signaling the other cells that when they have a viral infection. So alpha interferons also we can use. Clear? That's a biological response modifier. Biological response modifier. Students, I am thinking, do you think we can do drugs now? Do you think another 15 minutes we should be able to do drugs? Should we do drugs, or can you people study by yourself? Tell me fast in the chat box. If not, we will do drugs. I have notes for it. Notes means I have NCERT lines. We can go through NCERT lines quickly, scam the NCERT lines. Okay, yes, BRM, biological response modifier. Okay, what will they do, students? They basically will activate the immune system. Okay, this interferons will activate the immune system. Okay, I don't feel nice teaching fast. Okay, I don't really like it because I feel like there's so much missing upon. Okay, but I hope students are understanding, because I recently did this in disorders, so you can actually check out the video as well, and my cancer and AIDS video also is on the chat box. Okay, yes, we will do. Okay, chalo, let's start drugs then. Cancer is clear. Any doubts in the cancer, please put a thumbs up. Anything we missed, please put it in the chat box. Okay, yes, do it ma'am, please do it ma'am. Okay, let's do it in 15 minutes. We will take 15 minutes so we can give the class to sir as well. Okay. Students, there are some anti-cancer drugs like Vinstrin, Vinblastin, okay, but it's not mentioned. So I have taught you before also this, okay. This is extracted from Vinca roseae. Okay, I've mentioned it here. Those who want extra information. Okay, chalo, let's do drugs. Okay, let's do it fast and furious. Okay, yes. So look here, drugs, opioids are the drugs. So what are drugs? Drug abuse is what you're studying, something that is used extremely more and not in good rates. It's going to be called as abuse. Okay, so here you're going to study opioids, cannabinoids and coca alkaloids. In this you're going to start with opioids. Now students, remember, drugs can be natural, semi-synthetic and synthetic. So in that category only you're going to study this. Okay, opioids are drugs which bind to a specific opioid receptor present in our central nervous system. This is the most important. Where are they affecting? Are they acting as a depressant or as they are acting as a stimulant? Okay, I cannot even talk this fast. All of you quickly like the video, those who joined right now, because I want all of you to enjoy the class, and please let me know in the comments how it was. Do not miss it. Last time many of you did not comment and I'm very upset about it. Okay, look here students, opioids are drugs which bind to specific opioid receptor present in the CNS. They're going to affect your central nervous system and gastrointestinal tract. These are the two regions that they are going to affect. Heroin, which is commonly called as smack, is chemically formed by diacetylation, okay, acetylmorphin, that is diacetylation of morphine. Clear? Okay, diacetylation of morphine, which is white, okay, which is white, odorless and bitter crystalline compound. So you should also know the color. You should also know the color, that is white, odorless, and it is also bitter crystalline. Now this is obtained by acetylation, okay, this is obtained by acetylation of morphine, which is extracted from the latex of poppy or Papaver somniferum, which we studied where students? MSP, we studied it in Apocarpus. Okay, Apocarpus in sexual reproduction and flowering plant. Okay, Dr. Siddharth. Okay, if you want to do the PYQs, just go to the description box. You have two links, human health and diseases and microbes. After my class, you can do the PYQs. Okay, generally taken by snorting or injection. Have you seen that? They keep it like this and they do like this. That is snorting, or by direct intravenous drug. Okay, injection. Heroin is a depressant. This is the question. Okay, heroin is a depressant and slows down your body functions. Students, questions will be asked directly. What are barbiturates? They will ask you, are they acting as depressants or stimulants? This is the question that is going to come. Okay, opioids, you have studied about heroin, which is also called as smack. It is derived from diacetylation of morphine. Okay, this is a semi-synthetic drug. Okay, semi-synthetic. Diacetylation of morphine. It is extracted from the latex of Papaver somniferum, which is commonly called as poppy plant. Clear? This is how the plant look. They can give you the plant picture and ask you to identify. Okay, identify. Oh yeah, structure is very important. Look here. They will ask you structure also. I saw in one of the PYQs. Look here students. This is the structure of your morphine. Okay, diacetylation of this will lead to formation of your heroin. Now look here. Morphine, no? So it will be M. Okay, M. M is the structure. Like this one, M should be there in your structure. That means this is morphine. Identify like this. Okay, identify like this. Now, cannabinoids are groups of chemicals which interact with cannabinoid receptors present principally in the brain. These fellow is going to be present in the brain and they are going to affect that. Natural cannabinoids are obtained by the inflorescence of plant called cannabis sativa. The flower, this cannabis sativa, from top to bottom, everything is used for making drugs. Okay, that means what? Flower, as well as top, flower top, leaves, inflorescence, resin, everything is used. Okay, flower tops, leaves, resins, every single thing is used of the plant. Okay, in various combination to produce marijuana, hashish, I don't even know if I'm pronouncing the name a little wrong. Okay, charas and ganja. Okay, these are the common ones that are used the most. Okay, these are commonly used by everyone in and around. Okay, yes, no problem Sukhmeet, you can continue from here. Okay, now students, it is important for you to know that these many combinations leads to the formation of marijuana, hashish, charas and ganja, which are commonly used by people, and it is extracted from cannabis sativa, whose inflorescence is used, leaf is used, resin is used, every single part of the flower is used. Okay, got it? Now, generally taken by inhalation or by oral injection, these are known for the effects of cardiovascular system of the body. That means they are going to affect the heart. Okay, because in the brain you have cardiovascular receptor spot, no? So it is going to affect your heart and it is going to slow down and make you feel more calm. Oh, nothing is going to happen. NEET and all is very easy. All this thought process is going to happen for people who take this kind of things. But tomorrow when you wake up, you're back to reality. NEET is everything. Your life is back to normal. Nothing has changed. Only your heartbeat has reduced. Clear? only your heartbeat is reduced. Please identify the structure, students. These people are going to affect the heart. So the structure is going to look like a heart. Okay, please study like this. Okay, my request, don't miss it out. They have given the diagram and they have asked you. I will show you the question. Look here, it looks like a heart. Okay, that means these people are going to affect the cardiovascular system, and this is gonna be extracted from cannabis sativa. Okay, cannabis sativa. This is cannabis sativa. Clear? Yes. Fine. Moving forward. Now, coca alkaloids. So, three categories you are studying: opioids, cannabinoids, and coca alkaloids. In opioids, you studied heroin, which is formed by diacetylation of morphine. In cannabinoids, you have studied about marijuana, hashish, ganja, and charas. And here you are studying about coca alkaloids, where you talk about cocaine. Okay, now coca alkaloids or cocaine. Okay, coca alkaloids or cocaine is from the coca plant. Okay, that is Erythroxylum coca. Native to South America, it interferes with the transport of neurotransmitter dopamine. Now students, dopamine is usually released when you get rewards. Okay, rewards. Okay, slow heartbeat does not reduce. I just meant it will calm you down, like in the sense as if you'll feel like, okay, nothing is going wrong in my life, I am the superior person. Okay, everything is perfect. Now look here, coca alkaloids or cocaine is very commonly used for people who feel very unhappy and they feel that, okay, I'm not very successful in my life, so when I take coke, it gives them a false positivity and false reward. They feel that they have achieved everything, but they have not achieved anything other than they have got drug abuse. Okay, so that means it is because they affect the dopamine. The dopamine rush comes when you are achieving something. When you get 700, 710, when you get 360 out of 360 in your biology, you will get a dopamine rush. That dopamine rush will be given by coke in a false way. Okay, false way. Cocaine, commonly called as coke or crack, is usually snorted. It has a potent stimulating action on the central nervous system, producing a sense. This question has come, I hope all of you remember. A sense of euphoria and increased energy. That means an extreme happiness, okay, an extreme level of feeling that, oh my God, I achieved everything. I am the superior. I am the CEO of the company. All that is just for the time that you are in that moment. After you are out of that moment, after you go to sleep and you come back, you will realize that you are still an employee and you still need to work, otherwise CEO will kick you out. Okay, so these people are going to give you an effect of hallucination, euphoria, extreme happiness. At the same time also, hallucination is something like, you know, you will see people flying, you will see multiple earth, you will see multiple tube lights, you will be able to see the sound and hear the light, something like that. Okay, yes, excessive dosage of cocaine causes hallucination. Okay, you will see yourself flying, you will see Spider-Man, Batman, everything flying around you. That is because of hallucination. Okay, other well-known plants with hallucinogenic properties are Atropa, Belladonna and Datura. Okay, this is Datura. Atropa, Belladonna and Datura. These days, cannabinoids are being abused by some sports persons. Sports persons use this for high energy because these people increase the energy. You feel like, yes, today only I will achieve Olympics, I will get into Guinness World Record. Nothing will happen, students. Hallucination will happen. So, don't do drugs. Okay? Don't think of doing drugs. Okay. Now, look here. Drugs like barbiturates, amphetamines and benzodiazepines are other some drugs. Okay, this question has come in 2019, so please make a note of this. Barbiturates, amphetamines and benzodiazepines are other similar drugs. Okay, some words does not come in my mouth, you have to forgive me for that. Okay, that are normally used as medicines to help patients cope with mental illness. Like normally these things are given by doctors. The doctors tell them that you can take it as sedatives. That means you are not getting sleep. Okay, not getting sleep, and since you are not getting sleep, doctor will tell that you can take this in this amount. But when you abuse it, that means, have you seen all in these movies, they take, instead of taking one sleeping pill, they take 20 sleeping pills and put it in their mouth and then they will sleep for, or they will die. So during this situation, it is an abuse. That means you're not doing according to the dosage given to you, but you're just randomly doing it. Okay, yes. Like depressions and insomnias are often abused. Understood? Morphine is a very effective. This is given as a painkiller. Morphine is a very effective sedative and a painkiller, and it's very useful in patients who have undergone surgery. Several plants, fruits and seeds having hallucinogenic properties have been used for hundreds of years in folk medicines, religious ceremonies and rituals all over the globe. When these are taken for a purpose other than medicinal use, it becomes abuse. Okay, it becomes abuse. Now students, all these barbiturates and all of this are used as sedatives and painkillers and recommended or given by doctors to patients. But what happens is when you take in excess amount, okay, excess amount, clear? Now, when these are taken for purpose other than the medicinal use or amounts frequently than the recommended one, then you will have physical, physiological and psychological functions. It constitutes drug abuse. Clear? Insomnia means you're not able to sleep, lack of sleep, and you're not able to sleep. That is the problem. Students, 3.5 hours I have been continuously talking. So now my tongue will start twisting and you people know it. Okay? Tongue twisting is a part of my life. Yes? Now smoking and alcohol abuse. Okay? Now, all this is there in your GK books. Okay? Tobacco has been used by humans for more than 400 years. It is smoked, chewed or used as a snuff. Tobacco contains a large amount of chemical substance, that is your nicotine, which is an alkaloid you would have studied in your biomolecules. Nicotine stimulates adrenal glands to release adrenaline and non-adrenaline into the blood circulation, both of which raise the blood pressure and increases heartbeat. Okay, increases heartbeat. Okay, now smoking is associated with increased incidence of cancer of lungs, urinary bladder, throat, bronchitis, emphysema important, coronary heart diseases, gastric ulcers, etc. Tobacco chewing is associated with increased risk of cancer of the oral cavities. Smoking increases carbon monoxide content in the blood. That means what? Carboxyhemoglobin. Okay? Carboxyhemoglobin. Carbaminohemoglobin should be formed. Instead of that, carboxyhemoglobin will be formed. Carboxyhemoglobin, okay, will lead to our body not having enough oxygen for circulation and reduces the concentration of hemo-bound oxygen. This causes oxygen deficiency in the blood. Now students, tobacco, that is your smoking, also is an abuse. Alcohol also is an abuse. That means what? People consume it first as fun, to be cool, then it becomes your status, then it becomes your lifestyle, and then it becomes an addiction. Okay? It all starts like this. First you try to be cool in front of your friends. One puff, ma'am, nothing is going to happen. Then it becomes as my status. I need to smoke because I am privileged. I'm elite. Okay. Then it becomes a lifestyle, and then it becomes something that you cannot get rid of. So nicotine is very, very harmful, and people chew it, people smoke it, and then comes alcohol. Now students, to prevent all of this, one thing, first of all, avoid peer pressure. As I told you, at your age, when you crack NEET and when you go to different colleges, you will meet all the kinds of people. You will meet the extremely rich kids, you will meet extremely poor kids, you will meet kids who have been very hardworking and come all the way there. Now, what happens is, when you meet different kinds of people will tell you different things. And NEET is such a kind of exam where age limit is not there. And you will notice that there are people at the age of 25, 26 sitting with you in your MBBS class. Now, those people would have used drugs, would have used smoking, would have used alcohol to get rid of tension or stress during that time. And they will influence you. So, what you have to do? Spread this knowledge of what is going to be problematic. Students, everything at the starting will look all wonderla and beautiful, but later when you admit in the hospital, when you go through it, and being an MBBS student, you should know where it is affecting, right? So, always I'm teaching the chapter, okay? Prevention, avoid peer pressure, go for counseling, talk to elders, take mentorship, ask help. These are the 5 techniques by which you can prevent it. Okay? Yes, self-discipline is the most important. If you don't know where to stop, please don't start. Simple as that. This is my motto for life. If I don't know when to stop, I will never start it. Because if I know that I cannot stop something, then I shouldn't even start it. Because if I'm not controlling myself, then what is the point? What is the whole point of mind, heart, everything connected to each other? Okay, so drop this in the comment. If you cannot stop, don't start. That is the line. Okay, yes. So alcohol abuse, I just told you in some words. Now will you do one question? Yes, do this one question, students. Last minute I thought we went rushing. I hope all of you understood, and I want to do justice to you people. Okay, yes, perfect. Now tell me the answer. Yes, yes. Wow, ma'am, wow, wow. NEET 2012 question. B, I told you how to remember. Now try to think of A. Identify A and B shown below and select the correct option. Okay, only correct option you have to tell. Okay. Okay, I know the answer. Do you know the answer? Yes. You're welcome, welcome. Advice is always good. Student, is that morphine? Is that morphine? Yes. So this is morphine. Okay, papaver somniferum and sedative painkiller. Here, what is the problem? Atropa, belladonna, no, datura. Here cannabinoids they have given. This is cannabis sativa. Okay, so it's option number this. So your marks are now 32. Okay, 32. And tell me this, which is particular drug you get from this? Yes. Yes, CC करके. I remember Diksha ma'am telling this. Yes, tell the answer for this also, just the name. Okay, datura, what does it give? Which is the drug that we extract from it? Both the answers you have to give. Drug that you extract from it. Yes, very good. Very good. This is datura. Okay, so you get plus 36. Okay, plus 36. Very good. Okay, done. Now tell me these two answers and then I'll give you a break and I'll inform sir as well. Okay, I took more time today. Ringworm in humans are caused by. Fast. Did you all understand everything? Was I too fast? Those students who are watching it recorded, please understand that we have been doing a long time. So the live students have requested me to go fast. But anything, every chapter, every topic is on the channel. Don't worry at all. Okay, yes. Ringworm is caused by 2010 question. Yes, all of you join my Telegram channel as well. Okay, you will get the notes as well as NEET English. Yes, perfect. The answer is going to be fungi. Ringworm is caused by fungi. And second one, answer is going to be typhoid. See students, typhoid is asked every single year. I don't know why they like typhoid so much. 44. Okay, 44 marks for GM today. And those who wants to do the quiz, students, this is the video that you're watching. Okay, 1 second. Yeah. Go forward, click on this link, okay, human health and diseases. When I give you a break, enter your phone number or your email ID, click on next and you will get a test. Okay, many of you send me your screenshot on the Instagram. Very good. So what you have to do, this is the video you are watching. Click on this link, All the students should do this. Okay, click on the link, go to next. Proper test will be there, question will be there. You have to select the option, go next, next, next. Finally, you will get a performance review, how fast you did and how much time you did. Everyone, during the break time, I'm going to give you another 15 minutes break. I think it's almost 9 o'clock. So, sir should start by, I think, 9:05, 9:10 and continue. Okay, and let me also tell you guys, there is a lot of offers happening on NEET Express Station 2. See, as you can see, all the course plans are here. Look at the price. Okay, you will get the NEET Express Station 2 at this price. Okay, it's going to start in 30th December and you will get the full course, that is combo plus test series, at this price. But now, okay, 1 second, yes. Okay, now I'll to show you the actual price here. Look here, you're going to get the entire thing at 3,500. Okay, 3,500. Clear? So make sure that you're joining the batch and also taking the test series, because you will get all the test series at your home, delivered at your home, in class doubt solving, practice test every Sunday, and assignment and notes. Okay, yes. Now go for a break, and during the break, please do the questions. Okay. So students, Biology 360 club members, you just finished day 2 with, I'm so sorry, reproduction unit, no, with, okay, after Baswa sir's class, please put biology in human welfare. So you can put biology in human welfare, BIHW marathon. You are officially unlocking the champion badge, and by the time you attend 3 sessions, you get the VIP badge. Okay, so yes, very good. Okay, very good. I'm not going to be doing microbes. Baswa sir is going to be doing microbes. So resume back at 9:10. Have your dinner and come. We are so sorry, students. We are not able to give you a longer dinner break. Okay. Yes. Let me just ask sir as well. Yes. By the time sir picks up, I will write the break timing. Okay. Resume back at 9:10. I will ask. Hello, sir. How long should it? Yeah. Please start. Okay. Yeah. Okay, sir. Yes. So, students, resume back at 9:10. It's not opening. Dr. Siddharth, use another ID to log in, or use an incognito mode, because every student will be trying to log in. Okay. 20 minutes. Okay. Wait 1 second. I'll just 9:15 students. 9:15. Okay. 1 second. Sir is calling. Sir, I'm still in the class. Okay. Yeah. Okay. 9:15. Okay. 9:15. 9:15 for microbes. Okay, microbes in human. Okay, thank you so much, students. I hope all of you enjoyed. Okay, and see you all in next Biology 360. Don't go anywhere. Definitely attend microbes. Lot of topics are connected to that. So I hope all of you enjoyed my class. Please let me know in the comments, and all the notes will be shared on Biology by Gopika and Vedantu NEET English. So see you all at 9:15 for Baswa sir's class. Till then, tata, bye bye.
So, hello students, class will be resuming at 9:15. So, all of you quickly run and get that break done. Eat something, drink a lot of water, and come back at exactly at let's say around 9:15. So, take a 15 minutes break. And I will see all of you exactly at 9:15 p.m. to start the microbes chapter. Okay, so with that being said, students, see you in next 15 minutes, all of you. Okay. Sound check, sound check, sound check. Hello students, sound check. So, hello students, welcome back to your own channel Vedantu NEET English. This is Baswaraj, your biology master teacher. Now students, quickly let me know in the chat right now if I am audible and visible to each and every one of you. Quickly let me know in the chat right now. Why do you let me know in the chat? Let me also add a message in Telegram. This is the Telegram. That is Baswaraj sir biology. Let me add a message in the Telegram that class has started. 1 second. Join soon. Let's go student, let's go. Okay, now all of you understood and completely learned the chapter of human health and disease. Now with that amazing chapter students, what is the most strongest takeaway? Tell me in the chat right now students, what is the most strongest takeaway you can get from that chapter? One thing is, don't do drugs. Yes, don't do drugs or don't do anything unnecessary. The second strongest takeaway is that microbes are bad. Yes or no? All of you tell me in the chat quickly. Microbes are bad. All of you tell me in the chat quickly, all of you. All of you. We learned that microbes are bad. But is it really true? Is it really true that microbes, all the microbes in the world are actually bad? Or do you also find some of the microbes which are actually helpful? Quickly let me know in the chat students. Right, quickly let me know in the chat. Are there any microbes which are actually useful to you? Are there any benefits to microbes? Should we actually care for them? The answer is yes. The answer is obviously yes. Let me ask you one more thing students. When I tell microbes, which organism is coming to your mind right now? Quickly tell me in the chat. Quickly. When I talk about microbes, when I say just the word microbes, what is exactly coming to your mind right now? Which organism? 99% of the time, the organism which is coming to your mind is a bacteria. Yes or no? All of you tell me. Something called as bacteria. Now, if you know bacteria students, tell me, bacteria belongs to which kingdom? Under your 5 kingdom classification given by R.H. Whitaker, the bacteria comes under which kingdom? Is it Monera? Is it Protista? Is it Animalia, Plantae or Fungi? You will tell me, sir, it is bacteria belongs to kingdom Monera. Okay, agree. Now what about amoeba and paramecium? What about amoeba and paramecium? Amoeba and paramecium are also microbes. Yes, they are microbes. That is, you cannot see them under naked eye. So if amoeba and paramecium are also microbes, can anyone tell me in the chat which kingdom do they belong to? Quickly tell me in the chat. amoeba and paramecia. Let me give a quick small clue to you. They belong under a group called as protozoans. Yes, protista. Absolutely. All of you very smart today. 5 marks extra for all of you, protista. Okay. So can I tell some organisms of kingdom Monera, kingdom Protista, as well as certain microscopic animals and plants also belong to microbes. All of you agree with that. Now what about viruses, students? Viruses. Do viruses also belong under microbes? The answer is yes again. Now tell me, think all of you, think very hard and tell me, virus belongs to which kingdom? All of you, this is the good question answer session where you get all the things out. Tell me, viruses belong to which kingdom? Now I want all of you want to think, start thinking. Viruses belong to which kingdom? Quickly in the chat, all of you, quickly. Viruses belong to which kingdom? You're like, no kingdom at all. Viruses have been removed out of the 5 kingdom classification. They are not a part of 5 kingdom classification also. Okay. So next time, students, when someone asks you, what is a microbe? You're supposed give me an example of microbe. Don't just say bacteria. Try including other organisms also, even fungi also. Okay. So the answer to this question, do all microbes make us sick? The answer is no. There are certain microorganisms which are helpful to us, and that is what this entire chapter is about, the useful bacteria or useful fungi. Okay. Now students, what is the weightage? What is the weightage of the chapter? Let me tell you, students, let me tell you. Before the change in syllabus, we used to hardly get around 1 or 2 questions. That is all. But after the change in syllabus, last year in NEET 2024, there were around 4 questions from this chapter. 4 questions. Generally, we get 1 or 2 questions, but last year in NEET 2024, there were 4 questions from this chapter itself. This chapter, 4 questions. So you cannot miss out. You cannot miss out on this chapter, even though it may be very easy, but there are very high chance that you might lose some marks. Okay. So with that being said, students, what is a microbe? Microbes are tiny living organisms that cannot be seen with the naked eye. Now, yes, microbe is very tiny. I cannot see with the naked eye. Then students, how do I see a microbe? For example, this is the pen right here. How can I tell confidently that this pen, on the surface of this pen, there are microbes? Tell me students, how can I confidently say that yes, if I take a sample from here and on this surface of a pen, there are microbes. In fact, on the hand, there are microbes. You're like, sir, with the help of microscope. That is correct. That is correct. With the help of microscope. That is on my hand, on the water which we drink. If it is not filtered, every single thing on this world is microbes, has microbes. Now students, do you think inside your body also do we have microbes? Anyone? Do you think inside our body also we have microbes? The answer is yes again. Let me tell you students, let me tell you. According to the recent research, inside our body, we have around 0.2 kg of bacteria. 0.2 kg, that is around 200 grams. 200 grams of bacteria inside our body, not anywhere else. 200 grams of bacteria inside our body. That is a lot. That is a lot of bacteria. Okay. So you find microbes everywhere. Now students, look at this beautiful diagram, background. Where else do you think microbes are? Yes, on the surface of the pen, I find microbes. Inside my body, I find microbes. But do you think we find microbes in the air? Answer is yes. Do you find microbes in the water? The answer is yes. Do you find microbes in the soil? The answer is yes. So air, water, soil. There is no place on earth where you cannot find microbes unless it is a sterilization area. Okay. So all the places we can find microbes growing there. Okay. Microbes are growing there. Now what about such acidic conditions, students? Such acidic conditions or deep thermal vents. Do you think, students, in such areas also we find microbes? Yes. There's certain bacteria called as archaebacteria. How many of you heard about archaebacteria? Raise your hands. Raise your hands, students. Raise your hands quickly. How many of you know what is archaebacteria? Raise your hands. Okay. Certain archaebacteria can be found in such hot springs. You can find. You can also find microbes in ice. In ice, let deep frozen -degree Celsius, you can find microbes everywhere. Everywhere it is there. It is omnipresent. Okay, not technically omnipresent, but is there everywhere. Right? All these locations we can find microbes. Now, students, all of you, let's look into the entire under microscope. For example, this is a simple compound microscope, right? And if I do 100x, I should be able to see certain type of bacteria at 1000x. I should be able to see fungi, microscopic in nature. I should be able to see protozoans, which are also microbes. I should be able to see viruses, which are also microbes. Not only viruses, even prions, viroids, all of these structures are microorganisms. If someone next time asks you, what are the examples? Don't just tell them the bacteria, tell them viroids, prions, and bacteria, protozoans, fungi. So all of these are different types of microbes which are given in your NCERT and also present in the entire environment. Okay, so next time onwards, you know what to tell. Don't just say bacteria. Talk about protozoans, amoeba and amoeba, paramecium. Talk about fungi, microscopic fungi, yeast. Talk about prions. Talk about viroids. Talk about viruses. Okay, so that is a small introduction to this particular chapter. Now there is a type of classification. Now, for example, I will tell some students are very serious in class. They will sit throughout. Some students will go out of the class. Okay. Now that is the basis of classification. Even in microbiology, we can classify the entire microbes into two broad categories. There is something called as gram-positive bacteria and gram-negative bacteria. Here is a simple example. If you can you see all of you? Here one is a gram-negative bacteria, one is a gram-positive bacteria. Okay. But apart from that, bacteria are also present in various shapes. For example, if you see the first one, it is said to be rod shape. Now students, can anyone tell me what is the other name of rod shape? Anyone? Rod shape is also called as, anyone? Rod shape is also called as, rod shape bacteria is also called as bacilli. Bacilli. Now what about this round on ball-like structure? The round on ball-like structure is called as caucae or spherical. Caucae or spherical. So rod shape is called as what? Rod shape is called as bacillus. Spherical is called as caucae. Caucae. Now apart from that, they have also given you one more diagram here. Now students, if you look at this bacillus rod shape bacteria, it has a flagella. It has a flagella. Now tell me in the chat, students, if it has flagella, does it show motility? True or false? Tell me. Can it show motility? Yes, sir. If it has flagella, it can show motility. Now apart from that, students, look at this diagram right here. Okay. Now here, the first microbe they have given you is called as a bacteriophage. Bacteriophage. Bacteriophage is the ultimate boss. You think bacteria are bad? If you think bacteria is bad, this is the dad. Okay, this is the dad of bacteria. Why? Because bacteriophage is a virus. Bacteriophage is a virus which can even eat bacteria. It is a big boss of which eats even bacteria also. Okay. The other example is adenovirus. It is a adenovirus which causes respiratory disorders. This is a TMV. This right here is a TMV, tobacco mosaic virus. So these are basic examples of different types of microbes given your NCERT. Here is a simple comparison also. Gram-negative, gram-positive bacteria. Okay. Now students, with that being said, how many of you have dogs in your house? How many of you have dog in your house? Raise your hands. Dog, cat, parrot, pigeon. Raise your hands quickly. How many of you have pets in your house basically? Pets in your house. Raise your hands. Raise your hands. Now if you have a, let's say like I have a dog in my house. Okay. Now if you love, if you have a dog in your house, at one point in your life you thought getting a separate bed for your dog. So I got a bed for my dog. But that stupid person, my stupid dog, he's not sleeping in his bed still. He's sleeping in my bed only. Okay. The whole point is every single animal needs a habitat where they survive. Okay. Like how he has a bed and everything, or he stays in the house and everything. But can I also try to grow this bacteria? See, I told you bacteria is found in soil, water, air, hydrothermal vents. But can you grow all of this bacteria inside a lab? Is it possible? The answer is yes. The answer is yes, you can grow the bacteria in the lab. But it cannot grow randomly anywhere. Just because it is randomly found everywhere in the road or in the sewage, in the air. But if you want to grow the bacteria in the lab, it is very specific. It needs a media. It becomes very picky. You know, right? Like I have around 4 or 5 dogs outside my house who are very happy sleeping on the road. I give them biscuit, they are very happy. But if I give the same biscuit to my dog, he'll spit it out. You're like, what are you giving me? Normal biscuit. If I give Parle-G, he'll spit it out. If I give him a cream biscuit, he'll eat. Okay. So even bacteria becomes very specific when you grow them in media. So if you want to grow the bacteria inside a lab, you need a separate nutritive media. This is a petri dish. On this petri dish, we have to grow media. So all of these are required for making a nutritive media. On this nutritive media, we can grow our bacteria. We can grow our bacteria here. Okay. Now, for fungus, there is a different requirement. For bacteria, also there is a different requirement. Okay, different requirement. Now, with that all being said, students, tell me, how many of you love curd rice? How many of you love curd rice? Raise your hands. Raise your hands. How many of you love curd rice? Tell me. I love curd rice. Without curd rice, there is no life. On curd rice, one nice apla, okay, papad, which you already call it apla. I call it apla in Kannada. Okay, a nice pickle. Okay, so that makes up the amazing curd rice. Now, in curd rice, apart from rice, which is the most important item, you tell me? Curd. Okay, so right now, students, right now, let me tell you. There are 2 ways which I'll teach you. There are 2 ways in which I can teach you this concept. One is, I can just read the lines of NCERT. I'm like, see, lactobacillus is there. Lactobacillus is a bacteria. It will convert milk to curd. That is one method. The other method is that, students. Let me show you. What if I tell you? What if I tell you, students? Okay. I will explain you this line in detail, and today you will understand how actually curd is made. You tell me, how do you want to learn? Do you want me to read this entire paragraph and tell me, or should I explain you in a way that you will truly understand how curd is made? Next time when you're eating curd, no, you'll think, oh, Baswa sir told this much. Okay, so I will explain you how does this entire thing happens, what is coagulation of proteins, what is actually happening. Okay, so can we start, students? Can we start? The most scientific way of making curd. Today you will learn. The most scientific way of making curd, not just by, just reading the lines of NCERT. Okay, full explanation. So if you promise me, students, you will comment on today's video, you will say you will stick with the bio club, you will say, yes sir, microbes learned very well today, then only I'll teach you the entire way of explaining, because I love explaining things. Okay, let's start. First of all, students, what is the main requirement to make a curd? Now all of you, imagine, you're making curd now, all of you are cooks now. So what you need to do is take a bowl. To this bowl, you need to add first milk. Add milk into it. Okay, milk is added now. Second step, you need to know what is present in the milk now. So if you look into a milk, magnify the milk. Okay, so inside the milk, there is obviously fat globules, which you see the yellow part. There is protein, 2 types of protein. One is called as whey protein, other one is called as casein protein. Remember this, casein and whey. This whey is used for your bodybuilders. Okay. Then there is something called as a sugar molecule called as lactose sugar is present. And then there is certain types of ions. So out of all this, all of us, we need to focus on 2 main things here. First one is the protein part, proteins. Second main thing is the lactose. These 2 are your main focus right now. Okay. So milk is there now. This is the milk. To this milk, I am adding some curd. Now this curd has, students, let me tell you, this curd right here is called as inoculum and also called as starter culture. Starter culture. In this small curd, there are millions of bacteria waiting, hungry bacteria waiting to eat. Okay, now I take this curd which has millions of bacteria like this, millions of lactobacillus bacteria, millions of bacteria, and we take, once we add this bacteria into the milk, once we add this bacteria into the milk has something called as lactose that is favorite for bacteria. This bacteria will go attack the lactose now. This bacteria will go attack the lactose. Attacking. I'll attack the lactose. Now what happens? Understand, all of you understand. Very focused. Lactose is a disaccharide, breaks down into glucose and galactose. Done. Glucose undergo a process called as glycolysis. Tell me, students, how many of you know how many steps are there in glycolysis? Tell me in the chat how many steps. In total, it undergoes 10 different steps in glycolysis. Okay. The end product is pyruvate. From lactose, it is getting converted to pyruvate. Now, pyruvate under anaerobic conditions, it gets converted to lactic acid. So, right now on our hand, we have the lactic acid on our hand. So, this lactose is completely broken down by the lactobacillus bacteria or the LAB. So, LAB is breaking the lactose into lactic acid. Till here, all of you understood? Understood. Now, students, understand. This lactic acid is an acid. It will decrease the pH eventually. So, the pH of milk will start to decrease as more and more lactose breakdown, more and more lactic acid is produced, the pH is dropping. It is dropping. Now, all of you aware, all of you know, my students, biomolecules have taught you. A protein cannot maintain the shape when the pH is changed or temperature is changed. Yes or no? So, as soon as the pH drops, the whey protein and casein protein will completely denature and they will start to coagulate. When all the casein protein starts to denature and they coagulate, that's how we get the curd. So this is the actual way the curd is being formed, not just the lines of NCERT. Okay. So did you enjoy, students? Did you enjoy? Did you understand how the curd is made? Now let me tell you one more thing here. Lactic acid bacteria is little cunning. When lactic acid bacteria is growing, it is making the entire surrounding acidic in nature. When it is making the entire surroundings acidic in nature, no other bacteria can grow there. How is the ability? It is growing in such an area that no other bacteria is able to grow. That is why lactic acid bacteria is the superior here. Okay. Now, students, how many of you know one more thing? There are some humans in this world who are lactose intolerant. How many of you heard about them? Lactose intolerant people. How many of you heard? I know so many of them. I know so many of my friends who are lactose intolerant. Okay. Now these people, they do a little bit of drama they do. What is the drama? They're like, no bro, milk I can't drink, curd I can't eat. Next time when you meet them, students, unless they have a very serious allergy, they can eat curd. Tell them next time. Why? Because in the milk there is lactose. But in the curd, there is no lactose. Lactose is almost consumed by the LAB, so it is safe. Even for lactose intolerant people, it is safe to eat the curd. Do not make excuse next time. Okay. So concept understood, all of you understood the concept. Lactose is there, but it is broken down by the LAB. Okay. So next time, catch hold of them. Catch hold of all the people who are telling you. Now, why curd is important? Curd is very nutritious, very tasty, obviously. Apart from that, students, it has increased nutrition value of vitamin B12, easy to digest, probiotic gut health, safe to consume by lactose intolerant. See, safe to consume by lactose intolerant people. Next time they should not do drama. Okay. Enhances texture and flavor. Nice. Now, students, nice, no? Nice, no? No. Now I will teach you how to make dosa. Ready? Ready, all of you? How to make idli and dosa. Ready, all of you? Want to learn? Like, to make idli and dosa, you need to add bacteria. But, let me ask you. How many of you have seen your mom or your dad or yourself? Do you ever secretly add any bacteria into the batter one night before? Never. Never. Right? Have you ever noticed that? Have you ever noticed that they will add some bacteria secretly, a nice powder of bacteria? Never. So if there is no bacteria added, how is the bacteria even developing here? Where is the bacteria coming from? Is it the nice hand flavor going into the mixture? No. Then where is the bacteria even coming from, students? I will tell you today. I will tell you today. Okay, so don't worry. If you do not know where the bacteria comes from, I will tell you today. Where is the bacteria coming from in the idli and dosa making? Now, first of all, does it happen in your house? Does it happen in your house, students? Let's say Saturday night, Sunday night, Big Boss is on. Sunday night, you're watching Big Boss or cricket match or some reality show you're watching on the TV. Whenever there is a crucial moment, suddenly from the kitchen one sound will come. What is that? One sound comes, right? How many of you heard that? And suddenly everything is inaudible in the house. Why? Because that time, one day before the actual idli or dosa making, the batter is being prepared in the house, one day before in the house. Now in that batter making, what exactly goes? In that batter making, what exactly goes, students? There is rice, there is water, there is something called as black gram, also called as urad dal. You might have heard urad dal. This is the urad dal, and nicely it is mixed, grinding for the batter, and goes, grinder happens. And next day, that time you have idli dosa. That time you have idli dosa batter. Next day becomes bigger. Now, students, I'll tell you two things here. Two things. Listen to me very carefully. It will change the way you think itself. Okay. Next day, what happens? First of all, you don't actually add the bacteria. Why? Because bacteria is present in that urad dal. Okay. In that urad dal we added, right? In that urad dal itself, there is a bacteria. So all of you know, bacteria is not needed. Okay. There we add a type of, there's a type of LAB present there. Second thing, students, if you make idli dosa in your house, when do they add the salt? Can anyone remind me? Like when I was making the batter, I never added the salt here. When do you add the salt technically? You add the salt in the next day. When the dosa batter is ready, before you make the dosa itself, you add the salt that time. Why? Because the salt will, if you add the salt one day before, if you add the salt in the night time only, it will not allow the bacteria to grow because change in the osmotic concentration. So it is very scientific, students. Next time when someone says to you, idli dosa is a science. The salt is added the next day to give the flavor because it will not affect the LAB now because LAB has grown significantly. Now I'll tell you one more thing. What will happen if you keep the batter, idli dosa batter, in the open for a long time? The batter will become what? The batter will become very tangy. You have tried tasting it? Have you tried tasting it? If you tried tasting the batter, little tangy. Why? Because the LAB is growing. The LAB is growing and it is creating more and more acid, lactic acid being produced. That is why it becomes more tangy. That is why we say keep the batter in the fridge. When you keep it in the fridge, bacteria is stopping to grow. So much of science, no? So much of science. So let me tell you, students, this is how I'll teach today. This is how I will teach today. I will not make you just mug up the lines of NCERT and tell some random things and go. No. My students should understand entire thing. Okay. No. So in the entire of all this, dokla making, pickles, cheese, buttermilk, idli dosa, flavored yogurt, every single place LAB is used. Lactic acid bacteria is used everywhere. Okay. It is used everywhere. Now students, next thing is cheese. Okay. I call this Tom and Jerry cheese. How many of you seen this? Tom and Jerry. Tom and Jerry cheese. This Tom and Jerry cheese is also called as Swiss cheese. Okay, it is my dream to eat it, but I have never tried eating it. It is called as Swiss cheese. Now students, something is advanced here. In making Swiss cheese, we don't use one bacteria, we use two bacteria here. One more extra bacteria is coming, which is very shy, I'll tell you now. Okay, shy bacteria is coming. So understand, same process goes on here, same process. Lactose is broken down to lactic acid by lactobacillus. Same thing as before, same thing as before. Okay. Lactic acid now, students, lactic acid now is used by one more bacteria. Lactic acid now is used by one more bacteria called as Propionibacterium shermani. What bacteria is that? Propionibacterium shermani is a second type of bacteria which converts the lactic acid into propionic acid and carbon dioxide. Carbon dioxide. So that is why when this cheese is being made, these small holes here. These small holes are nothing but they were trapped. Who were trapped there? CO2, carbon dioxide was trapped there. And when the cheese started aging, the CO2 came out from there, and when the CO2 started coming out from the chamber, the gaps will produce. So all these small gaps you see here, those gaps or small holes are because of CO2 initially being trapped there. Okay, so CO2 is initially trapped there, then later on CO2 became completely free and the holes were formed. Okay, so remember Propionibacteria shermani. Now students, now do you want to learn how to make bread now? Raise your hands, raise your hands. Tell me students, do you want to learn how to make bread now? Today I will teach you a little cooking class today, cooking class. Shermani those, yeah. Do you want to learn how to make bread now? How to make bread? Okay, I'll teach you how to make bread now. And what is the science behind bread? Like what is the science, actual science behind bread making? Okay, now understand, cooking class. Okay, now what do we need to make the bread? There are few requirements. For example, to one container you have to add flour, sugar, salt, oil and water. Okay, then we have to do something called as kneading. To this kneading, you should also add one more fungi called as yeast. It is also called as baker's yeast. It is called as baker's yeast, called as Saccharomyces cerevisiae. I'll tell that later on. Saccharomyces cerevisiae. Okay. Now you understand the science behind it. Okay. Now students, when the flour is being added, in the flour, the atta, there is a molecule called as gluten. You might have heard about gluten, right? I want gluten free. I don't want gluten in my life. Gluten free Starbucks they add, no, gluten free. The gluten molecule is there in the wheat flour. Okay. Now once you add the gluten, right, this gluten molecule, when it reacts with water, when you add water to it, when you make a add water to it, tongue twisted became suddenly. Okay. This gluten will make a mesh. This gluten will make a mesh like this, mesh. Okay. Now in this mesh, the CO2 is being trapped here. CO2 is being trapped. Now students, when you buy the bread, on the piece of bread, do you see the small holes here? When you're eating, like why this hole? One mine has big holes. You see that, right? Holes are there on the bread. That holes are because of in this entire mesh, CO2 is again trapped here. Okay, so that is the making of bread. The bread here. Okay, so the yeast cells are used here. Yeast is used, and as the CO2 evolves, the bread becomes bigger and bigger. Okay, so donuts, bread, croissant, all of these are basically made with the help of yeast called as Saccharomyces cerevisiae. Okay, Saccharomyces cerevisiae. Okay, now students, I don't like this. Okay, how many of you tell me now? You tell me how many of you want to eat this? How many of you want to eat blue cheese? I don't know why. I'm not personally, it doesn't look very appetizing to me. But tell me, how many of you want to eat blue cheese? Blue cheese. If you want to eat blue cheese, I will tell you what is blue cheese now. You will be shocked. You will be shocked after I tell you what exactly is a blue cheese. Try eating now later on, okay? Okay, tell me how you want to eat blue cheese. Students, let me tell you. This blue part, you think, no, it is not blueberry. This blue part is not blueberry. It is not blueberry flavor. Nope. The blue part, there is a fungus. The blue part growing there is a actual live fungus called as Penicillium roquefort. It is called as Penicillium roquefort is the fungus which is growing there. Now, how do they do it? How do they do it? Listen to me. First, they pasteurize the milk. They add rennet to it. Rennet help in coagulation of proteins, curd formation. Then they strain it. Then they add the fungus into it. Allow the cheese to age. The fungus will grow there. The blue color part of the fungus is growing there. And apparently, some people in this world, very exquisite taste apparently, very exquisite, I can't even say it, very fancy word, exquisite taste. Okay, so blue part of the fungus, apparently it gives a very, I don't know what taste, students, I don't even know what taste, but some taste it gives. But if you are daredevil, try it someday. Okay, not for me. Blue cheese. Now students, what is the other use of household in bacteria? Bacteria can be used to help in even in the keeping the things concealed. Okay, even pickles jar, everywhere bacteria is added. Beer making, wine making, everywhere bacteria is there. Okay. Now students, how many of you been to Kerala? Tell me students, how many of you been to Kerala? Raise your hands. If you've been to Kerala, you will see such nice palm trees everywhere. Nice palm trees. And on this nice palm trees, you will see one uncle. On this nice palm trees, you will see one uncle growing on top of it like this, and he will go there. He'll make a small cut here. From the small cut, something will come out. What will come out? The sap will come out. Okay, sap is basically the blood of your plant. It has all the nutrients in it. Now, he'll keep a pot there. Once you keep the pot there, all the sap is added into the pot. Okay. Now, in this pot, what happens? In this pot, because of the microorganisms present in his uncle's hand, in the pot, in the air, all the microorganisms will consume the sap and convert alcohol, and that is called freshly served, not even aged, freshly served toddy. So toddy is a fermented traditional white juice. I will call them white juice. Okay, it's white juice. Okay, fermentation of toddy. So with that, we come to the end of first half. Okay, So what we learned till now, students? Till now, we learned that to make curd, what do we need, students? Which bacteria do we need? Let's do a quick revision. To make curd, what do we need? We need lactobacillus. We need lactobacillus, also called as LAB, lactic acid bacteria. Yes, lactic acid bacteria. Done. Now, what about to make bread? What do we need to make the bread? We need yeast. We need baker's yeast. The other name of baker's yeast, it is called as Saccharomyces cerevisiae. Okay, Saccharomyces cerevisiae. Okay, then cheese, tell me students, cheese. What do we need? To make the cheese also we need LAB. All of you remember LAB is required to make normal cheese, but to make Swiss cheese, what do you need? To make Swiss cheese, what do we need? We need a bacteria called as what, students? Propionibacterium shermani. We need Propionibacterium shermani. Then last one was your toddy, not toddy. Let's say what is that, students? What is that? Roquefort cheese. To make Roquefort cheese, we need something called as Penicillium Roquefort. Okay, so all of this you should remember. All of this you should remember. All of you will remember, right? Easy, no? Students, tell me, is it fun? This chapter is very fun, no? This chapter is very fun chapter. Similarly, I'll teach you the entire thing today. Very smoothly, I'll teach you. We will learn how trust me, when I teach you STP, sewage treatment plant, when I teach you STP, you'll be like, wow, how does it even happen? Okay, so you'll be enjoying that also. Okay, done. Now here's a quick question. Let's see who can tell me the answer. Let's see who can tell me the answer. Quick question to all of you. Quick question to all of you. Conversion of milk to curd improves its nutrition value by increasing the amount of. What increases? I want everyone to keep a track of your marks. If you tell the right answer, 1 mark to you. 1 mark to you. What is the answer? Vitamin B12. Exactly. Vitamin B12. Okay. So remember that 1 mark. Remember that 1 mark to all of you, because there are many PYQs today. Many PYQs today. You will enjoy it thoroughly today. Okay. So with that first part of the class over, now we go to the second part, that is called as microbes in industrial products. Ready, all of you? Ready? All of you? Quick some fire emoji in the chat. And all of you quickly smash that like button. What is this? No likes for me today. All of you quickly smash that like button. Quickly, all of you. Keep it as 1 mark. Otherwise, you'll forget off. 4 marks not. Just keep it as 1 mark. Okay. If you take 4, 4, 8, 8, 1 mark enough. You do a simple addition. 1 plus 1. Okay. Now, students, industrial products. Now, here we'll be studying 3 main categories. 3 main categories. Okay. 3 main categories is fermented beverages, antibiotics, and chemicals. There are certain chemicals which can be obtained. Okay, certain chemicals which can be obtained for industrial applications. Okay, these 3 are the main category for today. Okay, 3 main category. The first one is called as the yellow juice category. Okay, yellow juice. Okay, adult juice. Now here, the first thing which I'm talking about is the fermented beverages, also called as alcohol. Also called as alcohol. Now there are multiple types of alcohol in the market. Like how Gopika ma'am told, don't drink, so don't smoke, don't drink also. Okay, that is beer, wine, brandy, rum, whiskey. Different types of options available in the market. There might be more also which I have not written, for obvious reasons. Okay, now here, students, do you remember in the bread making there was a yeast involved? The same yeast has a twin brother. The same yeast has a twin brother. Who is the twin brother? The twin brother is called as brewer's yeast now. It is called as brewer's yeast, also called as Saccharomyces cerevisiae. Same thing. Dual role, bro. Dual role. Okay? Dual role. Now students, I'll ask you a very simple thing. Okay? In your house, when you're preparing food, do you use a small cooker or do you use a large utensil? If you want to make a rice for 4 people, you will use a small cooker in your house. Yes or no? Okay. 5 people, same cooker. Now, what if some relatives are coming to your house without even telling you? Worst kind, okay? Without even telling you, relatives are coming to your house. Randomly, they are coming to your house. They're like, I will stay in your house for 10 days. Now, can you make the food in the same small container? Or your mother will bring out the big weapons. The large container will come. Remember, have you seen them? They use in marriages, in weddings they use, right? Weddings. The big one, like you can make on 10 kg biryani in it, 100 kg biryani in it. Okay, the big ones will come out. Now, similarly, based on the requirement, the utensil size also increased. It increased. Same concept also applies here. When you want to make the alcohol for yourself, small container. For the whole country, big container. So the big container is called as fermentator. In this fermentator, the alcohol is prepared in a very, very large quantity for everyone to be happy. Okay. Now, large container. Now, students, small flashback. How many of you remember alcoholic fermentation? Raise your hands. Tell me, alcoholic fermentation, raise your hands. I'll come to everyone's house. Only if you call, only if you make nice food, I'm coming to everyone's house. Don't worry. Okay. Now, how many of you remember alcoholic fermentation? In alcoholic fermentation, what happens? First step, glucose is converted to pyruvate. 2 molecules, 10 steps, 10 steps. Okay, 10 steps. Converted to pyruvate. Now what happens here, students? Pyruvate gets converted to what, students, tell me? Pyruvate gets converted to basically the, tell me, alcohol plus, tell me. Pyruvate gets converted to ethanol plus CO2. Now I will check your memory. Flashback time. Tell me what are the enzymes present here, enzymes? Tell me. The first enzyme is pyruvate decarboxylase. Pyruvate decarboxylase, yes. The other enzyme is, what is the other enzyme? It is a dehydrogenase enzyme. The other enzyme is alcohol dehydrogenase. Students, tell me in the chat, do you want forgot only now. Do you want respiration in plants chapter one more time in 3D? Let me know in the chat if you want to do in 3D one more time. Okay, I will make it happen. Now this is the basic process which is happening. This entire process happens inside the fermentator. See, bacteria is happy and is releasing CO2. It is releasing CO2. Okay, so inside the fermentator, fermentation happens and alcohol is made. Now students, let me tell you, I told you earlier there are multiple types of alcohol. The reason there is multiple types is because there are different types of ingredients involved in every single alcohol. For example, see here, wheat, barley, sugarcane, potato, grapes, all of these can be used as the raw materials to make alcohol, raw materials. And each one of them will result in a different type of alcohol. For example, wheat will result in beer, and barley will result in whiskey, sugar, rum, grapes and potato, wine and vodka. So every single type of ingredient will result in a different type of alcohol. Okay, remember that. Now apart from that, students, some alcohol are much more stronger. If you remember the chapter respiration in plants, I told you there the maximum concentration of alcohol is 13% naturally available. That is, if it goes above 13%, what happens? Yeast poisoning happens and all the yeast will die. But in the market right now, if you go, there are alcohols which are more extreme concentration of alcohol, 30%, 50%, 70%. I don't know who is drinking 70 now. 71 hole will become in your esophagus and liver. Hole will become. 70% alcohol is there. Okay, 70% alcohol. But how do we achieve this? How do we achieve the 70%? How do we achieve the 40%? A process called as a distillation. Do you want to learn how it happens, students? Do you want to learn? Do you want to learn how exactly distillation happens? Let me teach you, students. Okay. Now, for example, here I have one container. Here I have one more container. Okay. Here I have, let's take I have normal alcohol. Normal alcohol is here, which has alcohol plus water. It has water content in it. Okay. Now, for example, here I'm adding a tube. Here I'll add a tube. Okay, I'll add a tube here. Let's take here is the alcohol. Now, what happens here? Understand. There is a process called as boiling point. Now, students, tell me in the chat, what is the boiling point of water? How many of you know? The boiling point of water is around 100 degrees Celsius. 100 degrees centigrade or Celsius. Okay. Boiling point of alcohol is around, let's say, depending upon the alcohol concentration, around 40 to 50 degrees Celsius, or even lesser or more in some cases. Okay. Now, students, here to this spot, I am adding some flames. Flames I'm adding. Now, tell me, students, all of you tell me, which liquid will evaporate faster? Tell me, all of you, which liquid will evaporate faster? Boiling point of alcohol is comparatively lesser than alcohol. So, alcohol will evaporate faster. evaporate faster. So alcohol will start to evaporate faster. So let's take all the alcohol, some pink color alcohol we have here, okay. Alcohol will evaporate and come this side. And all the water will be left here. So eventually what happens? Alcohol without water is achieved. Now in reality it doesn't happen so easily. There is something called as fractional distillation, which you learn in chemistry, not here. So that method is actually used. Okay. So all the pink color alcohol is coming here, and remaining water is left here. And that's how the distillation happens, and we get more concentration of alcohol with less water in it. Students, again, disclaimer, don't try it at home. Do not try it at home, let me tell you now only. Later on, I don't want any complaints from any parents telling that Baswa sir is teaching so many information, I will try it in the house. Do not try. Okay, do not try all this at home. Disclaimer I should add next time. Okay, the process is called as distillation. Separation and removal of alcohol from the other components from alcoholic beverages, that is called as distillation. Now here there is something called as non-distillated and distillated. Now what is non-distillated, students? Non-distillated is basically beer and wine. They are non-distillated. Without distillation, they are not distillation. Now what about distillated? In distillated, the other thing comes in. Whiskey, rum, vodka, and all of that will come here. All that comes here. That is distillated. Okay, that is all that is distillated. Okay. Now students, that was fermented beverages, basically yellow juice. Over now. All of you understood? All of you understood how it is made? Fermenters. All of you understood the chemical reaction? Understood. Distillate, non-distillated? Understood. Also we learnt about different type of ingredients, raw materials? Understood. Okay. Now we go to something called as antibiotic. Antibiotics. Antibiotics are basically anti means against life. They're against life. Okay, against life. They're anti-bio. Anti means against, and bio means life. So they're basically against life. Now students, how many of you remember the chapter, the ecology chapter, where I taught you about different types of relationships? Not that one. I'm telling you about commensalism, amensalism, mutualism, predation, competition. How many of you remember that? Raise your hands. How many of you remember that? Raise your hands quickly. How many of you remember that? Remember? Yes, you remember. Now in there we learnt about a biological relationship called as amensalism. Now what happens in amensalism, students? In amensalism, one species is affected, other species is neither benefited nor affected. One is simply chilling, other one is getting destroyed. There was no example given in your NCERT. There is no example given. But the example for that is here. Antibiotics. Example, penicillium. Let's take a penicillium for example. Penicillium notatum. Penicillium is killing all the bacteria. Penicillium is not getting any benefit from it, nor it is getting harmed, but it is harming the bacteria. So this is an example. Antibiotics is an example of amensalism. Okay, remember that point. Okay, amensalism. Okay, so to bacteria it is against life. But for us humans it is pro-life, because we love antibiotics. Keep taking antibiotics. Okay, keep taking antibiotics. Now students, understand what exactly is the definition of antibiotics. Now I want everyone to write this down so you can remember it better. Okay, it is not banned in India, don't worry. Antibiotics I also take, all of you take. Okay, now these are basically chemical substances. They are chemical substances. The chemical substances which are produced by the microbes. Interesting part is they're produced by microbes, also affecting the microbes. Produced by the microbes, okay, that can kill or retard, right, or retard the growth of other microbes. Have you tell no? Humans bring down other humans. Humans bring down other humans. Microbes are the same thing. One microbe is killing other microbe. One microbe is killing other microbe. But let me tell you, students, let me tell you a small story here. Let me tell you a small story. Ready? Ready. How did the microbes antibiotics came into existence? Like, how did we get to know about antibiotics? Like, one microbe can be helpful to you. The answer is very simple. The answer is by accident. Okay, by laziness, basically by laziness. Okay, how exactly? Now here is a great scientist called as Alexander Fleming. Namaskar all of you, quickly give the namaskar to the scientist, respect. Okay, Alexander Fleming was working on a bacteria called as Staphylococci. Right, Staphylococci. When he was working on Staphylococci, this is called as a petri dish. He kept some of the petri dish unwashed in the sink. Okay, he kept some of the petri dish unwashed like that. Later on, he noticed that the Staphylococci is not growing only. Even though he's not cleaning the petri dish, even though he's just keeping the petri dish as such there, the bacteria is not growing. Why? Because then he noticed that there was a fungi growing there. The fungi, later he called it as Penicillium notatum. Penicillium was growing there, and when the penicillium is growing, around the penicillium, no other bacteria is growing. It's like very interesting, very interesting. Okay, that is how antibiotics work. That's how the Penicillium notatum came into existence. Penicillium notatum came into existence, but the entire work on usage of antibiotics was later worked and established by two other scientists called as here Ernest Chain, MS Chain and Howard Florey. These are two scientists who worked on penicillium, and later on, all the three scientists, Alexander Fleming, Ernest Chain, and Howard Florey, all the three of them got the Nobel Prize in 1945. And because of this antibiotic, it saved hundreds and thousands of lives during World War II. Okay, World War II. Now, are there different types of antibiotics? Yes, there are different types of antibiotics. Some of you might have heard about them, not given in NCERT. Cephalosporin. You might have heard about gentamicin and streptomycin. These two are very famous people, very famous celebrities. These are the celebrities among antibiotics, streptomycin and gentamicin. Whatever happens, take one gentamicin or everything good. Okay. So antibiotics can be useful. Now, how are they useful? They are useful against different type of diseases like plague, diphtheria called as Gulghotu, whooping cough called as Kali Khasi, leprosy called as Kushtrog. So all of these are different types of bacterial diseases which can be stopped with the help of antibiotics. Okay. So with that, antibiotics is also over. Two concepts done. Students, tell me how smooth the chapter is going. Smooth like butter, no? How smooth you're learning today? Very easy it is. This chapter is very easy. All you need to do is remember certain examples and concepts which I'm telling you today, and you're good to go. You learn from me today, you will never forget all small stories. Okay. Now we come to something called as chemicals. Now chemicals, there are three main categories. Okay. This all the one fact I added fact. Streptomyces. Streptomyces is the famous person here. Streptomyces is the largest antibody producing genus. Remember that. Streptomycin. Not given NCERT, but small thing. Okay. Now chemicals, there are three main type of chemicals here. The first one is called as your basic chemicals, which can be used for different purposes. Enzymes and bioactive molecules. All three are very important. For the next 10 minutes, I want everyone to focus, ultimate focus now, maximum effort. Next 10 minutes, you will learn these three things with examples today only, and you can solve 50% of PYQs right now after this. Can we start? Students? Can we start? All of you. And quickly smash the like button. Yes. Okay. The first one is chemicals. When I say chemicals, I mean acids. There are different type of acids derived from bacteria and fungi. Now, which is bacteria, which is fungi is also important to note. There is something called, students, listen to me. There is something called as, all of you. First of all, lactic acid. Lactic acid is by your lactobacillus. Lactobacillus or LAB. Okay, lactobacillus. Okay, lactobacillus is the lactobacillus, first one, lactic acid. Clostridium butylicum is butyric acid. Acetic acid is by Acetobacter aceti, there is a bacterium. Citric acid by Aspergillus niger, which is a fungi. Now this paragraph right here, student, this paragraph right here, if you remember this paragraph, you will be able to solve almost, let's say, around 10 to 12 PYQs very easily. Now let's play a small game of photographic memory. I'll give you exactly 30 seconds and then I'll ask you a question. You should be able to tell me the example of each one of them. Ready? Bacterium, fungi. Butyric acid, Clostridium butylicum. Lactic acid, Lactobacillus. Acetic acid, Acetobacter aceti. Citric acid, Aspergillus niger. Exactly 30 seconds. Time starts now. Try to completely stare at it. Stare and remember. Okay. 3, 2, 1. Time starts. 30 seconds. Done. Okay. Now, time starts now. The first question. Acetic acid is produced by dash. Acetic acid is produced by dash. Acetic acid is produced by dash. Acetobacter aceti. Okay. Citric acid is produced by. Citric acid is produced by. Citric acid is produced by. Quickly in the chat. Citric acid is produced by. Citric acid is produced by Aspergillus niger. Very good. Lactic acid is produced by. Lactic acid is produced by, students. Lactic acid is produced by Lactobacillus. Done. Very simple. Very simple. Okay. So you have to remember this. Next thing is called as enzymes. Now there are different type of enzymes here. 3 main type of enzymes. You need to know the function. That is very important. For example, lipase. Students, lipase is a detergent. Basically, lipase will help in what? It will help in removing the oils and stains. Completely remove the oils and stains. Okay. So lipase will help in removing it is a type of detergent. Remember. Next thing is called as your pectinase and proteases. Pectinase and proteases, students, are called as clarifying agent. Now what do I mean by that? Now, if you buy fruity from outside and if you make a mango juice in the house, the first major difference you will see that mango juice in the house is very less sugar, and also mango juice in the house, which you make it in the house, will have fibers, will have different types of things in it. But the same fruity which you buy from the market is very clear. Very clear it will be. Why? Because of lipase, because of protease and pectinases. Protease and pectinases is added in fruity to make the juice very clear. Okay. So it will protease and pectinases will completely denature all the pectin and everything there. Okay. Will make it completely clear. Okay. So clarified bottle juice. Enzyme which cleaves pectin and peptide bonds respectively, clarifying the juice. Clarify means it is making it more clear. Okay, clear. Super fruity. Now the next third one, the third different enzyme is called as streptokinase. Streptokinase is obtained from? Streptokinase is obtained from Streptococcus. Very important point. Obtained from Streptococcus. Now, what is the main function here? The main function is it acts as a clot buster. What is a clot buster? If this is the artery or a vein, if this is a blood vessel, in this blood vessel, sometimes what happens is the blood gets clogged like this, which can cause heart attack, myocardial infarction. Now, when we add streptokinase, when we take a tablet of streptokinase, it will go and it will burst open the clot and the flow of blood will happen properly. So, removes blood clots from patients who have undergone myocardial infarction. Okay, so clot buster. Now, those were the three different types of enzymes: lipases, protease and pectinases, as well as streptokinases. Now, the next thing is called as bioactive molecules. Two examples. Just two examples. Okay, two examples. The first one is called as cyclosporine A. Let me give a simple example. Okay. Now imagine tomorrow I go to buy an iPhone. Tomorrow I want to go buy an iPhone. Okay. But I don't have any money. What do? I'll sell my kidney. I'll give my kidney. I'll sell it. I'll get an iPhone. But later on I get to know I need my kidney. Okay. Now how do I get my kidney back? I will ask my sister. I'll call her. One kidney, place one transfer on Zepto one kidney. She'll give me her kidney. Okay. Now I'll take her kidney and fix it in mine. But my body is rejecting it. Graft rejection. Okay. My body is rejecting the kidney. I'm like, no, I don't want to come to you. Okay. So in such cases, what happens? In such cases, to make sure the organ is not saying tata bye bye, the organ is not rejecting, you decrease your immunity. That is called as immunosuppressant. So you take a tablet called as, or take a liquid, basically any form, you take a immunosuppressant, we will decrease the immunity and the process of transfer can happen very easily. Okay. So what happens is cyclosporine A, it's an immunosuppressant. It is obtained from Trichoderma polysporum, which is a fungus. So Trichoderma polysporum fungus, cyclosporine A, immunosuppressant. Students, how beautiful is it written? Can you see? So please download these notes. It is available on Telegram channel, Baswat Biology. Download the notes. Please write these notes on your own also. Okay, so well it is written, so well it is mentioned. Next one is called as statins. Statin. Now, what is a statin? What does it do? Where does it come from? Statin comes from a fungus again called as Monascus purpurus. Monascus purpurus is a fungus which gives statin. Now, statin will help in what? Statin will help in blood cholesterol lowering agent. It will decrease all the cholesterol in the body. Done. Students, here concept is very important, this much, but it is your job to remember this statin, Monascus purpurus. This you have to remember at any cost. Okay. Done, students? Done? Can we solve some questions now? Some questions based on whatever we learned right now. Can we solve some questions before we go ahead to STP? Can we start? All of you? Yes. The first question on the board, let me tell you, all the questions are based on the examples. Chill questions. Okay. First question on the board, students. Clostridium botulicum, butyric acid. So A is going to be 3. So try solving before me. Okay. Clostridium botulicum is butyric acid. Saccharomyces is used in ethanol preparation. Okay. Trichoderma polysporum we just now learnt is cyclosporine A. Streptococcus is streptokinin, which is the blood clot. Done. So A is 3, B is 1, answer is 2. Done. Next question. Saccharomyces cerevisiae, again ethanol. Same question, no? No, different question. Cerevisiae A is 1. Aspergillus niger is citric acid, it's a fungus. Trichoderma polysporum is cyclosporine A. Clostridium botulicum is butyric acid. A is 3, B is 1. Answer is 1. Answer is 1. Yes. Answer is 1 this time. Yes or no? A is 3, B is 1, C is going to be 4 and D is going to be 2. Yes, answer is 1. Answer is 1 now. So, such questions can come. Such questions will come, and this is a NEET 2024 question. NEET 2024 question this is. Okay. Next question, students. Identify the microorganism which is responsible for the production of immunosuppressive molecule cyclosporin A. Tell me. I'll give you exactly 5 seconds on this question. 5 seconds. 5, 4, 3, 2, 1. Trichoderma polysporum. Same thing, students. Do you realize? Same thing is repeating everywhere. Repeating everywhere. Next question. Okay, match the following again. Do it before me. Try doing this before me, students. Aspergillus citric acid, Acetobacter acetic acid, Clostridium butyric acid, lactic acid. So A is going to be 3, B is going to be 1. No, B is going to be 1. So it's option number 3. Number 3. Done. Done. Yes or no? Done. A is going to be 3, B is going to be 1. So that's the only option given there. Okay. Easy question, easy answer. Next question, students. Which of the following commercially blood cholesterol lowering agent? Commercial blood cholesterol lowering agent. Let me answer quickly. Answer in the chat. Quick answer in the chat. Quick answer in the chat, all of you. 5. I'll give you 5 seconds. 5, 4, 3, 2, 1. Time's up. Statins, lowering agent. Okay. Here. Same thing we learned here. Next question. For commercial and industrial production of citric acid, which of the following microbes will be used? Citric acid. Quickly in the chat. Quickly answer in the chat. Quick answer in the chat. 5, 4, 3, 2, 1. Aspergillus niger. Very simple. It's a fungi. Cyclosporin A used immunosuppressant agent produced from. Again, same thing. Same thing is coming. Cyclosporin A. Trichoderma polysporum. Trichoderma polysporum. Same thing, students. Match the following list of microbes or what they're used for. Okay. Saccharomyces cerevisiae. Students, I'll give you 5 seconds. Answer this question again one more time. Saccharomyces cerevisiae, ethanol. Monoascus purpurus, resting agent. Trichoderma polysporum, immunosuppressant. Propionibacterium shermani is wrapping of Swiss cheese. So A is going to be 3, B is going to be 4, C is going to be 1, D is going to be 2. Option number 1. Option number 1. Yes or no? Yes or no, students? Yes? Done. Very easy question. So that was the end of the first part of the chapter. The first part of the chapter is over now. Now we come to something called as STP, also called as sewage treatment plant. Now tell me, honestly students, tell me, how many of you find this part, particularly this part, difficult to understand? Raise your hands. Raise your hands. All of you, quickly. How many of you find this part of the chapter difficult to understand? This part of the chapter, little difficult to understand. Telling that, sir, I have read STP so many times, but I do not really understand it, or I can't really tell how does each and each step by step happens. Tell me, all of you. Because today I'll be teaching you in a way that you will remember forever, because I'll be teaching you in a stepwise manner, which is very important to understand in such a way. Okay, can we start? Can we start now? Can we start? Yes. Now, first of all, tell me, students, what is the wastewater? Wastewater is called as sewage water, which is coming from your house, my house, your houses, from city and village, from everywhere the wastewater is coming, and this wastewater is sent to the natural body. But let me tell you, this sewage is very dangerous. This sewage is very, very dangerous because the sewage will has organic matter microbes, it has pathogens. Now tell me students, can I directly send the wastewater into the water body without treating? It will be very bad. For example, students, if I don't send it unfiltered, if I send it unfiltered, what happens? Imagine here, near to this water body, there is one more family living here. Near to this water body, if there is a village here, all the village will get dirty water. They'll get water with a lot of microbes. So you cannot send it directly. It is not possible. Because if you send directly without treating it, unfit for use, waterborne diseases, everything can be transferred. Everything can be transferred. So that is why before sending the water directly, you need to introduce something called as sewage treatment plant. Now students, tell me, how many of you have seen a sewage treatment plant in real or in photo? How many of you seen? In real or photo? Tell me. Tell me students. In real or photo? This right here is a sewage treatment plant. Can you see? This right here is the massive sewage treatment plant. We can see multiple different types of tanks here. And finally, when the water is finally filtered, entire water is removed to the nearby water body here. It is sent here directly. So this right here is called as a sewage treatment plant. And I'm going to explain the entire process in the next 15 minutes. Next 15 minutes, you will understand the entire process. Ready? Ready students? Ready? Entire 15 minutes. Next 15 minutes. I will be dividing the entire sewage treatment plant into 5 different chambers. Chamber 1 or tank 1, tank 2, tank 3, tank 4 and tank 5. Every single tank is one step, and after every single step, the effluent is transferred to the next one. Okay? Every single step it is transferred to the next one by one. Before we understand that, the entire sewage treatment plant, students, let me tell you, the entire sewage treatment plant can be broadly divided into 2 categories. One is called as physical treatment, one is called as physical or primary treatment. The other one is called as secondary or biological treatment. I'll repeat again. The first one is called as primary treatment, also called as physical removal. The second one is called as secondary treatment, also called as biological treatment. Okay. So students, understand, out of these 5 different chambers, the first 2 are primary treatment. 3, 4, 5. The remaining 3 are biological treatment, also called as the remaining 3 are secondary treatment. Okay, so remember that primary treatment, secondary treatment. Now what exactly happens in the primary treatment? Understand all of you. In the case of primary treatment, the first tank right now, let's say wastewater is accumulated. We have taken some wastewater and we pour the wastewater into the first tank. The first tank here, students, the first tank is called as filtration tank. The first tank is called what, students? The first tank is called as filtration tank. Now understand, students, understand. Okay. Have you noticed in your house, when your mother is making chapati or any type of chapati with maida or anything, the chapati, we take the flour and we allow it to go through a mesh. Yes, we allow it to go through a mesh. You might have also seen when they are making buildings, they keep a large mesh or over which a soil is added, where large particles are remaining on the top and the fine particles are coming out. Same principle is applied here. Same principle. And this principle is called as sequential filtration. It is called as what? Sequential filtration. Understand. In filtration tank, there are small grids like this. Grids. As we go from here to here, the grids are becoming smaller. Can you see? Smaller. So what happens then? Larger particles are held here, more smaller particles, least smaller particles. By the time we come here, we can only find something like coins and everything, coins or small, even more smaller particles like a cap, all that will be present here. So as we go from here to here, the size of the particles left behind is becoming smaller and smaller and smaller. That is called as sequential filtration. It is called as sequential filtration. Removal of floating solid debris method is called as sequential filtration. That is the first tank. Done, students. Done. First tank is done. All of you. Tank 1 done, all of you. Tank 1 is called as filtration tank. Physically, some particles are removed out. All the notebook, bat, cricket bat, ball, everything is removed out here. Now understand the second tank. The second tank is called a sedimentation tank. It is called a sedimentation tank. Now what exactly happens here? In sedimentation tank, we use gravity. With the help of gravity, the entire dirt will start to settle down. All the pebbles, rocks, even the tiny of the rocks will start to settle down because of gravity. Can you see here? In sedimentation tank, all the pebbles and girls will start to settle down. And whatever is remaining on the top is called as the effluent. What is going down is called as primary sludge. The primary sludge is taken and again sent back to the filtration tank for more and more filtration. And the top part called as effluent is sent to the next chamber or the next tank. Okay. So until here, what happens? Supernatant floating sewage is sent to the next tank. So filtration tank, sedimentation tank. In these two tanks, all the physical removal of particles is over. Done. Removal, done. Now remaining sludge is again sent back for filtration. Now remaining top part called as effluent, the effluent is sent to the next chamber. The effluent is sent to the next chamber now. Follow, all of you follow. Okay, follow through. Sedimentation is sent to the next chamber. Till here, primary treatment is over. From now onwards, from 3, 4, 5, all the 3 is called as biological treatment. They are called as biological treatment. See, here in sedimentation, the solid debris is there on the below part. It is again sent back to the primary, again filtration. The top part called as supernatant, is sent to the next chamber. Okay. Now, from now onwards, second treatment starts. From next one. Okay. Removal of organic matter from the sewage through microbial action. Now onwards, we are using microbes to our advantage. We are using microbes power now. Okay. Now what happens? Next tank is called as aeration tank. Now what exactly happens in aeration tank? In aeration tank, obviously there is a lot of air. Okay. Can you see here? There will be something called as fans, agitators. They will rotate and air is released. Now tell me, students, if there is aerobic bacteria present here, the aerobic bacteria will consume all the organic matter. It will grow. Now imagine you only. If I take you and I'll lock you in one chocolate factory, next day morning you'll finish entire thing. Same thing is happening here. Now here, like how I locked you in a chocolate factory, I am locking all the bacteria here with all the organic matter present here. Okay. Now with the help of aeration, there is more air. So there is air here, there is food here, there is microbe here. So microbe will eat all the organic matter and it will keep on growing. Okay. Can you see here? The microbe will keep on growing. When the microbes keep on growing, here, mechanical agitation and pumping of air increase the number of aerobic bacteria, microbes. Now here on top they make a mesh-like structure. The mesh-like structure is called as a flock. So all the fungi and all the bacteria, they make a separate layer on top. That layer is called as flock. Okay. The flock is what? Mesh-like structure form the mass of bacteria associated with fungal filaments. So flock is basically bacteria plus fungus filament. That is called as flock. there is a flock formation on top. Okay, now that is aeration. Okay, microbial flocks consume the major part of organic matter. So all the, can you see here, all the organic matter will be consumed by the bacteria. Now I'll teach you one small concept here. Listen to me very, very carefully. One small concept. Okay, small concept coming up. What is this BOD now? What is BOD? And how does the BOD affects the purity of water? Understand, very important, understand all of you. Microbes consuming the organic matter decrease the level of BOD. So if there is very complex, let me teach in a very simple manner. Let me teach in a very simple manner. See this, listen to me. Listen to me, student, listen to me. Let's take, I have two water, you have one water here, second water. Let's say this is water A, this is water B. Okay. This water is less polluted. Less polluted. This one is more polluted. More polluted. Okay. Less polluted, more polluted. So if it is less pollution means, can I tell organic matter is less here? Organic matter is less? It is less. In more pollution, tell me now. In more pollution, organic matter is more. Okay. Now students, if organic matter is less here, will the bacteria be more or less? If food only is very less here, if the food itself is very less, bacteria also will be very less here. Bacteria will be very less. What about here? The food is more, bacteria is more. Bacteria is more here. Now students, you tell me, if there is less bacteria here, what will be the demand of oxygen? Will it be more or less? Less bacteria, less demand. Less O2 demand. What about here? In this case, demand of oxygen is more here. It is more here. If demand of oxygen is less, biochemical oxygen demand BOD is less here. What about here? Biochemical oxygen demand is more here. It is more here. Now you understand the definition one more time. Now you understand the definition one more time. Microbes consume the organic matter. More pollution, more organic matter. Consume organic matter, decreases the level of BOD. So they will decrease the level of, see, if organic matter is decreased, BOD will also be decreased eventually because less organic matter. See this. Amount of oxygen consumed by bacteria for oxidation of all organic matter in 1 liter of water. So any water body has certain amount of organic matter in it. More the organic matter, more is the BOD. Less organic matter, less is the BOD. Okay, less is the BOD. Less is the BOD. Now what is biochemical oxygen demand? Amount of oxygen consumed by the bacteria. So how much of oxygen is being consumed by the bacteria? To break the organic matter. To break the organic matter. Now tell me students, if BOD is very high, it means that pollution is very high, yes or no? Tell me. Tell me all of you. If BOD is very high, it means pollution is also very high? Very high. It means more organic matter is there? It is there. This right now is the sewage. For example, this now is the sewage. What is our goal? Our goal in STP, sewage treatment plant, our goal in sewage treatment plant is to from here to here. What do I have to do? From here to here we have to become. That is, we have to decrease the BOD level. When we decrease the BOD level, what happens? Organic matter decreases. Demand for oxygen also decreases. How do we do that? In this entire polluted water, we add bacteria. Bacteria will eat the organic matter. Bacteria will eat the organic matter and eventually will decrease the organic matter. If organic matter is decreasing, BOD is decreasing. That is our entire goal. The entire goal of STP, students, the entire goal of STP is to decrease the BOD. Okay. See here. Biochemical oxygen demand is indirectly measured for organic matter present in the water. Greater the BOD of wastewater, higher is the amount of organic matter, greater is the pollution potential of the water. A sewage water is treated until the BOD is reduced. Very important point. Why? Because less polluted water will have less BOD. I hope all of you understood the small concept of BOD. BOD is basically how much of oxygen is required by the bacteria to destroy the organic matter. Okay, to destroy the organic matter. Hope all of you understood the concept of BOD. Okay. Now students, now this was aeration tank done. Aeration tank is done. Now from aeration tank, next tank is called as settling tank. It is called as settling tank. Okay, settling tank. In settling tank, all the bacteria will start to settle down. Okay. Now all the bacteria and the fungi filaments, the flock which is settling down is called as activated sludge now. Remember, in the starting we learnt in the sedimentation. Later in sedimentation we learnt whatever was settling down, we call that as primary sludge. But right now, whatever is settling down is called as activated sludge. Why? Because this sludge right here has a lot of bacteria in it, and all this bacteria is very hungry bacteria. Okay, hungry bacteria. Now whatever is present on the bottom is called as activated sludge. Whatever is present on the top is called as treated effluent. Okay, now here is very important to understand. Now remember students, when we wanted to make curd, what did we do? To make curd from milk, we added some curd as inoculum in the starting to make the entire process going on. To this random milk, we added some curd and the milk became curd eventually. Same thing happens here. In this aeration tank, how does the bacteria come into existence? What we do is, from settling tank, we take some amount of activated sludge. Okay. Some amount of activated sludge is taken and it is sent back to aeration tank. Why? Because to make the bacteria culture constant there. Okay, it is keep on adding back. Remaining top part is sent out here. Top part is sent out for the water body. Now listen to me very carefully, the next point here. Some amount is sent back here. Now what about the majority of the part? Majority of this bacteria, students, majority of this bacteria is sent to one more tank. See here. Majority of this bacteria is sent to one more tank. See, the top part is sent out. The top part is sent out from settling tank. The majority of the bacteria, some amount of bacteria is sent back. Majority of this bacteria is sent to one more tank. The last tank is called as what? The last tank is called as anaerobic sludge digester tank. Now what exactly happens here? In anaerobic sludge digester tank, there is no oxygen. There is absolutely no oxygen here. If there is no oxygen here right now, can the aerobic bacteria survive here? Tell me yes or no in the chat. Can the aerobic bacteria survive here? Tell me, all of you. Tell me, can the aerobic bacteria survive here? Is it possible? Will they survive? The answer is no. They cannot survive at all. Not possible. So they will not survive at all here. Aerobic bacteria will completely die. Now what is remaining here? The dead matter of aerobic bacteria is eaten by anaerobic bacteria. So all the anaerobic bacteria will feed on the aerobic bacteria. Next level we are doing, students. Till now we use the bacteria to break down the organic matter. We use the aerobic bacteria to break down the organic matter. But as soon as we do not require them, we will kill them now. So what happens here is, all the aerobic bacteria is dying because of no oxygen, and all the anaerobic bacteria will consume the matter, and this anaerobic bacteria, students, this anaerobic bacteria will release some gases such as methane, carbon dioxide, and hydrogen sulfide. All of these gases are called as biogas. They are called as biogas. Okay. So what happens, students? What happened here? Understand? Filtration, sequential filtration, sedimentation, aeration, settling tank. Some part is sent back, some amount is sent here. Anaerobic sludge digester tank. Biogas is released. Biogas is released. Now the question arises is that, do we even need to cure this anaerobic bacteria, or do we just release them in the wild? We can release them. Why? Because as soon as you release anaerobic bacteria, they will anyway die. So we don't have to take care of them. So this is the entire sewage treatment plant. Primary treatment, secondary treatment. Students, tell me, with the help of flow chart, doesn't it seem very easy now? Doesn't seem very easy? Filtration, sequential filtration, sedimentation, aeration. In aeration tank, what happens? In aeration tank, BOD decreases completely here. Settling tank again, settle downs. Activated sludge is sent back. Set something here. Okay. Don't worry, human health will be sent back to you again. By tomorrow, human health will be available. It takes some time for it to come back. Okay. But students, there is a problem now. There is a problem in India. The biggest problem in India is the population. We have a lot of population, lot of waste being generated every single day, but very less STP. Biggest problem. So, and this major problem was identified near the rivers such as Yamuna and Ganga. Okay. So Yamuna and Ganga, lot of waste was being generated. To counteract this, government took an action that is called as Ganga Action Plan and Yamuna Action Plan. In Ganga and Yamuna Action Plan, it was made a rule that near this famous holistic rivers, we need to have STP. So all the waste cannot be released into the river or lake. It has to be treated first, then only it has to be sent here. Okay, see here. So build sewage treatment plants in the large numbers. No untreated sewage water must enter the major rivers. This was the main action plan given there. Ganga and Yamuna Action Plan. And that is the entire sewage treatment plant. Remaining the superintendent here is sent to the natural water body like that. Primary treatment, secondary treatment. Primary treatment is physical, second treatment is with the help of bacteria. Okay. So I hope all of you understood this one now. Students, if you want these notes, all these notes will be available in the Telegram. I am telling you now only. In Telegram will be available, and also on NEET English Telegram. Both the Telegram will be available to all of you. Okay. Now with that, we come to small question. Can anyone solve this question? Which of the following is sewage treatment plant removes suspended solids? Click me the answer in the chat. I'll drink some water in that gap. Which of the following is in sewage treatment plant removes suspended solids? Basically, physical removal. Primary treatment. A high value of BOD indicates that. What is the indication? High value of BOD. Tell me. If there is high BOD means what? High BOD means water is highly polluted. Yes, sir. Measuring biochemical oxygen demand is a method used for. What is the use of this? Okay. Estimating the amount of organic matter in sewage water. Working out efficiency of oil-driven automobile engines. Measuring the activity of Saccharomyces. Working out the efficiency of RBC. So what is the answer? First one. This right here is a AIPMT question 2012. Okay. Now students, with that we come to next topic. With that we come to the next topic called as gobar gas. Now students, tell me, how many of you notice this? In village side, how many of you have noticed this? There will be one auntie, old granny, she will be taking gobar and she will be slapping it on the wall. How many of you seen this in real life? Because I have seen, whenever I go to native, I've seen this in native. How many of you seen this? Yes. Seen or not seen? Seen or not seen, students? Seen or not seen? You have seen, I have also seen. Very good. Okay. This entire gobar is obtained from a cow. Like a cow is there. From the cow, all the gobar is obtained. Okay. Now, what is so special about this gobar? In the grass, there is cellulose. This entire cellulose is taken into the gut. In the rumen, in the rumen of this cow, there is a bacteria called as methanogens. These methanogens are very special. They are anaerobic bacteria which can take the cellulose and produce biogas. Produce the biogas. When the cow dung is produced, this cow dung has bacteria called as methanogens. These methanogens, example is methanobacterium is an example. This bacterium can produce under anaerobic respiration, very important. Anaerobic respiration, not aerobic. Under anaerobic respiration, it produces the biogas or gobar gas. Gobar gas has methane, CO2 as well as H2, all the different components of biogas. So all this biogas can be used for cooking, fuel or lighting also. Now all this gobar gas, students, the main question is, how can we use this gobar gas in a large scale? Obviously, humans are greedy. We got to know we are getting some gobar gas from this cow dung. How can we use this cow dung to large scale? Like, how can we make it in large scale and make a maximum profit? There came an invention like this. This is called as gobar gas plant. Gobar gas plant. Like, I have never seen this in real life, but I have seen only in pictures. This is called as a gobar gas plant. Okay, gobar gas plant. Okay, greedy gobar gas plant. Now, what exactly happens here? Before what exactly happens here, who developed all of this? There were two organizations who developed this. One is called as Indian Agriculture Research Institute, IARI, and also Khadi and Village Industry Commission, KVIC. These are two different agencies or institutions which benefited, who were the pioneers in the entire work of development of gobar gas plant. Okay. Now, to understand what happens. Understand. Okay. This is a diagram given in your NCERT also. The first thing here we need to understand here is, this is called a slurry tank. This is called a digester where main work happens. This is called as a sludge tank where all the waste comes out here. All the waste comes out here. Okay, all the waste comes out here. And this is called as a gas holder. Why gas holder? Because you might have seen, students, when you're trying to boil water or milk, when you keep a plate on top, does the plate keep coming out like this because of the more gas inside or more water vapors coming out? Same way, to make sure the biogas is not escaping out, there is a gas holder on top to make sure the gas is not escaping out, and there is only a single outlet like this. Now understand what is the procedure here. Okay, Okay, the first thing is called as, you want to make something called a slurry. What is a slurry? Okay, slurry is nothing but cow dung plus water. You take cow dung, add water to it, nicely make one paste, chutney, cow dung chutney this is, okay. Make a chutney and add the chutney into this slurry tank, here. Okay. Now once we add the slurry tank, the entire slurry will move everywhere. Now what exactly happens in digester? Understand. Inside the digester, we have the bacteria that is called as methanogens. Okay, methanogens or methanobacteria. There is methanobacteria, there is a methane, there is a methanobacterium, there is cellulosic material from the cow dung is there, Then there is methane gas, CO2 gas, hydrogen gas. These three are the biogas produced as a byproduct. When cellulose is completely eaten by the methanobacteria, these three are the byproducts here coming out. Can you see here? The gas are being produced, and all the gas produced here goes out from the outlet, and remaining waste is removed as a sludge in the sludge tank. And all this waste removed here, all the waste which is removed here, all of this waste can be used as a fertilizer later on. Fertilizer. And that is the story of biogas. How simple this is, how simple it is, students, how simple it is. This is how simple biology is, if you understand with the help of diagrams. Okay. See here, cowdung plus water, slurry tank, digester. In the digester there is biogas production, outlet, remaining part is sent to sludge tank. That is all. Simple flowchart to understand. Simple flowchart to understand. Here is a small question, solve the question. Small question on the board. Small question on the board. Sangeeta, which village? Tell me which village? Recently someone asked, what is my native? My native is North Karnataka. Okay, just saying. Okay. My next 20 more minutes over, don't worry. Which of the following statements about methanogens is not correct? They can be used for production of biogas, correct? Correct? Incorrect. Correct. Why, students? Why is C incorrect, students? Can anyone tell me? What is the imposter here? Who is the missing agent here? Who is the... Tell me. Bellari. Oh, nice. This is the imposter here. This is the imposter here. Okay. Done. Next question for all of you. What gas? What gas? Hey, I am all right. Why are you telling me that? I am all right only. I am feeling all good only. Don't worry. Don't jinx anything. I am feeling all right. I'm a little tired. That's all. Okay. Next question. What gas are produced in anaerobic sludge digester? Gases. Methane, hydrogen sulfide, and CO2. Very easy question. Very easy question. Very easy question. Solve this question, all of you. Let's see. Okay. Why will I be sleepy? Okay. Select the correct statement from the following. Biogas is produced by the activity of aerobic bacteria on animal waste. Wrong. Word here. Methanobacterium is aerobic bacteria. Again, wrong. Aerobic bacteria. Biogas, commonly called as gobar gas, is pure methane. Wrong. It is not pure methane because there is CO2, there is H2S, everything is there. H2 is there. Activated sludge medium in settling tank of sewage treatment plant is rich in source of aerobic bacteria. Yes. Aerobic bacteria. Correct. So all these are wrong. This is correct. Okay. Next question. The technology for biogas production was developed in India mainly due to the efforts of. Who were the two main institutes who took upon themselves? They're like, I will do the work. Tell me. Two institutes. Students, smash the like button, all of you. I want to see 400 likes. I want to see 400 likes today itself. Smash the like button. Share the video to everyone possible. I want this video to be shared to every single one of your friends, telling that yes, there are two teachers in biology called as Baswaraj sir and Gopika ma'am who are doing extreme hard work for all of you to clear your NEET 2025. So share this video to every single one of your friends. Okay. Both A and B. Exactly. So with that, students, we come to the end of the chapter. We come to the end of the chapter today. End of the chapter. So we learned about microbes in household. Yes, we learned that. Industrial, chemicals, organic acid and everything. Sewage treatment, done. Production of biogas, done. The only thing remaining is how can we use microbes in the usage of farming now. Farming. Can we start? Students? Can we start now? Farming now. I don't have friends, sir. Oh my God, heavy. I am your friend, don't worry. I am here. We are not that very old from you. I can also be your friend easily. Nice. Now students, this is my sister's bottle. If she finds out, I'm dead. Okay. Now students, can we start the last part of today's chapter, that is called as farming. Here there are two main applications, let me tell you. The first one, how can we use microbes for biocontrol, and how can we use microbes as a fertilizer. Two main things. Okay. Let's start. Start karo sir. Start. Wait. Patience. Let me drink some water. Okay. Okay. Now the first thing, how can we use in farming? Like all of you know, the traditional farming, all of you know. In traditional farming, the main usage of chemicals is there, and when we add a lot of chemicals, it is very bad. For example, all the vegetables which you eat are very harmful to you in many levels because of the pesticide, fertilizers, chemical, weedicides, everything nice coating is there for you. But there's a new concept called as organic farming. And let me tell you students, half of it is very fake also. Don't believe everything. In organic farming, let me tell you what they do is, they don't use chemicals, but they rather use biological agents to farm. Let me tell you. Okay. Agrochemicals too expensive for farming to afford. Okay. Agrochemicals cause environmental and health related problems. Lot of them. Okay. The alternative is organic farming. The problem is this is very expensive also. Very expensive. You take normal apple, organic apple, very expensive. Okay. Problem is there. Now two main methods, two main things. Biocontrol agents, biofertilizer. I will teach you biocontrol agent with the help of a simple example. Listen to me very carefully. Listen to me. For example, let's take who wants to volunteer. Who wants to volunteer? Raise your hands. Any one of you raise your hands. Who wants to volunteer? Anyone? Let's see who wants to volunteer. Parvati, I'm not tired, don't worry. Let's take Yashu. Okay. Let's take Yashu. Okay. Yashu has a lot of land. Very rich person he is. Rich big person. Okay. Yashu is very rich and big person here. Okay. This big person has a lot of field. Okay, lot of field. Okay. In this lot of field he has a lot of grasshopper growing there. The problem is this grasshopper is eating all the crops. Very bad. Now there are two main things you can do. Either you can add some chemicals and kill the grasshopper, or we can bring a frog. Who will only and only eat the grasshopper. Smart no? Tell me which category has to go. If you use chemicals, all the chemicals will be in the soil, damaging the soil. But he brings that frog, a nice golden frog. Okay, it will eat only the grasshopper. Grasshopper population is reduced. Problem solved. That entire concept is applied in biocontrol agent. The whole concept is that predation concept. There are certain insects which we don't want. Bring the predator. The predator do the job. We don't have to do anything. One more simple example I'll tell you. For example, there is a deer. The deer is coming to your farm too much, destroying everything. Bring one tiger and keep. The tiger will eat all the deer. Eat you also. Okay. But don't do that. Now here we need to replace the frog. With microbes. Okay. Now, see, microbes used to control pest disease based on natural predator, do not cause pollution to the environment, do not eradicate pests but keep them under the manageable levels. Very important, because we need biodiversity. We can't just keep on killing every single organism. We can't keep killing it. So we need to keep it manageable levels, not kill everything. Okay. Now see here, one simple comparison. Harmful, beneficial to species. Here under biological controls, harmful pests will decrease, but beneficial species are maintained. So the main advantage of biocontrol is useful things are maintained and not destroyed. Okay, not destroyed here. Here both are destroyed. Okay. So the first example, bacteria as biocontrol agent. How many of you heard about BT cotton? Okay, how many of you heard about BT cotton? All very famous. This person is in BT cotton. What happens is There's a bacteria called as Bacillus thuringiensis. Fancy, no? Bacillus thuringiensis is a bacteria. It will go and kill all the different types of butterfly larvae. Only larvae, not the adult moth. Only larvae. Okay. Now what happens is this BT, right? It has been used in plants such as BT cotton. Such as BT cotton. Here, BT cotton. Now what exactly happens here? This toxin is there, no, BT toxin. This BT toxin will not affect us. Why? Because our stomach is acidic in nature. At the same time, the larvae's gut pH is basic in nature, alkaline pH. And this BT toxin under alkaline pH will kill the larvae and not affect us. Awesome, no? Awesome. Students, don't worry, more about this BT cotton you will learn in the biotech chapters, cryogens and everything later more there. But remember, this is a biocontrol agent. Okay, we can use fungus also. There is a fungus called as trichoderma. This fungus will be growing in a symbiotic relationship with the tree. And this fungus, trichoderma, will kill all the plant pathogens in the root. So it will keep the plant safe and happy. Okay, trichoderma, it will prevent plant pathogens. Okay, next is viruses. There is a virus called as baculovirus. Baculovirus belongs to a genus. It belongs to the genus called as nucleopolyhedrovirus. It is called as nucleopolyhedrovirus genus. This baculovirus will kill only and only arthropods and insects. And at the same time, this baculovirus will not kill vertebrates, invertebrates and plants. So very amazing, no? It is very species specific. It is very species specific. It will kill only certain species. So very beneficial. And this property is very important. This property is the most important property here. Okay. So integrated pest management, economic approach to pest management, involves multiple approaches. Baculovirus as a desirable factor of PM. Why desirable? It is desirable because it is very species specific. It is not killing anyone else. It is only and only killing that very specific insect. That is all. Okay. Very specific. Okay. And students, let me tell you one more thing. Randomly, anyone can't do this. Not possible. If a person has to do IPM, if a person has to do integral pest management, he or she needs to know the entire food web, the entire interactions among different animals, the dosage. So everything needs to be known. Then only a person is qualified to do IPM. Not everyone can't do it. Okay. See, knowledge of pest, their life cycle, their interactions with environment help in developing a holistic approach in managing the pest. Okay. Other examples are given here. Ladybugs will kill the aphids. Dragonfly will kill the mosquito. All different examples. Okay. So please remember these examples for the next time. Okay. Do you want me to take the class till 1 AM? We will do it. Students, do you know what is in part 3? Do you know chapters in day 3, students? Do you have an idea about the day 3 chapters? Day 3 chapters. Any idea? Any idea about day 3 chapters? In day 3 we'll be doing genetics. And in genetics, molecular base of inheritance will take 4 hours. Principles of inheritance will take 3 hours. So 4, 5, 6, 7 to 8 hours. You'll go to 8 hours, almost 8 hours. So if we start the class at 5 PM, 5 PM to 8 hours, you do, students, tell me. 5 PM to 8 hours, what time does it get over? I am from South India only, right? I am from Bangalore. Tell me how many hours? It will go until 1 AM, midnight, above the midnight will go. Okay, so that will come up. And let me tell you, students, genetics will only happen if you are serious again. We do not want to stand here for 8 hours combined together and teach if you are not serious. And we will know your seriousness. We will know your seriousness by only if you are ready for it, if you give the same respect which we give it to you, the same love which we show to you by liking the video, because it is all free content, right? It's all free content. Commenting on today's video, telling that, yes sir, your hard work is appreciated. We understood the concepts, right? Do you see this PPT today? This is the PPT which I made today. This PPT requires, and let me tell you, I have spent around 4 to 5 hours. 4 to 5 hours to make this PPT today. This PPT today. 4 to 5 hours I've spent on it. I may be teaching the entire thing in 2 hours, but it took me 4 to 5 hours to make this entire PPT. Finding all the images, making all the boxes, sewage treatment plant one by one, finding all these images on the internet, making all this entire, all these decorations. Because when I make such things, it becomes more effective in learning also, right? So all this takes time. Now we go to biofertilizer. Last point, listen to me very carefully. Okay. In biofertilizers, microbes that enhance the soil quality. They are basically making the soil better and better. How do they do it? Students? How many of you seen this in real life? How many of you know what this is? This right here is called as nodules. These nodules are found in the leguminous plants. They are found in the leguminous plants. Okay. And these are found where? In the rhizobium. This rhizobium forms the nodules in the leguminous plants, and with the help of these nodules, the plant is able to get more and more nitrogen. This rhizobium will fix the atmospheric nitrogen into the soil nitrogen. Rhizobium, remember students, rhizobium is in a symbiotic relationship, symbiotic. But at the same time, Azotobacter is free living. Very important difference. They can ask you a question on this. Azotobacter is free living. Rhizobium is nodule formation. Azotobacter. Both of them will do what? Take the atmospheric nitrogen, pull it down, make into organic nitrogen. Okay? Organic nitrogen. Now what is this? What is this structure? What is this? Does anyone know what is this? This right here is normal roots with mycorrhizae in it. Mycorrhizae. Okay. The next one is called as fungi as biofertilizer. Okay. So mycorrhizae, genus Glomus. Now this genus mycorrhizae will form symbiotic association with the roots of higher plants. And when they become symbiotic association, students, they will help in absorption of phosphorus more. Very important line, very important. They help in absorption of phosphorus. At the same time, fungus gets a shelter, but the plants gets a lot of importance also. So biofertilizer. The other example is Anabaena and nostoc. They help in atmospheric nitrogen fixation. Again, cyanobacteria. Even cyanobacteria you might have seen, green color fields. Cyanobacterium fatty field adds biomass to the soil also. And all of these are commercially available as in the small tins. Like can you see rhizobium? In like small packets they are available, packets. Rhizobium, Azotobacter, nostoc, Anabaena, all that are available in small packets for everyone to buy. So if you have a garden in your house, try buying them instead of fertilizers. If you have a garden, if you are like Yeshu, if you have a field, try buying them. Okay? Try buying them. Now students, if you want to remember this entire chapter, like how do you remember this entire chapter, students? How can I remember this entire chapter? How can I remember the entire chapter, students? Here is a flow chart. Every single thing: curd, lactobacillus; bread, Saccharomyces; Swiss cheese, Propionibacterium shermani; biofertilizer, we just now learned biofertilizers; bacteria, symbiotic, rhizobium; Azotobacter, free living; fungi, mycorrhiza, glomus; cyanobacteria, symbiotic, Anabaena and azolla; free living, nostoc, Oscillatoria; microbes in industries, Saccharomyces cerevisiae, brewer's industry; antibiotics, Penicillium notatum; organic acids, citric acid, Azotobacter, citric acid, Aspergillus niger; acetic acid, Azotobacter; butyric acid, Clostridium; lactobacillus lactic acid; lipase, pectinase, proteases; septokines, clotbuster; statins, Monacis purpurus, blood cholesterol lowering agent. Cyclosporin A, that is basically your Trichoderma polysporum, used as immunosuppressive agent. Primary treatment, secondary treatment. Microbial, biogas production, biocontrol agents, Bt cotton, baculovirus, right? Everything, entire chapter is right here on the board for all of you. So if you download the notes, it will be available to you. If you download the notes, it will be available to you here. Okay. Genetics is going to be next. Genetics is, let's do next week. Let's do it after New Year. You know what, students? I have decided right now. The first class on the channel for the new year, let's do genetics. New year, new start. Let's start genetics for the next year. We will do genetics next year. Okay, we will do genetics next year. Let's see who can solve this question. Small question to all of you. Bacterial infections by Bacillus thuringiensis can kill the only caterpillar larvae? True. I told you already. Only larvae it can kill? True. Bt cannot kill the adult moth? True. I told you, it cannot kill the adult moth, only the larvae. Okay, only the larvae. Next year, new chapter, next class. So obviously option number 1. Both are correct. Aphids and mosquitoes are natural pests. Correct. Ladybirds and dragonflies are natural pest controllers. True. Very easy. NEET 2024 student. NEET 2024 question. 24 question. Which of the following can be used as biocontrol agents in treatment of plant diseases? I told you, Trichoderma, a fungi. In plant diseases, Trichoderma. A biocontrol agent is part of IPM. IPM should be. What is the main requirement? Main requirement is, I told you, species specific and inactive and non-target organisms. This is the most important, and the best example is baculovirus. Among the following pair of microbes, which pair has both microbes that can be used as biofertilizer? Biofertilizer is cyanobacteria. Cyanobacteria and Rhizobium. An organism used as biofertilizer for raising soya bean crop. I'll give you a small clue here. The small clue here is soya bean is a legume. Tell me the answer now. It is a legume. So the answer is going to be Rhizobium. Leguminous plant, Rhizobium. Yes. Next question. Leguminous plant are nitrogen fixers? True. Leguminous plant have Rhizobium in root nodules? Also true. This right here is a AIMS question. Answer is obviously A. And with that, the chapter is done. Students. Before I go, let me tell you a small good news. I think Gopika ma'am also already mentioned that many of our paid batches, and let me tell you, more than paid batches, what I want everyone to buy right now is the PYQ books. This is going to be very important. Right now you solved some PYQ books, right? Right now you told this is our half the price of the NEET Express, which is already going on. We have already finished 3-4 chapters there, NEET Express. This is going to be very important to you, student. This is basically the all-in test series. This is the most important at 764. I don't know what random number, I guess, but this is the ATS. And let me tell you, students, I have personally worked on this. And I have made beautiful questions for all of you. So try attempting this. And when I make questions, they are difficult questions. Okay. So this is the all-in test series. 22 online tests, student. 22 online tests. 16 full and 6 part tests. Okay. And PYQ book, again, very low price of 900 rupees. A PYQ book. So unlimited PYQ books. Basically, you will get chemistry, physics, biology. 3 separate books. Can you believe? 3 separate books of PYQ for 899. So it cannot get more lower than this. Okay. So that is the end of today's lecture. If you go to the description of today's video, you will get a link where you can attempt the quiz for this microbes also. So try attempting solving more questions, and that's it for today on my end. Thank you so much for today, students, for attending today's class. Let me know in the comment section how was today's lecture. Just type day 2 done. Try typing day 2 done and dusted. Day 2 done and dusted, and also you have achieved a new rank. Probably you are a pro subscriber now. Okay, so with that, students, thank you so much for today's class. I enjoyed, I hope all of you also enjoyed. Let me know in the comment section about today's class. I hope Gopika ma'am also wants to see the comments. So bye bye, take care, have a great night ahead. Bye bye, students.